looking at HIV, you could look at it and study it as the entire genome. However, this is quite cumbersome. So sometimes it's quite critical to look at it by specifying or focusing on specific genes. For example, the envelope gene, which is quite critical for viral entry into the cells and the body. So here we look at how we generate envelope pseudoviruses. You start with a blood sample. Using specific technology and editing bioinformatics tools, you end up with the envelope sort of amplicon and genome. You then insert this in a viral vector and then do co-transfection. And then you into a backbone and you end up with the pseudoviruses as highlighted. As highlighted down there at the bottom, you see those little pseudoviruses. So it's not quite easy to build this capacity. However, over the past 5 years, we are able to do this at the Uganda Virus Research Institute. And you may wonder, so what? The key role here is when you have pseudoviruses, you can then do the downstream experiments. For example, you can then determine that these envelopes, what receptors on the cell do they use? We know what subtype B and C are using because of the research that has been done in the Western world. Is it the same case in Uganda? So we were able to characterize our viruses here, and we know the co-receptors they're using, and this is quite informative for therapeutic interventions. How do they come in and block so that viral entry is prevented? Additionally, you're aware that we have these new broadly neutralizing antibodies that have been isolated and have the potential to neutralize several antigens, meaning several viruses of HIV, different viruses of HIV. We also have the opportunity to test our virus strains against these broadly neutralizing antibodies to see if they can be neutralized. And this will inform the manufacturers to see if Uganda actually needs a particular prototype or we have one standard. So we are contributing to research that is actually relevant in our environment. We have also, in addition to the envelope, which is a critical gene for viral entry, we have also been able to look at the GAG and the GAG pole. These are structured genes, but they may also be relevant in informing about properties or characteristics of the virus that are actually important for consideration during vaccine design, as well as disease progression. So we were able to amplify the GAG pole, and then using the NL4.3 backbone, we were able to insert our GAG and GAG pole into the NL4.3 backbone. And then we perform downstream characterization assays using cell lines, commercially available cell lines. Specifically, we focused on replication capacity of the GAG poles. How does this gene impact on replication? And how does it relate to disease progression? These are all critical in understanding which of the viruses may be more detrimental to an individual during disease progression of the HIV or AIDS. We have also moved further, advanced from specific gene target studies to full infectious molecular clones. Here it means you're looking at the entire genome, which is quite informative because then you get to compare your work, the full genome with the specific. Are there variants, and can you come up with certain hypotheses for why there are differences? So with the full genome, we specifically used a totally different strategy because it was a bit challenging to come up with the full infectious molecular clones. So here we use a strategy referred to as chemical synthesis, whereby you identify the genome of the virus you're working in, you share this genome with a commercial company that then chemically synthesizes the entire genome of HIV from the first nucleotide to the last. They send it back to you in 3 pieces because of biosecurity. And then when you receive it at the lab, you come up with primers, and then you amplify the different sections and cause overlap and end up with the circular, I'm sorry, the pointer is not working, the circular full plasmid of the HIV at the bottom. The importance of this is additionally, you can label with luciferase or GFP and then use this in viral inhibition assay. The overall goal here with your viral inhibition assays is to identify key epitopes of HIV that can then be included for vaccine design. And it's very relevant here because working with our viruses, then you're able to identify those key epitopes that are relevant to Ugandans based on our prevalent subtypes. This work is still ongoing. As you're all aware, we still don't have a vaccine, but we do know more. And hopefully we are almost there. And we have the capacity, I'm happy to report, we have the capacity to participate and have our data compared on the international scene and contribute to new immunogens under design. Yeah. I will now switch to COVID-19 vaccine design in Uganda. I'm happy to report that the UVRI and MRC have had an opportunity, and specifically the novel adeno COVID-19 vaccine is a collaboration between Uganda Virus Research Institute, MRC, UVRI, London School of Hygiene and Tropical Medicine, as well as Makerere University College of Veterinary Institute. The key objectives for the COVID vaccine study were to develop a Ugandan non-human primate adenovirus vector. Why would we be making a vector when they are commercially available vectors? One, as Brenda highlighted yesterday, it was quite challenging to access these adeno vectors outside existing patents. So this was another challenge. Secondly, there was the issue of pre-existing immunity for those that were readily available on the market. Ugandans, through previous studies, had been shown to have pre-existing immunity like to the AD5 that is already commercially available, meaning even before you make your vaccine, our population has the ability to neutralize the carrier or vector that is to help you deliver the vaccine in the body. So that was not an option for us. We then looked at our capacity and realized with the training we had received under HIV, we could embark on coming up with our own adenovector backbone that could belong to Uganda and is easily available. The second objective was then to generate SARS-CoV-2 vaccine using the strains that were prevalent in our environment here, and then capacity building for future epidemic responses. Before I get into the progress we have made, some people may think that COVID vaccines are not relevant anymore. However, if you look at that map, specifically, you can see Africa is light green. That is based on June 21st, 2022. It's an indication that in Africa, only 24% of the population has received the first dose and only 18% are fully vaccinated. Therefore, compared to the world, we are still not doing okay. And in Uganda specifically, we are at 34% vaccinated and only 23% fully vaccinated. And as you're all aware, these are viruses and they are changing and changing pretty fast. Recently, the genotypic report highlighted that we did have a recombinant between Omicron and the Delta. It seems harmless, but we cannot be sure how all this will progress. So it's always better to come up with ideas and ensure that the population is fully vaccinated. So to address the objectives I previously highlighted, we collected a total of 108 chimpanzee fecal samples from the sanctuary and the wild, and we got relevant approvals. We then conducted sequencing using next generation sequencing technology, and we were able to come up with a range of viruses. We focused on the samples that gave us a signal for adenoviruses, and there were about 9. From the 9 samples, we were able to use, again, next generation sequencing, specifically the Oxford Nanopore, and we generated the entire genome of the adenoviruses. We compared our adenoviruses with those that are actually in the environment. Those highlighted in blue are our samples from Uganda. And as you can see, they are significantly different from what exists in the public database. So we picked 1 or 2 to begin with, because this means these are unique viruses and the backbone we end up with is different and is patentable. So we then proceeded to the cloning strategy. The cloning strategy here, we procured a commercially available PAC19 vector. It's an open system. However, you need to customize it so that it's able to receive your adenoviruses here. So we amplified small sections of the LF1, LF2, LF3, and inserted them into the vector. That way, during the homologous recombination approaches, where we are inserting the full genome of adenovirus, we are then able to insert it into this commercially procured PAC vector, the carrier of the whole genome into one set. I'm sorry, the pointer is not working. No, yeah, it's fine. So we now generated at the bottom on the right-hand side, we have our adenovector down there at the bottom that has the E1 gene deleted. This is the one responsible for replication. The overall goal here is to ensure when we make our vaccine, we don't want it to continue replicating in the body. So you knock out this gene and you're sure replication will not happen. We then proceeded and amplified the spike gene from the COVID strains. And then again, using homologous recombination, we inserted it in the vector. And I'm happy to report we have several vaccine candidates. 1, 2, and 3 are actually ours inserted in our viral vector generated from our adenoviruses. And then we got the CAF2, which is already characterized, and we also inserted. So this is like a positive control that will help us benchmark what we have made here. I'm happy to report for the third objective, we have also included 2 PhD fellows, Christian Dekize, they are all registered at Makerere University, and Dennis Omara. So Christian will be involved with characterization of the chimpanzee adenoviruses and determining the level of their neutralizing antibodies in the Ugandan population. That is pre-existing immunity of our backbone, how relevant is it? And then Dennis will look at immunogenicity and efficacy of our adenovirus vector vaccine. I'm happy to report that we are already in the preclinical. We've set up the groups and we are now beginning to test our products in mice. Our next steps will be to complete the preclinical studies and get this information. If it works well, we'll then plan to progress to the clinical studies. And then it's quite critical, at least for the backbone generated, if we don't have pre-existing immunity as Ugandans, to proceed to patenting this. It's an open system and can be used beyond COVID-19 vaccine in animal health and other priority diseases. For HIV vaccine, as I highlighted, we still are doing the basic science research. At the moment, we are looking into viral diversity of the contemporary viruses and effects on recombination for vaccine design, specifically the broadly neutralizing antibodies. Thank you all for listening. And I would like to thank the funders of this study, Ayavi, USAID, as well as the Uganda government for the COVID-19 funding we've received. It's 100% funded by the government of Uganda. Thank you all for listening.
Thank you very much, Sheila, for that interesting and exciting presentation. Next, we have the next speaker, a colleague and a friend who has always been following me. Followed me at the medical school, followed me at the Zambia hospital, but now she's the executive director of the Joint Clinical Research Center. Dr. Sise Chitio Mutuluza has MBCHB, a master's and a PhD, and a fellow of the Uganda National Academy of Sciences. She's a medical doctor, public health specialist, seasoned researcher with over 28 years experience in conducting and coordinating AIDS research and care for HIV patients. She has a master's degree from Johns Hopkins University School of Public Health, USA, and a PhD from the University of Amsterdam, Netherlands. She is currently the executive director of the Joint Clinical Research Center. She's truncated, but a long history and a good, very, very strong CV, has published quite widely, as I've mentioned, and she's now going to present to us on the opportunities in gene therapy in Uganda. Dr. Sise.
Thank you, full professor, and my senior and mentor. Today, I will talk about opportunities for gene therapy in Uganda. And Sheila has set the stage. I will not explain gene therapy in my presentation. I only have 20 minutes. But really, the basis is gene editing, to replace, to silence, or to introduce genes for purposes of cure of different diseases. So gene therapy is the next generation of medicine. So that is the one opportunity already there. And while the rest of the world is moving ahead, if we don't join them, that will be our problem. So the opportunity is there, so we take it. And cure prospects using gene therapy presents a very promising new paradigm for management of HIV and sickle cell disease. And I focus on these, but gene therapy can be used for many other diseases. First of all, basically it is a one-time treatment to cure incurable diseases. It is potent, safe, and provides quantity and quality of life where it is applied. So the gene therapy, gene and cell therapy is booming around the world. You can see on the left-hand side, those are the numbers of studies that are taking place in the world. And that graph shows the number of applications that have been made to FDA in the US. It is projected that about 10 to 20 new cell and gene therapy products will be coming onto the market by 2025. But also, importantly, there is a lot of money being spent, but there is also a lot of money to harness. So as a country, we should really take advantage of this. This is a sample of therapies that have been approved by FDA in the US. You can look at the price tags one time, but look at the price tags. And all of these are given once, and then you get cured of your disease. So this slide here shows the red or the crosses, the countries in the world where gene therapy is taking place. And on the right-hand side, you see the pie chart. It shows USA is the biggest contributor here. When you look at sub-Saharan Africa and Uganda, I don't know if you can see Uganda there, there is no gene therapy taking place in our region. So we've been excluded. So should we continue to lament? What should we do? This is a survey on the clinicaltrials.gov, and it shows the ongoing gene therapy trials. This was accessed latest November 2021. You can see Africa, one gene therapy trial. This trial has taken place, it has actually ended in South Africa, and this is for hemophilia. So you can see where we are. And this, I think, alerts us on what we should be doing as a country. I'll now focus on two diseases. I say gene therapy can be done and used to cure many different diseases, but I'll focus on two, HIV and sickle cell disease. As you can see in the blue circles and the numbers above, sub-Saharan Africa has the biggest problem of these two diseases in the whole world. For HIV, we have about 67% of 38 million patients of HIV in the world. So While we have the least technology, we have the biggest problem of diseases where trends are moving. So for HIV in particular, we have had 34 years of antiretroviral therapy, but HIV remains the leading cause of death. And still many people can't access antiretroviral drugs. We are seeing donor fatigue because our governments are not the ones putting in money to treat HIV. So if we had a cure, wouldn't it be wonderful to treat everybody who has HIV? And one of the products that has been licensed is called Zynteglo. Zynteglo is used to cure beta thalassemia. Beta thalassemia is closely related to sickle cell disease. They are both hereditary diseases. And below you can see the price tag. If you don't have 1.8 million, you don't get this product. So this slide shows the progress that has been made elsewhere in the world with regards to these two diseases. At least 7 clinical trials have been opened between 2014 and 2021, and 36 clinical trials have used some bit of gene therapy to treat HIV. And how many of these have been done in sub-Saharan Africa? 0. So it goes without saying that in Uganda, no gene therapy trial has been done. It is also 0. But when we hear news of cure, it sparks hope. Many of you may have heard about the Berlin patient, the first one, Timothy. Now there is a London patient. There is Victoria Gray, who has been cured from sickle cell disease, but there are others, and these have been cured through participating in clinical trials. You have recently heard of the New York patient, and the numbers are growing. So now we want to hear the Uganda patient. So why is gene therapy important for Uganda? When you look at HIV, we have a high prevalence. We have about 1.4 million people in Uganda with HIV disease. And we have antiretroviral drugs, but people get tired of taking these drugs. And this has led to development of resistance to these drugs, which is another problem. Again, as I mentioned, there is fatigue. There is donor fatigue. This donor is going to continue to provide drugs for HIV for Uganda for 30 more years. We don't know. Nobody knows. When you look at sickle cell disease, Uganda is the 5th highest prevalence of sickle cell disease in sub-Saharan Africa. We know that there are drugs like hydroxyurea, but very few people can actually access this. So I think our goal, given all these challenges, but they also present opportunities, is to move ahead and embrace gene therapy and cures for these two diseases as a starting point. These are some of the opportunities for gene therapy in Uganda, and I can't read that text, so I will face this way. There is a promising cure, and there is a lot of research going on. I've already shown you. We could be in Uganda a regional center for care and treatment, actually for cure, and that is what we are aiming for. And when we develop infrastructure here and do work here, we can get IP, but we can also generate money as a country. We know, I've already shown, it goes without saying, if I cure you of HIV, the quality and quantity of life will increase. And then we have an opportunity now to transfer technology. We don't have all the technology we need, but we can do the transfer. This is a slide that shows the roadmap to getting antiretroviral drugs in Uganda. We rolled out antiretroviral drugs for treatment in 2004. But the first drugs were available much earlier, and treatment was happening elsewhere. So with regards to gene therapy, should we wait for 17 years? or should we get proactively engaged so that we can shorten this timeline? So what now? What should we be doing? So gene therapy, because of these issues and non-involvement, we have established what we call the Global Gene Therapy Initiative Working Group, which was formed in 2020. And the goal is to enable access and implementation of gene therapy research in low and middle income countries. And specifically, we have selected 2 countries, the Uganda at the Joint Clinical Research Center, and then India. And the goal is to have the first trials for these 2 diseases by 2024. And the founders of this Global Gene Therapy Initiative, which we call GGTI, is that doctor who is based at the JCRC, and Professor Jennifer Adair, who is based at the Fred Hutch in the US. And we have built a network of, this slide is very slow. We have built a network of about 70 experts in various fields around the world, and we meet every week to discuss and see what the way forward is. We have community and patients at the center of the discussions, and all these, the experts, these are different groups, and these are the different organizations where the people in GGTI come from. So how are we doing this? We have selected 4 pillars, and we are working on these 4 pillars concurrently to make sure that gene therapy comes and we meet our goal by 2024. This is one of the capacities which is very key, to collect the first cells that you have to modify, genetically modify, to produce the product, and we already have that. This product was developed by an NGO. This is the product that we are going to use at the JCRC for clinical evaluation. And the other product for sickle cell disease was produced at the NIH. It has been licensed to Caring Cross. And I talk about these products and evaluation here because they are very reasonable costs, so we can afford them. We wouldn't be evaluating anything that we are not going to be able to access. We've already gotten some seed funding, and we are doing capacity building. Lois is one of the trainees. She's at Fred Hutch and learning how to manufacture gene therapy. But we have others that have clinical placements and fellowships in the US. Manufacturing is the most costly aspect of gene therapy. And we are not going with the gold standard because we cannot sustain it. So we are already designing new platforms, and also working on a mobile GMP facility within a container, so that we can look at sustainability and manufacturing in country. Community is very critical and is part of this program, because we have to engage and train the community. There are a lot of myths. We did the first vaccine trial for HIV in 1997, 99. And on the very day when we had to vaccinate, a politician went on radio and said we are guinea pigs and what, and some participants didn't turn up. So we have to educate the community about what we are doing so that they embrace what we are planning. And regulatory, now this is very key. And we don't have guidelines and necessary regulations, because this is the first time we are doing this. We've been talking to National Council of Science, we've been talking to the Biosafety Committee, we've talked to National Drug Authority, we've talked to the President, and WHO, they also has been engaged, both local and in Geneva. And WHO has just completed consultative process to develop guidelines on gene editing, especially for low and middle income countries. And these documents, they've already assigned a consultant to help us to incorporate gene therapy for health into the JERA bill. And in conclusion, gene editing tools are fast evolving and their applications are vast. So we need to domesticate governance and regulatory frameworks for research, clinical care, community engagement, infrastructure, and capacity building. Uganda will serve as a test case. That is the opportunity for implementing of the WHO governance framework for human genome editing. And JSTRC, in collaboration with experts within the GGTI and with support from government institutions, regulatory authorities, ethics, and biosafety committee, is pioneering the development of gene therapy products in country to address public health needs. And I have a short video. Can someone help me to play this?
My name is Brenda. I am a youth living with sickle cell disease. I am always in and out of hospital. But one thing I'm sure of is that there is someone always out there searching for a cure for HIV and sickle cell disease. And one of them is GGTI.
Global Gene Therapy Initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders to bring gene therapies to LMIC. Global Gene Therapy Initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders to bring gene therapies to LMIC. We're gonna fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We're gonna fly high with GGTI. I am a person living with HIV. He's a person living with sickle cell. Adherence and access to medication is a daily challenge we face in our lives. That's why we love the work you do. GGTI. We treasure the time you spend to save our lives. That's why we love the work you do. GGTI. We treasure the time you spend to save our lives. We gonna fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We gonna fly high with GGTI.
Majority of the people with HIV and sickle cell reside in low and middle income countries. The Global Gene Therapy Initiative Working Group will initially focus on establishing a sustainable pathway to implement cell and gene therapy at the Joint Clinical Research Center in Uganda and CMC Center in India. It's time to train, translate, and initiate clinical trials for sickle cell disease and HIV in Africa and Asia.
Global Gene Therapy Initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders to bring gene therapies to LMIC. We're gonna fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We're gonna fly high with GGTI.
Doctor, my question to you is, what is your message to us as community regarding the GGTI in our country?
GGTI has identified pillars which are challenges and gaps in the delivery of gene therapy in low and middle income countries. And it is working concurrently on these pillars to fast track gene therapy delivery and access. However, this is a long journey. And in the meantime, We strongly advocate for care for HIV and sickle cell with available best treatment so that these patients are alive and available when gene therapy cure comes.
Doctor, what kind of support do you need from us as community of people living with HIV, but also those that have sickle cell?
The support we need beyond even community is expertise in gene therapy, which we largely have in GGTI, but we need to build local capacity to lead the way. We need funding, absolutely. We are talking to the government of Uganda. We've gotten seed funding from Gates Foundation, and we are also talking to development partners. And then, very importantly, we need community support in advocacy, also for funding, and also information to the rest of the community, demystifying gene therapy, and then also participation in the trials when they are here. Thank you very much. And we are still lobbying government to help us with money, with gene therapy.
Thank you very much. As a colleague, I gave us some extra minutes. So thank you for that. So next, we are going to have now another presenter, and that will be Dr. Joseph Nkodio, who is going to present to us on biosecurity interventions for national development. He is the National Biosafety Officer, Minister of Health, and currently coordinates the Biosafety Biosecurity Program in Uganda. He holds a Master of Science in Microbiology and MPH from Mbarara University of Science and Technology. He has more than 10 years experience in the safe and secure handling of infectious biological threats in the country. He has received his formal training in biosafety, biosecurity from the National Defense University, USA, and the Sandia National Laboratories, New Mexico. He is certified by risk management and biosecurity by the International Federation of Biosafety Association of Canada. He's IATA certified, and he has also vast experience in outbreak investigations and response to emerging and re-emerging epidemics and other public health services. So it's my pleasure to welcome Joseph Nkodio to give his presentation.
Yeah, thank you, Professor. Joseph Nkodio, doctor in making. I'll take on the journey. Good morning, ladies and gentlemen. All protocol observed. Like he has mentioned, my biography, I will not repeat it, but I'm glad to be here to share with you what the National Coordination Office on Biosafety Biosecurity Program in the country has so far managed to support our different stakeholders. I'll take you back to the year 2015, when we were reminded of the IHR requirements that state that state parties develop, strengthen, and maintain their capacities to respond promptly and effectively to public health threats. And this came as a call when we had the Ebola outbreak in West Africa, where we had most of our continent citizens being wiped out, and it gave birth to the global health security agenda, which one of the call was to make sure we have our laboratory capacity developed, and under the laboratory capacity, we had the component of biosafety, biosecurity being measured for the good practices. Prior to the IHR, we had to launch as a country the support for implementation of the global health security agenda, and it became very clear that we needed to strengthen our biosafety, biosecurity action package, which was under the laboratory capacity building. I am bringing this very vividly because all of us are talking about biotechnology, biosafety, biosecurity, and we have labs that are dealing or handling these very interesting technologies that we are trying to buy into our country to be able to embrace the call for a safe and secure work environment. And our country, like I mentioned, when we embraced the global health security agenda, we had to do a baseline assessment. And what I'm projecting there is what our status was. And it was measuring two items in regards to biosafety, biosecurity. We had to take on the whole of government biosafety, biosecurity systems. And we found out that much as some MDAs or government agencies were practicing biosafety, biosecurity, but it was under, the score was limited capacity because some other ministries were actually in the slumberland. Then when it came to assessment on biosafety, biosecurity, training and practices, by that year, 2015, we had no capacity. And that was it. So we realized that I want you to measure, start measuring. After 7 years, what has happened? I don't want us to miss that slide. After the assessment, all of us know that all the technologies we are manipulating for the betterment of our country, in case anything went wrong, our home becomes Ministry of Health because we run there to seek for rescue. So in response to the assessment, Ministry of Health established a well-structured risk management program and created also the National Biosafety Biosecurity Coordination Office and put it under the Department of National Health, Laboratory and Diagnostic Services. And also we had to work with the different MDAs. As mentioned or as projected, we had Ministry of Agriculture, Ministry of Defense, Ministry of Water, Wildlife Authority, and the primary focus was on making sure the biosafety, biosecurity competences and compliances are achieved. Allow me share with you what transpired since then. We managed to have a national training curriculum and program for biosafety, biosecurity, together with our biosafety manual. And initially, this training focused on the laboratory workforce, since everybody thought biosafety, biosecurity should be kept in the lab. But in 2016, we had to expand the scope and reviewed our curriculum to embrace and created a standardized curriculum, which has three tracks, track 1, track 2, and track 3. Track 1, which is the basic track, we managed to train our support staff, the nurses, the medical doctors, and our maintenance engineers in the different institutions. Track 2 remained for our technical staff who are doing the real manipulation. Track 3 targeted our top and middle level managers, where most of us lie. And I would pose a question: how many of us have taken on biosafety, biosecurity training at a management level, so that you get to know what you need, what is your role, what do you play with to make sure biosafety, biosecurity is up to date in your different institutions. The glaring gap in implementation remains due to the limited knowledge and buy-in from the top management. And this is where decisions are taken. This is where allocation of resources for biosafety, biosecurity takes place. So far, the National Coordination Office has managed to have 67 certified professionals by IFBA. IFBA is the International Federation for Biosafety Association. We have a number of them stipulated. We have those who have been taken through biosafety, biosecurity. Then we have the people who have been certified under biosafety cabinets, and then waste management. We also have the pool of national trainers, and these are helping us to cascade biosafety, biosecurity training in the different institutions under the One Health concept. In 2017, we had to do business differently. After training people for those 2 years, we realized that training alone could not produce results. So we had to come up with a biosafety checklist using the ISO standard 15190, which by then was the only standard which was supporting safety. And when we did an audit, the score, as per that time, it was 33%. And when we conducted another audit in 2018, after a year, we got 45%. This year, with support from Dr. Cisse of GCRC, we managed to conduct another audit, and I want you to bear in mind that with her support, we have another result to share with you in this forum as we go on. Yes. Without wasting time, you can see that in 2017, we were on 33. 2018, we went to 45, but we had a decline in 2022 by 12%. And the reason is very simple. We are now doing things differently. We've embraced international standards. We have the ISO 15190 and the ISO 35001 that support biosafety, biosecurity management for all institutions and laboratories. So I call upon the members here to pick interest in this, because we are all doing a lot of work in biotechnology and we are doing them in the lab. But what is the status of those labs in regards to the ISO standards that we are supposed to embrace? I want to share with you another assessment. After the 2015 assessment by Global Health Security Agenda checklist, we had another assessment recently, that is last year, and this is what is the picture. I hope you kept in mind the score from the assessment of 2015, and this is what is happening in 2021, and this is published. We are still having developed capacity, but still we are scoring 3. 3 means we've not moved as per the international standard, and also in terms of biosafety, biosecurity, it shows that we've developed capacity, but the level of capacity does not allow us to move to higher scores, because some of our MDAs have not yet taken on or done their roles as the way they're supposed to do them. So, and we can't just score a Ministry of Health facilities alone, because this is a national scorecard that targets a One Health approach. Yes, after that score, we've now decided to make sure we review our biosafety manual and the national curriculum to cater for what would have been missed in the previous one, in the previous versions. And also we've gone ahead to create the national mentorship toolkit so that we create biosafety, biosecurity mentors to support our facilities so that we can improve the projected score. We've also gone ahead to develop the national healthcare waste management guidelines, which used not to be there because we already now have a policy, and under One Health, it will be reviewed to embrace the different MDAs to make sure biosafety, biosecurity is part of the different sections. Now, like I mentioned, we conducted an audit with support from Joint Clinical Research Center, and this is part of what is happening. We have what we call the acceptable, non-acceptable, and then risk levels. So where you see very high risk, that is poor. Where you have high risk, that is fair. Moderate, that is average. So if you saw the colors, that means Uganda, we are on average. Low risk, which is good, we are not yet there. And then very low risk, which is very good. That is our desire. The scores you see there were determined to have our checklist scoreable. The audit I mentioned, this is what has happened. We conducted an audit, safety audit from 52 facilities, and we don't have any under green. We don't have any under low risk. Rather, we have 3 under low risk. We have around 4 under moderate risk. And then high risk, we have 15. Very high risk contribute to the biggest number, which is 57%. The categories of facilities that we visited under the One Health. We had the animal labs and the human labs from national to health center 3. I want to interest you. These are part of our sections from the checklist. But look at specifically laboratory biosecurity. That's where I want us to focus. It is among the poorly scored sections, and it is 23.2%, meaning our biosecurity measures or mitigations are still staggering, and we need to improve. So I want to interest you in this checklist so that we can be able to have it across our facilities. Another performance shows that from the facilities we visited, you realize that we had both government and the private facilities. And because I am government myself, that is what I've managed to score. So I call upon the other different government agencies to do their part to make sure we can improve that score. Otherwise, if I hide my head in the sun and say we are doing so well, then I'll be compromising on the standards. So I call upon you to make sure we can improve that score in the next gathering so that we can work with our heads up. Otherwise, that is what is happening. But some of our private labs, which were only 4, they indicated a low risk. You might ask me why only 4 private labs, but the audit exercise targeted those identified only 4 because of the funding gap that we had. Yes, performance of facilities on establishment of occupational safety and health. It is very clear that 55% lacked an occupational health and safety program, which is a backbone for our biosafety and biosecurity, since we are all employed by those different MDAs. So we need to think about the occupational health and safety as we do our biotechnology studies. Yes, I want to share with you a way forward as I conclude. The results clearly indicate that we need urgent engagement with our different stakeholders to develop robust biorisk management mitigation strategies. And then we also think that deliberate efforts will proactively identify the potential biosafety, biosecurity risks so that we can be able to avert these identified hazards and threats. Management commitment plays a very big role when it comes to biosafety, biosecurity. So our top managers who are here, you are the biosafety officers of your institutions. So I call upon you to pick interest and make sure the decisions that you take in regards to biosafety, biosecurity are well located for. And then in that, we shall be able to promote a safe and secure work environment. I want to make some recommendations. We've been in existence for the last 7 or so many years, hearing about biosafety, biosecurity. But I want to say that we urgently need a legal framework to be able to design our strategies. Africa CDC is trying to come up with a regional legal framework on biosafety, biosecurity. And we always do benchmarking. So I call upon the different stakeholders here to make sure we can benchmark and have our national legal framework on biosafety, biosecurity, because that's the only way we can operationalize our biotechnology strategies, and then people will be able to embrace them. Training and certification programs. We need to have our professionals certified. So, and I don't know, you've had some universities are now creating training programs for bachelor's degree and master's in biosafety, biosecurity. And I think we have one in the region, that is Tanzania. They're already creating a program there, and they're calling people to go and study biosafety, biosecurity as a profession. The issue of limited funding, that one is on everybody's lips. But what is bringing that is we are doing a lot of siloed implementation. We need to have cohesiveness as partners so that we support the coordination office for biosafety, biosecurity, and be able to expedite our mandate. We have the pathogen economy approach, which is embracing biotechnology. And this is very key if we are to identify our stakeholders clearly, if we know we are going to produce vaccines, we are going to produce our very good organisms, we are going to produce our drugs, then we need to still take you back to the legal framework so that we can be able to do our science when we are well guided. We have national repositories, and I will not mention where they are because for the purpose of being a biosecurity threat. How are we, what is their mandate and how are we managing them? What is in lab X or in a repository X? Do we know what they have and its status? So I call upon the different stakeholders here to please build systems to make sure our repositories are well secured to avoid the talk emerging and re-emerging threats, because some of them might come from repositories. I won't feel comfortable if I don't recognize our people. I'll begin with the National Council for Science and Technology for this opportunity to share with you what Biosafety Biosecurity program does in the country. CDC that started funding Biosafety Biosecurity way back from I think around 1999. They were funding Biosafety. FAO, WHO, IDI, JCRC, NADEC in particular that we are partnering with, and then Ministry of Health, Ministry of Water and Environment, Wildlife, and then the USAID. Most of us here get grants from USAID, and they're all targeting some biosafety, biosecurity component. Thank you for listening to me.
Okay, thank you very much. We need to move on. We are behind time. The next is a colleague from Uganda Virus Research Institute, Dr. Jonathan Kayondo, who is a principal scientist and is heading our Department of Entomology. He's trained, he has a PhD in vector biology and genomics from the University of Notre Dame, USA, and he's doing a lot of research on vectors, bioinformatics, molecular genetics, and he's also leading our project on preparing for the genetically modified mosquitoes. So he's going to talk about GM mosquito for malaria control. Jonathan.
Yeah, thank you very much. All protocols observed, and allow me to remove my mask. Yeah. So I'm going to share our ongoing work at UVRI with regard to genetically modified mosquito preparedness. So what we've been doing up to now is laying the biosafety foundation, so that when the time comes to do work involving genetically modified mosquitoes, we have systems and we have facilities and we have the expertise and we have the SOPs that can support that kind of work. Probably you've had a lot of misinformation, miscommunications that GM mosquitoes are here. Maybe I can start right away. They are not. We haven't started that kind of work. We're still far away from that, but still we've been working very hard to make sure we have the right facilities, the right infrastructure, the right teams on the ground to carry out this kind of work. And so that's what I'm mostly going to share today. Also, I want to add that we've had a stall here exhibiting for the last 2 days, and it will continue tomorrow. And that's a good resource to go in case you want to know more about what's going on and where we are at. And we have information and communication materials there that you can feel free to pick, and they'll give you more, because I can't talk more, a lot of that in the 20 minutes that I have. So the structure of the talk will be simple. So I will talk about the malaria problem in Uganda. It will be a background, and then also the mission of Target Malaria in Uganda. And then I'll talk about the laying of the biosafety foundation or groundwork that we've been up to at the UVRI. And then I'll talk about the future plans involving the contained use studies that we'll do, and then the regulatory requirements that go along with that. So malaria remains a big challenge health-wise globally. and lots of people, actually millions, are afflicted worldwide. And most of the problem or the burden is borne most by Africa, especially children, the poor infants and expectant mothers. The economic toll is also enormous, like we know. And unfortunately, progress in the control has stalled, and we've seen rising cases since 2015. And so new tools are needed if we are talking about, eradicating it. The case for Uganda in particular is no better. I could just summarize that we're among the 5 most highly afflicted countries when it comes to malaria burden. So we still have a lot of work to do. Now we have interventions that are there. You've heard about nets, you've heard about anti-malarial drugs, you've also heard about probably insecticides, and those are good, and they have saved lots of lives, averting death and morbidity extremely, but they're not adequate by themselves to eliminate. And that's because they're grappling with a number of challenges. If you look at the insecticide, for example, there is resistance that emerges in the mosquito vectors, making them, of course, less effective. If you look at the drugs themselves, again, there's the fear of drug resistance coming, making those therapeutics also less effective. And if you think of things like nets, the mosquitoes are also giving a counter fight so that they evolve and they try to evade the net. They try to bite you when you're not sleeping under the net. So there's an ongoing battle with the humans and the vectors. And so that means the tools that we're throwing at them in our current toolkits are not sufficient to eliminate. So genetic modification can help us combat the disease in novel ways, and that's why it's of interest. The WHO encourages research and exploration of its potential. It's also on the novel technology tool list for APET, and APET is the AU's high-level panel on emerging technologies, and they recommend that genetic modification be seriously considered as a possibility when it comes to integrated vector management, and they think it could be part of a toolkit that complements other tools that are there if it's researched and developed. Actually, in 2017, the high-level panel at the AU and the NIPAD and the Council of Ministers and the Science and Technology there, they recommended that more countries and regions consider that kind of approach in their development and research agendas. And this vision has mostly been taken up by West Africa, where you have the WHO there, the West African Health Organization, WAHO, and also coming together with other players like ECOWAS, and then health regulators, environmental regulators, and ethics experts to kind of see how this could be developed and how it could be integrated in a multidisciplinary integrated vector management, control tool. So they're busy working, trying to look ahead, see how they could harness this kind of technology. So for target malaria, we are a group of experts or a network of, let's say, researchers comprised of more than 200 experts coming from 11 institutions based in Africa, Europe, and the US. And the Uganda Virus Research Institute is one of the collaborating partners in this consortium. So in the malaria endemic countries in Africa, we have teams working in Uganda and in Mali and in Cape Verde and in Burkina Faso and Ghana. And you can see a lot of the activities taking place in West Africa. Now, the mission for target malaria is to develop and share a new genetic-based vector tool that can complement other existing interventions in our fight against malaria. So the approach there is to achieve reduction of malaria transmitting mosquitoes, obviously through genetic modification. And the idea is that if we can reduce the numbers, there's that threshold where you reach that they're so low that you can break the transmission. So that's really the big idea behind this idea. But before you do that, you need to put in place the required infrastructure and the competence and the capacity to carry out that work effectively. And so that's the first phase that the project in Uganda has been carrying up to this point in time. And that's what I'm going to focus on during the rest of this talk. So our research depends on stakeholder engagement and also regulatory compliance. And we have dedicated teams that help facilitate that kind of work. So what capacity have we put up in place over the years? So we have a facility that's built to arthropod containment level 2 standard that we've established back in 2019. And up to now, we've been refining it, trying to make sure it's fit for purpose. So it was designed to comply with ACL2 features, and arthropod containment level 2 has specific features that are characterized by a number of things. Some of those include, for example, limited access to only authorized personnel, and then you have rigorous protocols that are implemented to ensure waste disposal, and you have mosquitoes themselves being reared in species appropriate containers, and these are being confined in rooms, so that gives a barrier between the mosquitoes and the environment. And then you have additional physical features. For example, the way you enter, you have to go through a set of doors which won't allow you to open unless one is first opened and closed before another one is opened. And then the ceiling and the plumbing for all the piping, and also the gaps that are there, it requires to have mosquito-proof netting or appropriate seal. So that's how it's structured, and that's the standard that's recommended by international bodies like the WHO and also the CDC for work that involves transgenic mosquitoes and those that are not, of course, infectious. And so that's how we built it. So once we built it, we needed to test for its functionality to work at ACL2 as required. Well, because we don't have genetically modified mosquitoes, we had to use a different strain. So we used the strain that we are calling the color variant. So these are natural mosquitoes that show variation naturally. So it's not because of genetic modification. It's like the way you see, if you have cattle, some can be black, some can be brown, and some can be white. So that's a natural variation. We call it a polymorphism. So the mosquitoes too, there are traits that you could key in, which have genetic bases and which we could use to create particular strains for particular purposes. So for this case, we use that natural polymorphism, which comprised of having a certain pigment at the dorsal side of the thorax and not having it, to distinguish between two strains that we used to try and rear and breed and test the functionality of our facility. Yeah, and we could of course sort and sex those strains based on their structures at the pupal stage. So it was not too difficult to do. So the objective for that was to test the capacity of the facility for physical practices and to operate under ACL2 standards. So how good is the containment, and can we implement and run ACL2 standard protocols? And also to test the team's ability to handle genetic crosses, not with GM, but of course with this naturally occurring polymorphism. Because if you are to maintain that trait in the colony, you need to be able to sort and do some crossing so that it doesn't disappear on you. So the team needed to be able to show that and be able to handle that over a number of generations. So in that picture, you see one of the traits where it has a pigment that's white on the thorax on the dorsal part of the thorax. That's one of the strain. And then the other one is the one that doesn't have that phenotype, and that's the alternative strain. So two strains that we can use with physical features, morphological features to check our containment. So I said it was creating naturally from local mosquitoes, and then we made the colony, and then we did crossing to maintain that colony, in the laboratory for a number of generations. And so we had to test a number of things. So once, if you do it well, there are certain expectations and certain assumptions that should be or should stay. For example, you should not mess up with the ratios of your pigmented and non-pigmented strain and the sex ratios involved if you do a certain cross in a certain way. So we did the back cross, which should give you a 1-to-1. And each time that's what was observed. And this was replicated over a number of generations, up to 9. So that was good. We also looked at the fecundity, let's call it egg laying or fertility of the 2 strains that we isolated, again, because they were the same mosquitoes that we derived them from. They shouldn't be really different from each other. And indeed, the pigmented and the non-pigmented all ended up having the same facility, even when isolated, and continued to be observed individually. So again, we were happy, and that shows that the crossing was done well. We also looked at development times to see how long it takes to move, for example, from egg to adult for each of the strains over many generations. And again, the result was there was no substantial difference between the two, and that's what those graphs are showing, meaning that the team did a good job isolating these 2 strains, and they're very much able in maintaining the strains and the traits in the colony as expected. And then we look at the environmental rearing controls. Because this is an insectary, you need to rear these mosquitoes carefully. Can we control the environment to specification that the SOPs call for? So there you just see some plots that show the humidity and the temperature are just one of the parameters that we monitored. And again, over time, we could see that in the facility, we're able to maintain the rearing conditions optimal to what the SOPs called for. And so we're happy that we are really very much in control and of the environment in our own facility. And then we also looked at the surroundings. So around the insectarium, what mosquitoes are there, just for us to have a baseline for, in case there was an escape, assuming our color variant escaped, what would they encounter? So we wanted to know what surrounds them. So we observed this through traps over a number of years, and we were able to compile, the distribution of the surrounding mosquitoes that are there. That's a very busy graph, but what it shows is that there's lots of Culex and others, but as far as the Anopheles goes, there were really hardly any Anopheles around our facility. So we know that in the immediate vicinity of our environment, there's really not a lot of Anopheles going on. So in conclusion, this exercise that we did to isolate and maintain a color variant mosquito strain and run it at ACL2, rearing conditions, proved to us that the team was successful. We had the SOPs that were required, and that the facility is really able to contain and operate, at that level. And it was important that we show this before we start to work on, genetically modified mosquitoes. So then what's planned in the future? Of course, in the future, we plan to start studies on genetically modified strains. And for those, I said the aim is to develop a tool that's modified, that will suppress the natural mosquito malaria transmitting mosquitoes. And to do the suppression, there are 2 strategies that we're envisaging that could be achieved. One way would be if we modify and target certain genes that will result in biasing the sex ratios of the progeny under the matings that happen. So if that happens, you're having more males at the expense of females. And over time, if that continues, you can see that if there are more male mosquitoes than female mosquitoes, it's the female mosquitoes that spread the disease, but also it's the females that account for the number in the next generation. They're the ones that directly are linked to how many mosquitoes are going to be there in the next generation. So over time, if they're becoming less and less because of that, a time comes when the population becomes low, and then the transmission can be curbed. The other flavor of the same would be we target genes that have to do with actual female fertility in the body of the mosquitoes. So if you deactivate those, again, you'll have a scenario where the females that are modified are not as fertile because they've been made less fertile. So they're producing fewer offspring. And again, the result is over time, if that trait can spread, then the numbers in the coming generation would be fewer. And so a time reaches where the malaria transmission can be curbed. So that's the big idea behind that kind of strategy, all right? So to do that, of course, there are other things that also needs to be in place. I said regulatory oversight is important. So we are already engaging and been seeking advice from the UNCSD. They're the competent body on these sort of things. And of course, we also have the institutional oversight, the local IBC, and then there's also NEMA. They also have, regulations regarding the environment, and all those will have to be cleared before, of course, we start our work on genetically modified mosquitoes. So that's just a snapshot of where we are at and what we have planned. And I think, yeah, that's the end of my talk. And I would like to acknowledge our funders, the Bill and Melinda Gates Foundation, the Open Philanthropy, but then also the government of Uganda, because a lot of salaries of our staff are paid by the government. So it's collaborative work. And then those are the logos for the other participating institutions. So thank you very much.
So thank you very much, Jonathan, for that. Colleagues, we are now going to listen to the presentation we missed in the morning from our colleague who is in Kenya. Is he connected online, ready to give us? So this is Dr. Mugira, from the CEO of the National Biosafety Authority of Kenya, who is going to present to us on the current regulatory approaches for gene editing and synthetic biology. Already? Okay.
Hello, good morning, colleagues. Here from Nairobi, Kenya. I would like to confirm that you can hear me from a location in Kampala. You can hear me? Thank you. I get from the chats that I'm on, and therefore, once again, good morning, colleagues from Nairobi, Kenya, and to you in Kampala, Uganda. We are delighted to be part of this engagement. And I will request that I put off my video to save on bandwidth. I have been having difficulties this morning. I do not know the reason, so that also I'm able to share my screen. Somebody can confirm that you can see my screen? Even by chat, our colleague at UNST. Yes, thank you very much, Dr. Wilberforce and the colleagues across there. Once again, it is a pleasure to join you. I want to say that I truly enjoyed the presentation by your minister at the opening of the sessions on Tuesday. And that was a great eye-opener to what is happening in Uganda in relation to positioning science, technology, and innovation for purposes of driving the socio-economic development of your country. And given that Uganda is part of the East Africa community, what happens in Uganda has a direct impact to what happens in Kenya, in Tanzania, in Rwanda, Burundi, and the rest of the EAC region. So I like to congratulate the people that are driving the science, technology, and innovation agenda in Uganda for the work that you're doing in positioning technology towards delivering service to the people of this region. Having said that, with all protocols observed, I recognize my colleagues from UNST in Uganda. I recall the presentation by your chairperson, Madam Teresa Singoba, whom we have worked with a long time, and therefore I send my salutation to her. We have been in this job of preparing a legal framework for biosafety together. So receive greetings, therefore, from your brothers in Kenya. And with that very simple opening remarks, I like to get into my presentation, in which I am privileged to share the regulatory framework for genome editing in Kenya. And again, as in the screen, my name is Roy Mugira. I recognize that Mugira is a name that is also in Uganda, but I guess Uganda pronounce it as Mujira, and in Rwanda as well. So this is an East African name. I am serving as the acting CEO of Kenya's National Biosafety Authority, the competent authority and the focal point for the Cartagena Protocol here in Kenya. I like to move my slides. Oh yes, good. As a background, genome editing proceeds in basically 2 or 3 approaches that either may result in an end product that is a classic GMO, or a product that is non-GMO. In certain other procedures, genome editing would start maybe with steps that are GM, and then the final result is not GM. So this is the background upon which the Kenyan regulatory system, the guidelines that I'm going to share, is based on. And we take note that this is what many other jurisdictions, many other countries have taken in terms of guiding their regulatory framework for genome editing. To just touch a little on the genome editing is a newer technology that is targeting the constitutive genomes of organisms without introducing a new gene component, a transgene. And therefore, this may not fit properly within the context of genetically modified organisms. And therefore, newer methods of regulation, newer approaches to regulation need to be developed. I am also glad that a colleague who presented a little earlier than before me has also shared the guideline that has been developed by FAO. I think it is in the final stages that is guiding countries on how to approach this. And I think that was a colleague from Global Gene Therapies Initiative. So the approach, therefore, is to recognize that products of genome editing could be either classic GMO, like what is presented in my slide on the extreme left, or that which is not GMO, that can end up in the same way as would have resulted from natural mutagenesis. So this is the approach, the background upon which the Kenya's regulatory framework, or rather the approach towards genome editing, has taken. So the objectives of the guidelines that we published a little earlier in the year is to address genome editing, given that there is no substantive law on genome editing. The guidelines are therefore designed to provide a technical guidance to applicants on the criteria for determining which genome editing organisms or their derived end products are regulated under the Biosafety Act. The Biosafety Act specifically regulates genetically modified organisms, their development, transit, handling, and use. So genome editing, therefore, having those features that I have indicated, may require a kind of special kind of guideline. The key features of Kenya's genome editing guidelines include: 1, an early consultation stage, where the applicant fills out a simple form providing information or data that facilitate decision making. So in essence, this is a very, very early stage where I would say we have this kind of genome editing process or procedure, we have this product, and therefore it has been developed in this manner. And very quickly, the authority is able to make a decision on whether the product will fall within the category that will be regulated under the Biosafety Act, or it falls outside that regulatory framework. The other feature is an expeditious timeline of about 30 days for decision making at the early consultation stage. And this has been made quite easy to facilitate a quick decision making. Our law allows us to take up to 150 days, between 90 and 150 days in making decisions. But in this particular one, we have allowed for about 30 days quick decision making, and the communication is made to the applicant on the kind of pathway their product or their project will follow. Number 3 is a clear flow of steps on consideration of the various options on the basis of information and data provided. We have presented this in a flowchart that I'm going to show in a short while. And finally, is categorization of genome editing outcomes expected to be regulated under the Biosafety Act. We have provided a table that says this category will follow the Biosafety Act. This other category will be regulated through other regulatory mechanisms. We take note that this is the same approach that has been taken by a number of other countries, including Nigeria, India, and other, Argentina, and a few others that have also developed this kind of guidelines. And the key feature is the early consultation. Now, we have made decisions on genome editing applications on the basis of the Biosafety Act under the contained use regulation, because these applications are all at research level. So not 11, sorry, 10 of these have been approved and are going on within Kenyan research institutions. Number 1, we have some work around the African swine fever vaccine. This is a genome editing project that is seeking to develop a vaccine for ASF. Then we have another project on goat for trypanosome resistance by the International Livestock Research Institute. Then we have a project on sorghum to develop striga resistance. Striga is a weed that affects sorghum production and also maize. And then also in sorghum, another project that targets anthracnose resistance. In yam, we have a project that is targeting vitamin A, the biofortification, and also disease resistance. In cassava, we have a project that is targeting nutrition enhancement. And in banana, a project that is targeting nano and colimoviruses and aphids resistance. And again, in cassava, a project that is targeting early flowering for purposes of enhancing breeding programs. And another in banana, where the target is fungal and bacterial disease resistance. And finally, potato for potato virus Y resistance. So we used the biosafety regulations under contained use to approve these projects. They are going on. We expect that they will come up with products that will require to go to the next stage of commercialization. And that is where an early consultation will be conducted to find out of this product that is good for trypanosome resistance. Is it within the classical GMO, or is it within the outside the GMO in the classical sense of genetically modified organisms? And then we shall advise the kind of regulatory framework that will follow. The scope of the guidelines is genome-edited plants, genome-edited animals, and genome-edited microorganisms. As is with the provision with the Cartagena Protocol, these guidelines are not applicable to genome-edited pharmaceuticals for human health use because these are regulated by the Pharmacy and Poisons Board. I recall the presentation by the person from the Global Genome Therapies Initiative that highlighted the potential use of gene technologies in addressing serious medical issues. So this guideline, therefore, may not apply to those because they are regulated under another regulatory provision within the Kenyan legal framework. This is the flow chart for the early consultation on genome editing. It has very simple and direct steps. To start with, the applicant will launch the early consultation form, and then it will be checked for completeness, the usual administrative checks. And the question is there integration of foreign DNA sequence? If the answer is no, then that particular product will be classified as a non-GM, and therefore it will be taken for regulatory systems that are outside the Biosafety Act. So after that, the applicant is notified that the technique or product will not be regulated under the Biosafety Act of 2009, and therefore this falls now within other regulatory systems. If the answer to the question is yes, that there is indeed foreign DNA sequences, then this is taken to be like a GMO, and it is followed by a further question. Is the foreign DNA sequence present in the end product? Like I said earlier, there is a possibility that the process of making the genome edited product could have intermediary steps that are GMO. But then what about the final product? Is it GM? If again the end product is as foreign DNA sequences, if the answer is yes, then it is categorized as a GMO, and notification is given so that the regulatory authorities, other agencies can take up the matter, and at the same time, then this is indicated to follow through the Biosafety Act. If the answer to that question is no, again, we go back and classify that as an non-GMO, and therefore it is regulated outside the Biosafety Act. So this is the review, within the table we are showing 15 days. That is supposed to be 30 days. We look through that quickly and we give an answer to the applicant so that the applicant would know what kind of regulatory pathway their product or their technique would follow. So there are two possible decisions on these applications or these early consultation. One, it may be determined by NBA that the genome edited organism or product has no foreign genetic material, and therefore the applicant is informed that this particular process or procedure or product is exempt from regulation as a GMO. Scenario two is a determination by NBA that the genome edited organism or product has foreign genetic material, and therefore the applicant is guided to make a full application that follows a full risk assessment and biosafety approvals in compliance with the Biosafety Act. Now we have made provisions for the review of guidelines. Remember, science is quickly transforming and changing and advancing, and therefore we have said these guidelines may be revised based on new scientific information. If we become aware of new scientific information that have impact on the approach that we have proposed, then we have made a provision for revising the guidelines. And also, the authority reserves the right to alter its decision if new scientific information previously unknown become available. So this is just to open it up for purposes of revision. And therefore, colleagues, that is what we are doing in Kenya. Thank you very much for the opportunity to share this with you. I look forward to the discussion and any other things that are coming through in this forum. Asanteni sana.
Asante sana. Thank you very much. I think that was very nice, and really to see what our neighbors are doing. It was a very good presentation. We have now, I know we are about an hour behind, but I think I was requested that the biosafety officer from the National Council will give us just a brief on the policy and regulatory matters in emerging biotechnologies. So if the biosafety officer is around, just to give us a brief, and then our discussion will be shortened. It will be very brief, Q&A, questions and answers, and then we go for break. So you can come and give your short remarks.
Thank you very much, Mr. Chairman. The Uganda National Council for Science and Technology, within its mandate, pursuant to the Uganda National Council for Science and Technology Act, has moved along to regulate aspects of modern biotechnology, taking into account the National Biotechnology and Biosafety Policy of 2008. We recognize that these emerging biotechnologies, such as synthetic biology, genome editing, gene drives, are actually expected to transform society, particularly in Africa, where we are expected to leapfrog. However, relevant policy development and regulation for these promising biotechnologies will certainly be a daunting task, as the risks presented will not be fully comprehended or understood until the technologies are further developed through research and development pathways. So what is our call for African governments? We need to adopt new governance models that seek to manage the dynamics of emerging biotechnology promises, taking into account the risk and also looking at broad constructive enabling governance systems that structure the evolving biotechnology developments in a stepwise manner towards product development. So our question would be, what would be the ideal regulatory model, looking at all those policy and regulatory considerations? We are looking at a science-based, structured, systematically, and data-driven approach. We are looking at starting by systematically identifying plausible pathways, potential harm, and other potential unintended consequences via problem formulation approach for investigative elements. We need to embrace a continuous evaluation and assessment of the social, environmental, legal, and ethical considerations for responsible development of these biotechnologies. I'll take a note on this by highlighting what was published regarding the Chinese scientist. Well, there was an element of CRISP babies, which actually was done clandestinely. So this is an element that we need to strengthen as Africa and also Uganda at large. But the good element is that we already have some established frameworks for risk assessment, such as the Cartagena Protocol, World Health Organization, and then FAO. These are resources that will help us move forward. Although at international level, the discussions are still ongoing regarding synthetic biology and also elements related to gene drives and the like. So you will note during the last Conference of Parties meeting, there was an indication to do a horizon scanning and a proposed group to study these deeply to ensure that there is a guidance framework at international level. Members are asking, should we have a separate protocol or we can work within the Cartagena Protocol? And then we need to think about incentives for widespread stakeholder cooperation to produce proactive, flexible governance to inform decisions around the delivery and sustainability of these technologies. So also the regulatory mechanism should be put in place for regular review and updating. At Uganda National Council for Science and Technology, we are working on guidelines for new breeding techniques, and we shall be calling stakeholders soon for input, but the policy proposals are related to issues to do with risk analysis and how we shall manage all those issues. So each technology should be handled on case-by-case basis because each has its unique characteristics. So we implore you to move with us when we call you upon as we deliberate on further policy proposals, but this will also call for reviewing of the attendant policies, especially the National Biotechnology and Biosafety Policy is actually not responsive to the needs of today, taking into account synthetic biology, and also the elements of biosecurity, a need for an extensive consultation related to the policy framework. So that's the current prepositions on policy and regulatory issues for emerging biotechnologies from the Council for Science and Technology perspective. Thank you very much, Chairperson.
Okay, thank you very much. This is the time if you have a question to any of the presenters. We are going to have allow a few of these. We have had, it was a very nice session because it took us through different areas, regulation in the labs, what's happening, the work in HIV, COVID, vaccines that we have seen, the work that is being planned in the gene therapy that is quite interesting, exciting as we're moving forward. Genetical preparations for the genetically modified mosquitoes, very interesting. So really we are on the move, and it's very good that National Council is putting us together really to regulate, to ensure that the work we do is moving on very well. We have heard what our neighbors in Kenya are doing, how they are approaching their policies and regulation. So if you have any questions, you'll come. I think we have the microphone. If we can have 2 or 3 so that we don't, one on the other side, there are people who are, yeah, on this side. And then you just mention your name and to whom you're addressing your question, and be brief. Yeah.
Thank you, Chairman. This is Dr. Chibuka from Synthetic Biology Africa Global Catastrophic Biological Risks Initiative. My questions and comments go to Mr. Nkodio. Mr. Nkodio, thank you so much for the presentation. One thing I wanted to mention or to ask, why didn't we have biosafety and biosecurity running together in this? Maybe even the organizers will have to ask, because these two are almost the same things. They complement each other. Then 2, I just want to inform members here that we have a project, Synthetic Biology Africa Global Catastrophic Biological Risks Initiative. We have about 4 objectives, but we just started with 2 objectives. One was on creating awareness about global catastrophic biological risks and advocating for legal frameworks. So just recently, 9th to 11th, we had our inaugural workshop where we had the members coming from the Eastern Africa, and it was, I think, a very successful workshop. We shall be engaging many of you here, and after this one, we will be moving to North Africa. So we are moving around the African continent. 3, Mr. Nkodio, please, there was a joint external evaluation some time back. Can you kindly tell us how far you have gone with the recommendations of that report? Thank you so much.
Okay, thank you. Let's have maybe 3, and then they will answer those, and then, yeah.
Thank you. Thank you, Chair. I'm Claude Chirumuhuzia, pharmacologist, ethicist. This morning, I read 2 articles related to gene editing. One was saying the good, the bad, and the ugly about gene editing. The other one was talking about the dark side of gene editing. The second and third presenter, they gave us beautiful presentations on the positive side, on the good. Can we have some glimpse of the bad and the ugly in relation to gene editing? Thank you.
Okay, thank you. Then we can have those 2. Yeah.
Okay. Thank you very much, Chair. My name is Josephine Naiga, and I just completed my MSc in bioethics. My question goes to Dr. Sise Chityo. Thank you for that enlightening presentation on the opportunities in gene therapy. My question is, how prepared are we as a country or regulatory boards to handle the huge ethical concerns arising from these emerging biotechnologies? Thank you very much.
Thank you. My name's Dr. Mpaji Joseph from Bustama University. I have two questions. The first one to Dr. Mujira from Kenya. They seem to have made progress on biosafety and biosecurity. I'm wondering whether measures have been taken to include aspects of biosecurity and biosafety in the training curricula of especially tertiary institutions. That the students as they graduate have a significant appreciation of that aspect. Secondly, to Joseph, my namesake, on biosafety and biosecurity, his presentations seem to have focused largely on containment or management of outbreaks. I'm wondering whether there also is an appreciation on the need to prevent the outbreaks, these outbreaks by sensitizing the communities, especially those communities in areas where there are risks for some of these outbreaks. Thank you.
Thank you very much. My name is Professor Damaina Kanjako from Makerere University, College of Health Sciences. Incidentally, my question is somewhat related to my neighbor, and asking what strategies have been taken or are going to be taken to involve this in training of the young scientists in high school and undergraduate education, even primary school, because these are the innovators of the next generation, and they need to start thinking about these things and how to innovate. If we are going to innovate and produce, these are the people we are looking at to bring this out. Thank you.
Okay, thank you very much. I think let's respond to those. Audio, I think there are a few. And the others also can chip in.
Thank you so much for these wonderful questions and the recommendations. I want to begin with the question on why we don't have biosafety, biosecurity together here. I think it is high time now to, I myself, to also push it to the National Council of Science and Technology, since we have the biosafety officer here and also the executive secretary in the house, to think ahead of time and review the National Biosafety Forum to have biosafety, biosecurity move under one umbrella, if it is agreeable at that level. Secondly, about JEE. Yes, JEE, the Joint External Evaluation. This is one of our tool that is checking our preparedness in regards to the Global Health Security Agenda Action Package, and specifically on biosafety, biosecurity. What the country has done is we've brought all the MDAs on board to make sure we take them through what the JEE checklist requires, and then they develop improvement projects to make sure we can score better in the subsequent assessments. So the work is in progress. Much as of today, we are still on score 3, which is a basic acceptable score. As a country, we want to be on 5 and above, but currently that is what is hindering us. But if you came and you only measured Ministry of Health facilities, then the score would be different. But we said under national laboratory services, and specifically biosafety, biosecurity, we take on all labs in the different MDAs so that we can move together as a whole. Regarding the prevention strategies, thank you, my namesake. Yeah, I take this as a commendation, and I will engage my team to make sure we can now widen our scope in identifying our stakeholders in regards to biosafety, biosecurity, so that we can be able to include that strategy. Yes, the last, I didn't get her name, but this happened, training of young science in biosafety, biosecurity. I hope that's what you mean. We way back in 2015, when we embraced the global health security agenda, one associate professor, Majalija from Makerere University, pledged that when I go back, I'm going to change the college and to what it is today. Cover B means biosecurity. So Makerere University is already embracing modules on biosafety, biosecurity, and they're training young scientists. But we want to encourage, I had another government university here, Sitema, and the other private universities, to review your training curriculum to include modules on biosafety, biosecurity, the way you did with quality assurance. I thank you.
Okay. Then we shall have Dr. Mujira also.
Thank you for the questions directed to me. One of them was the bad, the ugly, the good of gene editing. So the title, what I was asked to talk about today in 20 minutes, which was not even sufficient, was the opportunities for gene therapy in Uganda. But certainly anything in the world has the good and the bad. And we need to look, the same with gene therapy. There are risks, and we have to look at how to mitigate this. And when we look at things that we are adopting, even technology, even when we look at ethics, when we are reviewing protocols for this biotechnology, we have to look, we always balance what is the benefit versus the harm, the harm in what we are doing. And if there are risks or harm, how do we mitigate them? So the risks will always be there. But just to mention a few, there's an ongoing study in the US for gene therapy for sickle cell disease by Bluebird. And this study was halted by FDA. Why? Because 2 of the participants developed blood cancer. And they went to investigate from samples taken even before those participants got into the studies. And they have concluded that these cancers were not due to the gene therapy that was given. So this study has been opened. But these are things and myths, and that's why we need to engage community and tell them, while this may be very good, these are some of the possible risks, like we see for COVID vaccines. The other one that could go wrong, when you edit, you can edit wrong. And when we edit outside of the body or in the body, which is called in vivo. You can edit the wrong area, and you can insert a gene that may stimulate other things. All these things, I think we all need to look at them. The other question was about readiness for these kinds of technology, in particular regulatory. I think we are not ready, but we have the executive director of National Council here. And for us, when we are planning our clinical trials, we found that, one, we didn't have policies, we didn't have guidelines in the country to address these technologies. And we've been talking to all the regulatory bodies, and we have been doing also information giving and education. But I think these bodies, they are taking this on head on to put in place guidelines so that we can do what we have to do as researchers. But yes, we are talking to all of them. But they're also here, they can give their comments. Thank you.
Okay, Dr. Mujira.
And I forgot, we have a brochure, again, to educate regulatory bodies and the community. They are out at the National Council of Science and Technology booth. Please pick them. We also have an online one so that we educate ourselves, all of us, as we look for the good, the bad, and the ugly.
Thank you. Thank you very much, Prof. Facilitator. I assume that I'm the one seated on the seat that is vacant next to the colleague who has just spoken. So in Kiswahili, we say, hiyo ni kiti yangu. So I'm seated there, we assume that, but joining you through this technology. Thank you once again, Prof. Facilitator. there were two questions. Number one, related to one another, measures for biosecurity in curricula for higher learning institutions. And I want to admit, yes, indeed, the Kenya's National Commission for Science, Technology, and Innovation is the national focal point for the Biological Weapons Convention in Kenya, and is therefore the focal point. And NACOSTI, as is said in short, is partnered with a project and Denmark, and together with Kenya's Commission for University Education, rolled out a training program for academia, both graduate students and faculty in Kenyan universities outside the formal curriculum. So this has not been infused into the formal curriculum, but there is a robust training program that is going on targeting academia, particularly postgraduate students and faculty. Number two, we also carry out regular inspection of research facilities and continuous training of faculty to build human resource capacity in biosafety and biosecurity. Going to the lower schools, we are rolling out a reviewed curriculum in Kenya that is targeting to introduce competency-based kind of approaches to curricula. And within this curriculum, there is a reasonable focus on discovery, creativity, collaboration, and cooperation in problem solving. So we are introducing at the very early stage the aspects of discovery, of wonder, of innovation, of creativity in young children at the very early stages in their education programs. Finally, Chair, I like to mention a little about the good, the bad, and the ugly, riding very closely on what our colleague from the Global Initiative has put through. I want to indicate that any technology has potential for abuse, and that is why you will be comfortable if you found a military officer in uniform or a police officer in uniform carrying a gun, for example, the AK-47. You won't be worried about that. But if you found some other ragtag people in different kind of clothing and a different kind of demeanor carrying a gun, you will run away. And it is the same gun. So it is the same technology in the hands of the wrong people. There is therefore potential for all this technology, genome editing and others, to land in the wrong hands and be used for the wrong intentions. And that is why the global community negotiated the Cartagena Protocol, particularly for GMOs, and also the Biological Weapons Convention, a convention that prohibits the stockpiling of biological and chemical agents. A good example is when the mobile phones were just introduced into our region, there were fears and talk that they will cause ear cancer, they will cause all manner of things. In fact, we went to buy some small gadgets that we were putting on the phone to prevent ourselves from cancer and so on. So any technology has that potential, and that is why genome editing and GMO technologies are highly regulated. It is possible, indeed, for a rogue person to enhance the virulence of a pathogen using genome editing. But again, focusing on the areas that have been presented by our colleague from the Global Initiative, they are very big and huge benefits. Thank you, Chair.
Okay, thank you very much. I think I'm told we are very much behind. I will request that those who have additional questions, you can meet the presenters during coffee time, interact, so that we can exchange more and ask more questions. For me, my final remarks, it's very good, this Biosafety Forum, Biosafety regulations that are coming, but I keep telling my colleagues at UVRI, let's hope, think, because we have had biosafeties for a long time, even before gene therapy and the rest. Let me hope that in your institutions, as you're talking, we don't only narrow our biosafety on genes, gene therapy, and modifications. Let me hope you are still strengthening our other safety in the labs beyond biosafety, because some of these discussions sometimes focus on genes and where we're going. It's interesting, exciting. Biosafety in the lab and biosafety is wider. And I hope National Council and the rest take this very much into consideration. Our tradition of biosafety in the labs is not forgotten, it's strengthened, and we don't concentrate on the genes and gene therapy as we're moving forward. So that's my plea. Thank you very much. Let's put our hands together to thank our presenters for the good and always. So go and have a good, enjoyed break, and we shall be back after. Let me leave the organizers to announce the next items on the agenda. Thank you very much.
Good morning once again. Thank you, Mr. Chair, Professor Chair, for steering us, for steering this session very well and helping us to catch up on time after the late start. And thank you, presenters, for those very informative presentations. The discussions still continue. I think we've formed a network for this forum. We'll ensure that we continue the exchange. We'll go for a short break of 20 minutes. We've lost a lot of time. Actually, one of the presenters in the next session on public engagement from Nigeria has been online and requesting that we give him an opportunity, but it wasn't possible to get him into this program because they were talking about something completely different. So we request that we have a 20 minutes break. The break tea will be at the exhibition area, and we'll be back at 12:04, and we'll try to catch up in the next session. I'll give a brief introduction of our next chair. The person chairing the Biosafety Communication and Public Engagement session is not Professor Karembu, but we have Mr. Josie Mohanji, a certified PR practitioner who is currently working with Uganda National Council for Science and Technology. Josie has previously worked with the Uganda Wildlife Authority as a PR manager. He has also worked as communications officer at the Economic Policy Research Center. He has worked as a content developer for the East African Community Women in Business platform, and also as a bureau chief with the New Vision. He holds a bachelor's degree in mass communication and a master's in project management. So you'll be in good hands as you discuss issues of biosafety communication. We welcome you all to actively participate in the next session. Thank you very much for your attention. Have a nice break. Thank you, online participants and presenters. We acknowledge your very useful communications, the questions, and the presentations. Presenters and the chair for the previous session, you can pause for a photograph before you go for break as a team on the stairs of the podium. No. Those are official photographs. The next level.
Yeah, I have but you better start their videos, becomes more engaging and speaking because of network.
Our farmers control ticks using acaricides, but they spray the ticks twice a week, which is very high. So now, if they spray twice a week, that means they are using more than a double the acaricide. So now we need a better approach, and this is urgent because 60% of the rural households keep livestock, mainly cattle. There are many tribes and cultures. Most of the cultures in this country require that you keep a cow, require that you keep livestock. However, acaricides have been failing because we have used them for a very long time. So because of that, we now have moved into development of vaccines. Vaccines for controlling ticks. Now, when we effectively control ticks, we shall also effectively control tick-borne diseases. So because of that, it requires a lot of biosafety design protocols. Under the Uganda National Council for Science and Technology, we have institutional bodies which perform their roles. For example, we have the Institutional Animal Care and Use Committee at the level of NARO. So those ones make sure that we do not mistreat our animals as we develop our technologies. Then we have the Institutional Biosafety Committee. It also ensures that we use the technology for doing something. We develop a technology which is safe. It is very safe and it's suitable for use in animals. Doing it in such a way that our experimental animals are confined within given premises. There are those ones at the station, which we call on-station clinical trials. And then there are those in the field, which we call confined field clinical trials. Now, here the animals are confined within paddocks, several paddocks. And within the paddocks, we must ensure that the animals have their freedoms. They should not fear. They should not they should have adequate feed. They should have adequate water. They should not suffer from any disease. In other words, even if we are so much interested in developing our technology, we ensure that our animals are safe and healthy. We don't allow them to fall sick, okay? And the animals should not live in fear. In other words, we should provide adequate security. So those roles played by the National Biosafety Committee are very important in that we handle animals ethically. We ensure that their welfare is maintained. And therefore, even when we have our vaccine eventually, it will not have a bad image. We shall have not worked against the animal rights.
I want to take this opportunity to congratulate the Uganda National Council for Science and Technology upon the 25th year anniversary, and thank them as a farmer and a farmer representative for the work they are doing for the sector and for the farmers. In particular, the National Biosafety Committee for the work they are doing in ensuring that the technology that is being put out there in the market is safe for the farmers, and we appreciate that. But also ensuring that the farmers are represented in this body, and we hope that we continue to add value on this. As a farmer and as a farmer representative, one of the key things that we would love farmers to have is increased changes, increased advancement in the different technologies that are coming on the market. Because of things like climate change, unfortunately, our traditional methods, traditional technologies can no longer be as effective all the time. It is therefore critical that technology catches up with the changing environment and climate. And biotechnology is key in ensuring that farmers can continuously produce despite these changes in the environment. We see increased incidences of diseases, we see increased prolonged droughts, and all of these things mean that the crops that we have, the animal breeds that we have, really may not cope all the time with the changes. And thus, as a farmer and as a farmer's leader, we think biotechnology is critical. We therefore hope that there is continued and increased investment in biotechnology, because if we do not, we'll be left behind, and this country is still dependent on agriculture. It was before I was born, it is still dependent now, and it will still be dependent in the foreseeable future. Therefore, it is critical that we make sure that the technologies that we keep pushing out to the farmers are relevant and can respond to the changes in the environment. So as farmers, we think it is important to create a space where this knowledge, or rather this information gap is closed, and we close the gaps for disinformation as well. The other thing I would like to urge government and all the other stakeholders is to ensure that we finalize and put in place regulations for biotechnology in this country. It is very dangerous, in my view, in my personal view, for us to operate in a vacuum where there is no law that is particular to biotechnology, especially in agriculture. Because whether we like it or not, these products will come to our market. They are already on our market. So you have a choice. You either have them on the market when they are well regulated by law, or you have them on the market when they are not regulated by law, which equates to anarchy. So in my view, it is one of the other key things that we would like to request from government is, please let us finish putting in place a law or laws that regulate the use of biotechnology. I think that will even guide the engagement between the developers of these technologies and the farmers.
The National Council of Science and Technology was created out of the National Research Council. The National Research Council was a government entity which belonged to the Ministry of Finance, Economic Development and Planning, which was handling research priorities, and identification of research funds and supervising research in the country. But then the National Research Council was a very small entity. The government decided to make it a bigger entity. So the National Council for Science and Technology was based at Uganda House. Now, it was first based at the now Ministry of Public Service, the veterinary section, where there was once an FAO building, where there is a small animal clinic. It was based there. That's where the National Council of Science and Technology started the National Biosafety Committee. And by then, it was necessary to establish the Biosafety Committee because research was beginning to take place on the use of HIV vaccines. And we wanted to be sure that we knew the safety of these vaccines. There was an attempt to vaccinate cattle against a disease which affects people, especially sore throat. And the idea was that if cattle were vaccinated, they would produce milk. If they drank it, then people would be healed from antibodies which cows produce. Then there was also a need to introduce agents to digest fecal material in latrines so that they can go down. Then there was a possible idea of looking at the control of mosquitoes using biotechnology. All those were things driving this. So it actually started as a human-animal biosafety, because that's where it began. Agricultural biosafety came a little bit later, because biosafety was very, very important in human protection, vaccine protection, and environmental protection. That was the main reason. Once it was established, the membership was created, quite a big membership, representing farmers, representing the academia. Makerere University had 2 representatives, one from agriculture, one from veterinary medicine, and one from human medicine, actually 3, one from human medicine. And then we had many other representatives from agricultural research institutes and from the medical research council by then, and so on. So we had very many people. So subsequently, it became necessary to look at crop biosafety. For a long time, we have had what we call the Biosafety Committee, and that was responsible to ensure that the work we do in the labs is done in a safe way in terms of how people dress themselves up, how they dispose of their waste, how they dispose of their chemicals, how they dispose of their radiation. And we had a safety committee. But there have been more developments, and now we talk about the Institutional Biosafety Committee with the new developments, especially the developments around biotechnology. We are working with a lot of organisms that have been, where genes have been inserted, and also globally and internationally. I think now there's a more talk about how do you regulate genetically modified organisms and recombinant DNA technology. And that, of course, now we're to support both at the national and institutional level. And also I thank them for the partnership we have created. Technology came in as a very important way of safeguarding the population, especially as a result of population increase. It became important to ensure that is there sufficient food for the population? Is there sufficient energy for the population? Is there sufficient shelter for the population? Is there sufficient clothing for the population? With an explosion of the population,
it became necessary to ensure that normal formal agriculture could not provide food, clothing, energy for this export. And we all know that human beings have for a long time used living organisms to benefit themselves. Living organisms have been used in the production of beer, production of ajono, tonto, mwenge bigere. All that is biotechnology. Sour milk, production of yogurt, fermenting of food. So all that is biotechnology. So it's not a new thing. The only difference now is how to enhance biotechnology at a genetic level. So, and that of course led to vaccine production and also led to drug production. For instance, the drug against diabetes, insulin, can now be produced using biotechnology. Is now the way of production of insulin. In the past, they used to produce it from pigs and from sheep. But that was not sustainable. Large-scale production was not possible. So we're now looking at how can we produce drugs using bacteria called E. coli, which if you introduce an insulin gene into them, they can produce the insulin in large quantities. That's what's happening in the world now. For plants, tissue culture was very important for production of clean material. Those seeds which are free of disease. So that when you plant seeds, you know that these seeds are safe. So clean planting material, as well as production in plants, production of disease-resistant plants. So all in all, biotechnology has been very useful in increasing production. That means you can now cover large areas of acreage without necessarily suffering the costs of labor. Right now in this country and in the world, especially in the third world and in Uganda, the biggest problem we have is labor, weed gardens. You go to the rural areas and you will see people can open land, but they will fail to weed it. In the developed world, acreage has been increased because they no longer weed. They spray and all the weeds die. So the crops must be resistant to the chemical which is used for spray. And only the weeds will die and the crops survive. So you can find one man has acreage from here up to Ginger. Biotechnology is the reason why the prosperous world is surviving better than us. And so the question is, we in Uganda, what are we doing? What are our scientists doing? Why are our farmers suffering? What are our universities doing? What is government doing? And that's precisely why biotechnology came on board. Not only to increase production and productivity, but subsequently to reduce poverty. The more you can produce, the more you sell, the better off you are. And this goes back to the Millennium Development Goals, where poverty reduction is a priority. And you cannot do that if you are not self-sufficient. Food security, again, hunger reduction, Millennium Development Goal. How will people survive if there is not enough food? So food security or hunger reduction is a priority. as a result of biotechnology. Lower cost of production. As you know, that to produce, you need to control pests. You need to control weeds. You need to make water available. Can we afford irrigation? Can we afford to fertilize soil? Can we afford to control pests like the locusts and so on? For us, using biotechnology, if we had a capacity to have plants which, if locusts ate those plants, they would die. That's biotechnology. Instead of trying to do all these things which we have had a problem with. So indeed, food security, lowering costs of production, and lastly, a reduction of use of pesticides. The more pesticides you use in production, the more you destroy the environment. And so if you reduce spraying, these days when people are growing, and they're growing maize, they're growing anything, even oranges, you have to spray 2, 3, 4 times. Where do those pesticides go? They go to the soil, the water washes them, rain washes them, rainwater washes them, they go to the water system, it ends up where we are, ends up in the food chain. It's all very dangerous thing. So if we can have plants which can resist pests, we do not need to spray them. The moment food is owned by someone else, you beg. But if you own your own food, you can eat it anytime you want. You can do what you want. So do we own our own production? Do we own and do we know the biotechnology which is used in crop science, in animal science, in human science, in to better health? For instance, nutrition, to increase nutrition, to make available certain minerals and vitamins in crops, which when we eat them, our health is better. And that's one of the things we try to do in Kawanda with bananas, to produce more vitamin A rich bananas, so that as people consume these bananas, they are more healthy. In other words, nutrient rich products.
I had the opportunity to represent Uganda in the negotiations under the Convention on Biological Diversity, whose main objectives are the conservation and sustainable use of biological diversity, and then the fair and equitable sharing of benefits arising from the sustainable use. Now, Uganda, of course, ratified the Convention on Biological Diversity, which was adopted in Rio de Janeiro in June 1992. And under this convention, there is a provision. There was already a concern globally about the developments in modern biotechnology or genetic engineering. And some people already thought there could be risks that could undermine, arising from the use of modern biotechnology, that could undermine the conservation and sustainable use of biological diversity. So under Article 8, subsection G of the Convention on Biological Diversity, parties to the convention were required to put in place mechanisms at national level to manage and minimize the risks to biodiversity that could arise from the development and use of modern biotechnology that arise from genetic engineering. But of course, this requirement was there. But at that time, this was 1992 and the early 90s, the level of knowledge and understanding of the products of modern biotechnology was still very limited, especially in developing countries. Now, the same convention in Article 19, Section 3, Convention on Biological Diversity, put a provision. They knew there would be shortcomings at national level for individual countries to go it alone. So they put a provision that parties shall consider the need for and the modalities of a protocol to especially govern the transboundary movements of living modified organisms resulting from modern biotechnology that may have an impact on the conservation and sustainable use of biodiversity and taking into consideration effects on human health. So in 1995, July, the Convention on Biological Diversity convened a meeting of all parties to the Convention on Biological Diversity. Even non-parties attended as observers. This is a meeting of experts on biosafety and biodiversity to consider this need for an international treaty or a protocol. The meeting was held in Madrid, Spain. I had the honor to represent Uganda in that meeting. And after 5 days of deliberations, the consensus was that indeed there was need for a protocol, an international treaty to govern the transboundary movements of living modified organisms resulting from modern biotechnology that may have an impact, a negative impact on the conservation and sustainable use of biological diversity, taking into account risks to human health. Now, this working group made a recommendation to the second meeting of the Conference of Parties that was to convene the same year in November, I think November 1995 in Jakarta, Indonesia. And the meeting of the Conference of Parties to the Convention on Biological Diversity, having considered the recommendation from this working group on biosafety, established an open-ended working group on biosafety to negotiate and elaborate this international protocol. So the negotiations took some time, and they were supposed to be concluded in the Colombian city of Cartagena, 1999, January. Then, but for some reasons, there were some disagreements with some countries not agreeing with what was the desire of the majority of the countries of the world. So the negotiations were suspended, and there were informal consultations that went on in between, both at the Africa region level and at global level. After some consultation, consensus was built that it was time to resume the negotiations. They were resumed in January 2000, and then the final text of what was called the Cartagena Protocol on Biosafety was adopted in Montreal.
Distinguished guests, researchers, scholars, ladies and gentlemen, welcome back from the tea break, and we thank you for having observed the time. We'd like to sincerely apologize to the Chief Executive Officer, National Biosafety Management Agency, Nigeria, Rufus Ebegba. I hope I have done that properly. He has been online waiting because we have lost some time before and after break. He's been online waiting. The session for this time is biosafety communication and public engagement. I am not Margaret Karembu from International Service for Acquisition of Agribiotech Applications. I'm standing in for her. She's in Nairobi, and I'm happy she's following us. The Chief Executive Officer, National Biosafety Management Agency from Nigeria, will introduce himself briefly, given what Professor Karepu expressed as a concern about the unupdated profiles and CVs. I would not want to dwell so much into the depths of this profile, and I will request him to introduce himself. But besides that, we'll have a panel discussion after the presentations, and I would like to invite Professor Arthur Tugume from Makerere University. Mr. Isaac Kongu is a presenter, and Mr. Herbert Oluka. I'll shortly be presenting their bios as we wait to start this session. Can we start as others trickle in? Distinguished guests, welcome back. I see the Executive Secretary, Uganda National Council for Science and Technology coming in. I'm not a scientist, a physical scientist, I'm a social scientist, but at least from my interactions with Dr. Hafashimana, I've heard a lot about the Cartagena Protocol, and I know that it emphasizes the need for informed decision-making through public participation. That's the reason we are engaging, and we shall continue to engage, and we shall not stop engaging. And I'm also aware that Article 23 of the CBD, the Convention on Biological Diversity, provides for need to promote and facilitate awareness, education, and public participation, including access to information, safe transfer, and handling and use of living modified organisms. So we have a good background for public engagement. Without wasting more time or taking more time, I would like now to invite the Chief Executive Officer, National Biosafety Management Agency, Nigeria. Please join me in welcoming him. He's online with a round of applause.
Good morning, everyone. Let me congratulate the organizers for sustaining the annual Biosafety Conference and also inviting me to this very important one for the year 2022. I'm Dr. Rufus Ebegba, the Director General, National Biosafety Management Agency of Nigeria. The National Biosafety Management Agency of Nigeria was established in the year 2015 with the sole aim of ensuring a holistic biosafety system within the country, so that biotechnology products, biotechnology and its products can be used in a way that it will enhance the economy, to diversify the economy as a whole, and also to come out with products that are environmentally sustainable, that do not have negative impact on the human health. My presentation today is the engagement on public engagement on environmental release of genetically modified organisms. My presentation has the following outlines: introduction, practices for effective public engagement, case study of Nigeria on public engagement, and under that we also have a brief on the National Biosafety Management Agency, GMO approval for commercialization in Nigeria, process leading to environmental release of GMOs, public engagement for environmental release of GMOs, and what are the next steps, and my conclusion. Introduction. Public involvement is a critical part of a development, evaluation, and acceptance of any new technology. It has been considered to be especially important as part of biosafety decision-making process, and is included in the Cartagena Protocol.
Can you please put your presentation in presentation?
Pardon?
Would like to have your presentation in presentation mode.
Okay.
Be visible.
Sorry. Okay. However, the inclusion of public participation in protocol is linked to the sovereignty of countries to determine how best to do such public participation. Effective engagement involves all important stakeholders. These involve products, those who are involved in product development, product users, and regulators. In order to be effective, these engagements must be timely, transparent, and trustworthy. Successful commercialization of genetically modified organisms require not only regulatory approaches, but also public engagement for acceptance. Specific public engagement practices create a foundation for systematic methods to gather, acknowledge, respond to, and even incorporate public input. Applying such best practices will increase transparency and optimize the value of input from the public. For effective practice for this, we have to in the utilization of range of resources to facilitate public education and opportunities for understanding complex technologies, defining in advance the goal of seeking input, identifying and communicating with the critical public groups from which input is needed, using a clearer, defined approach to gathering and assessing what will be used in making the biosafety decision, and communicating using clear and simple languages. The Nigerian biosafety system as a case study. The Nigerian biosafety system, as I said, officially through an amendment, through an act of 2015, came into a legal and administrative mode for actions to be taken. It is true that the issue of commercialization of GMOs have been possible. With the creation of the National Biosafety Management Agency to ensure legal and administrative mechanisms for the regulation of modern biotechnology and its products, the agency has specifically the mandate to be in charge with the responsibility of providing regulatory framework, institutional and administrative mechanisms for safety measures in the application of modern biotechnology and the use and handling of these products in Nigeria. For the agency to effectively carry out its functions, various instruments have been put in place. We have policies, we have laws, we also have guidelines, and also have a GM detection analysis laboratory. Since the existence of the agency from 2015 to date, we have approved 4 crops for environmental release. The first was 2016, a year after the existence of the agency. And let me also relate to you the experience we have had in the area of public engagement because of the first release of the genetically modified BT cotton. When these products was released in 2016, there was serious public outcry, demonstration at the Parliament building, seeking government to remove me as a chief executive officer, seeking government to prosecute me that I've taken bribe to approve genetically modified cotton for Nigeria, seeking government to remove me that I'm trying to kill Nigerians with GMO. It was a serious challenge for me as a chief executive officer. I had no option but to go all out to convince Nigerians that the decision I have taken is for the best interest of Nigerians, and not just for the best interest of Nigerians, but the GM cotton which we have granted approval for. It is safe and it's going to be useful for the Nigerian economy and also empower Nigerians in the area of employment and other economic endeavors. This was not easy. Appearances in media houses, publications in print media, and not only that, having various workshops, seminars to sensitize Nigerians on the role of the National Biosafety Management Agency. And not only doing that, also requesting the agency in charge of promoting biotechnology, which is the National Biotechnology Development Agency, to also come out to support, and also gathering all stakeholders, both from the academic community, academia, and also partners to support the process of enlightenment and public engagement. It's on that led to the reduction in the constant attack and harassment of the agency. It came to a point that as a chief executive officer, I could not walk freely in the Nigerian streets. I had to be going about with armed security for protection. Even the Secret Service advised that I shouldn't travel out of station without then giving me a backup. That was the major challenge of holding a public office in the regulating a GMO within the United States. But I can tell you, despite all those challenges, our hope was the fact that we believe we have the confidence, we have the knowledge, and also have the courage to go ahead to do what we have been mandated to do by the Nigerian government and Nigeria. We were not deterred, we were not discouraged, we were not intimidated. We went ahead to do what we need to do. We received further applications, both for confined feed trials and also for commercial use, and also for food and feed processing. So far, we have granted 4 approvals, as you can see. And this I've also granted another approval this year. You can see that nearly every year we have been granting approvals. That is our responsibility. Once we are convinced that any product is safe, we see no reason why we should not grant such approval. But the fact that the process is transparent in all the processes, not that the agency does it alone. Invite other in the institutional arrangement of National Biosafety Committee and National Biosafety Technical Subcommittee to do independent review of all applications and advise the agency. The agency is given discretionary powers to take decisions on behalf of the government and people of Nigeria on matters of GMOs in the country. And the agency has done that effectively. I want to let you know that the agency has about 350 staff doing various areas, thematic areas on biosafety. And these staff gradually every year acquire new knowledge and skill. It's on that note of the fact that we have been moving. A new mandate was given to the agency through the amendment of the Act in 2019 to put in place measures in the area of to ensure biosecurity within Nigerian space. And the mandate to also regulate emerging aspects of modern biotechnology was also given to the agency. In view of that, we have developed guidelines for gene editing, and we are currently developing guidelines for in the area of synthetic biology and also gene drives. And we have successively developed a national policy on biosecurity for the Nigeria, embracing One Health approach. The processes for reviewing application in Nigeria is what we see here in this thematic area. First, submission of application, acknowledgement of application, checking for completeness, requesting for additional information if need be, and public views. And how do we do this public view? Publication in 3 national dailies for the members of the public to take a critical look of every genetically modified application for the permit for a genetically modified application in Nigeria. This dossier is deposited at various locations and is published in the media, asking the members of the public to go and take a look at it. And we also put it in our website for the members of the public to take a critical review of such and also come up with their position. Their position guides, their position sometimes guides our decision-making process. So far, none of the public, those who have objected to some of these applications have been able to provide any scientific evidence why such applications should not be granted. We do not dwell on emotions because most of those who have condemned our action, which are civil society environmental rights, they bring up evidences that do not stand the test of scientific verification. As such, we set them aside and take decisions in the best interest of our country. This is the National Biosafety Committee. It's one of the public processes where the members of the public committee. And this is the table of how we take our decisions. Our public participation, the Nigerian approach to communication with the public about the use of GMO into the environment is determined by legislation and by administrative processes established to achieve consistent, sound regulatory decisions based on science and economic consideration. Who do we really talk to in the area of public communication? We talk to the applicant and also the permit holder, the National Biosafety Technical Committee members, which are drawn from various ministry departments and some of the experts from the general society and academia, ministry departments and agencies. They are also communicated. We also involve other regulatory bodies, the Nigerian Customs Service, the Standard Organization of Nigeria, the Agricultural Pension Service, Nigerian Agricultural State Council. These are all involved in our processes. We only notify them, also constitute them into the National Biosafety Committee so that they will have advanced information about all we do and the general public through media publications. our own this thing. What are the periods we do this? We do this before and even within the period of the application. When an application, before an application, the engagement process, before approval, public in national dailies, public display of application and strategic locations in the country, the constitution of National Biosafety Committee and National Biosafety Technical Subcommittee drawn from various groups, principal investigation presentation to National Biosafety Committee and also to the public. One thing we also do is that at particular time, after we are even at the process of taking a decision, we give opportunity to the principal investigator to address the press, to also members of the public who are willing to listen to why the application should be approved, the importance of the application to the economy, and what are the due safety diligence they have put in place, and all the risk assessment data they have collected. They present this before the public so that the public will be aware of what is coming, and also hear from the applicant that this is what we have done, and we are convinced that what we are presenting before government for approval is safe and also has economic benefit and does not have any negative impact on our social and our cultural values. And also after the approval, we also make publication.
CEO, you have 5 minutes.
Okay, I'll be rounding up. We also make publication in our website. We submit our sole decision to the National Biosafety Clearinghouse, and our quarterly newsletter also carries such our decision. Our short documents also made public. This is the publication we make and the public engagement. Why we do it to ensure confidence in the process. The members of the public to also make up their mind to accept or not accept the document. In conclusion, public input to biosafety decision can be a valuable resource for improving risk assessment, communication, and risk management. Public education and enabling initiatives can play an important role in supporting input to biosafety decision-making process. Distinguished participants and listeners, I want to say thank you. That is what we do in Nigeria. Thank you for the opportunity. This is our communication channels. Thank you.
Thank you so much. A round of applause for our CEO from Nigeria. The key take home has said we should not only emphasize the regulatory approach, but also public engagement for transparency and buy-in. They should also have a clearly defined approach to communication. We should have an objective for communication. And he also emphasized the simplicity with which we should put out this communication while doing public engagement so that people really understand us and they can even be our champions. And this has led them to releasing those 4 products so far. And the public has the right to know before some of these products are released to the environment. So as a result, they have gained a lot of confidence and is no longer worried about being stoned or questioned about how they have done this. And of course, he has also emphasized the issue of mapping audience, identifying audience, packaging the messages properly to suit certain interests, and finally, identifying various channels of communication to reach the people, the website, newsletter, blogs, policy papers, and briefs, etc. We thank you so much for those insights. Now, without standing between me and your lunch, I would like to invite Mr. Isaac Ongu to the stage, to the podium, to tell us about the regulation. This talked about public engagement. Mr. Isaac Ongu will tell us about regulation. As Isaac comes to the podium, I would like to celebrate him that he's the Executive Director, Science Foundation for Livelihood and Development, SICFOD. He has a bachelor's degree in agriculture, master's in public policy and governance. He has expertise in science, communication, policy research, and advocacy, and he coordinates communication and public engagement for the virus-resistant cassava for Africa project in Uganda. He's also a practicing Christian, Mr. Isaac Ongu. And the topic is addressing key stakeholder biosafety concerns through regulation and public engagement. Welcome with me, Mr. Isaac Ongu.
Good afternoon, ladies and gentlemen.
You have 15 minutes.
As it was mentioned, this is a joint presentation. We are supposed to handle concerns, biosafety concerns, and how those concerns can be addressed through public engagement or regulatory means. I'm going to handle the regulatory component, and my colleague from OFAB will speak on public engagement.
Therefore, you are sharing the 15 minutes, the two of you.
Yeah, from the graph, you can see the trend in global adoption of GM crops. And the only significant country in Africa is South Africa, just because its agriculture is highly formalized. On the first day, we recognize the importance of formality. That graph of Africa shows the regulatory landscape. Those countries that are green, we've already heard from Kenya and Nigeria, which have used new laws to have their regulations and guidelines. And they also have stars in their countries showing they have commercialized genetically modified crops. Uganda has some stripes, green and stripes, because we've functioned under existing laws. We don't have a new law, and we have been trying to have it. Yeah, the global concerns as per the Convention for Biological Diversity, there are risks to human health, risk to environment, disruption to ecological systems, and other questions that law should answer are: who should pay for harm in case there is any harm? How should biological resources be accessed? How benefits accruing from use of biological resources be shared? And also traceability. That shows the regulatory concerns in Uganda as taken from the 2 letters that the president wrote, rejecting to assent to the biosafety bill. There are so many concerns, but I've managed to put them into those categories. The institutional, the key concern there was what he called the home of the regulator. He wanted it to be under the presidency, and that concern has already been addressed. The regulatory agency right now is under the presidency. Then there are issues of conservation of indigenous crops. And that has also been handled administratively, where NARU has a conservation component, but also NEMA's law covers the need to conserve biodiversity, and they're supposed to come up with regulations and guidelines to that effect. And then liability and redress. If you look at these concerns, most of them are environmental, and the issue of liability is mostly the environmental law. In the last year, the major issue was whether the liability should go to those who intentionally cause harm, or the researcher should be strictly liable. And the issue of strict liability, by consensus from scientists, was that it would drive away scientists from conducting research in gene technology. And we have seen countries without strict liability like Kenya and Nigeria, they have gone ahead despite the threats that are associated with the technology. Then there are issues of coexistence. There was need that if you are to introduce GMO crops, how would you keep away the current crops or the crops you are modifying? And this was supposed to be addressed through regulations that can either come from NEMA or from the Ministry of Agriculture, but also technically from the technology itself. There are some crops that are vegetatively propagated, like cassava. It's very difficult for it to mix. Yesterday, we saw provitamin A banana that is orange. It's very easy to identify the genetically modified banana. And we are also told it's easy to see the genetic makeup of the GM bananas and be in position to identify. But that issue is also a regulatory issue that can further be addressed through guidelines or regulations. And then there was the need to take precaution that we should not proceed without putting in measures, and the president was talked about we shouldn't go into human cloning. And I think the health people who are here, we've not heard of human cloning. We only heard about gene therapy as a treatment to diseases like sickle cell anemia. And then access and benefit sharing. That how you talked about if you are to introduce a gene in the existing crop, how would those who have been having that crop benefit? So it needed clarity on the benefit sharing. And also access and benefit sharing is actually an environmental law, and NEMA has regulations for that, and guidelines for accessing genetic resources and benefits that accrue from it. And then the issues of regulations that have been lacking. And I remember the president asking, has NARO already released the GMOs in the environment? And it means that shows he understands the mandate of NARO and that their responsibility is to release improved crops. His only concern was that when released, some of the issues he raised should be addressed, but his problem wasn't with the GMOs per se. These are some of the global responses to addressing biosafety concerns. Establish, designate regulatory administrative systems. We've seen in Kenya and Nigeria, there are new systems. In Uganda, we designated the Uganda National Council for Science and Technology that has been in existence since 1994, and also the National Biosafety Committee that has been in place even before the Cartagena Protocol came into existence in 2002.
5 minutes.
And also enact laws, regulations, and guidelines to govern research and development and involve people in decision making. I've tried to show the regulatory status in Uganda, and there's that gap in between research carried out by NARO or health research. It's still it started from the laboratory and field trials, and they have a product that should go to the farmers. The farmers are holding that product to those two individuals in the middle, but there is a disconnect, and who is not doing their job? That's the question that we all need to ask, because we have all the framework in place, we have all the institutions in place, and we have the product. What then should come next? Yeah, that's the regulatory environment that we are in. We have cabinet and parliament that are responsible for policy and legislation. And parliament also delegates that power of legislation to ministry to issue regulations. And people who stay in cabinet and parliament, they are mostly politicians, and they have various interests. They care about their voters, and at times when they are making decisions, they put their interest first, not necessarily the interest of the general public, because they define the public differently according to their interest. And that is what is done basically by politicians and all of us, that individually we are inherently selfish. The decision that we make, we first consider ourselves first. And there's that green, those circles in the middle, showing how problems are identified, who identifies the problem. If the problem is say, lack of vitamin A, and people are coming with solutions. Some people are saying we need to biofortify. Others are saying, no, we don't need biofortification. We need to give pills. And whoever takes the day would influence the decision in the middle, and that decision would go through policymakers and would determine what comes out. And that could be the reason why the policymakers, they seem reluctant to come up with legislation that would inform regulations by the ministers in charge, so that the guidelines from agencies and the standard operating procedures can also be in place. And below there, we have the regulatory agencies. We have the UNCSD, we have the Ministry of Agriculture, we have the National Drug Authority, and NEMA and UNBS, they are more with the consumers. And also under UNCSD, they have the IBC. UNCSD has highly qualified individuals, both at the council, but also in the NBC and in the IBC as well. So on issues of competence and capacity, we are not lacking. This is the political will. We need the political will. And in Uganda, I think we may not say we don't have political will, because we have some trends that show political will. In 2003, His Excellency the President opened the biotechnology lab in Uganda. Uganda has allowed biotechnology research trials across all regions of Uganda. And I mean all. All major crops in Uganda, including matoke that we love most, have been improved using gene technology. And Uganda's university, spearheaded by Makerere, has trained students in biotechnology. Uganda has invested money in developing tick vaccines as well as COVID vaccines using gene technology. And most recently, the president noted that biotechnology could help Uganda improve sorghum to make it tastier. And our major challenge is willful ignorance, or that someone does not want to accept facts because the fact would not agree with their value. And that one, they were talking about the nuclear power, but we could include it, we could say GMOs. Someone saying, I hate GMOs. Why? Because when you plant, it does not germinate. And then the science provides you with the evidence, but you still go ahead and say, I don't want to know. And you go ahead to refuse the solution that they are offering to you. And this kind of challenge, even public engagement may not address that much. In conclusion, Uganda has the best research infrastructures for all biosciences. Relevant regulators should convene and decide on ready products like banana, cassava, and maize and potato. Time for regulatory clarity is now. Thank you.
Thank you, Mr. Ongu. Okay, we are consulting because this is public engagement. So I was engaging him on certain issues. He has identified public concerns, has also identified global concerns. I don't know if we have domesticated those ones to be our national concerns, but they are mostly fears and risks, the fear of unseen. Oh, we are supposed to have a presenter from OFAP, Nairobi. I don't know if he's online, Colin. Can we invite him then to talk to us? Is he the one, Dr. Chinoko Vitumbiko? The one? Okay, and he's going to talk to us, still addressing the key stakeholder concerns, biosafety concerns through regulation and public engagement. Is he ready yet? As he comes, this guy raised the question, this gentleman, the presenter raised the question, who is cheating on the job? I read a novel by Arthur Koestler, I think was talking about a revolution in some country, and he used your science. He said, oh, our science. He said, we have diagnosed a problem with microscopic exactness, but whenever we apply the surgical knife, a new saw appears. So where is the problem? Is this the knife? Is it the microscope? Where is the problem? Once we identify the problem, then we can look for the proper, you call it what? Prescription. So can I invite online, via online or virtual, Dr. Chinoko Vitumbiko to come and also share insights about the public engagement. This is not around. There's someone who is supposed to represent him. Is it Philip Chemonges? Okay, if that's the case, now we are going to go for a panel discussion, very brief as we go for lunch. I would like to kindly invite Professor Arthur Tugume. I hope he's around. Professor Tugume needs no more introduction. He is a professor of plant pathology and virology at the Department of Plant Sciences, Microbiology and Biotechnology, College of Natural Sciences. They call it CONUS, Makerere University, Kampala. He has been the head of this department at 2 different times, between 2012 and 2022, and currently is the Dean, School of Biosciences, the same college. Between 2006 and 2010, he was part of the core team of senior Ugandan scientists that created, designed, and championed the creation of an undergraduate program on Bachelor of Sciences in Biotechnology under the Millennium Science Initiative project. This program, BSc Biotechnology, aims at building a critical mass of young scientists with basic knowledge of biotechnology and provides support for the development of this country. He also runs different research projects. So welcome with me, Arthur, or Professor Arthur Tugume. I would also like to invite to the stage Mr. Herbert Oloka. Herbert Oloka is a biosafety specialist with more than 14 years of continuous experience in biosafety, policy and regulatory development, as well as risk communication. Herbert currently works as the country coordinator with the Program for Biosafety Systems, a partnership capacity development initiative of the International Food Policy Research Institute. He loves to see Uganda and Africa transform using science technology. I think this one shares the minister's vision of we must make Uganda the best. So you are welcome, and I would also like to believe that our CEO from Nigeria agency is still online. So we are going to have some few questions or insights or discussion about public engagement as far as biosafety is concerned. Yeah. So where have I written events? Starting with, I expected also Mr. Philip Chemonges, if he can come to the stage. Philip Chemonges works with OFAB. He will tell you what it is in full. But as he comes to the stage, would like really to know, the CEO from Nigeria has told us about identifying the audiences, segmenting them, and addressing them differently. For example, after this forum, we are going to have a communique. It will be a general communique. Will be there a special one for the members of parliament, or the Committee on Natural Resources, or Science and Technology. Dr. Boa yesterday disclosed here, but he did not give full information. He's under immense pressure from lawmakers and the policymakers. They want already to take the plants, the products. Maybe they have already taken some, but he's not under liberty to disclose. So who are our target audiences, or which audiences should we target, or stakeholders that we really identify or help us overcome the current obstacles as far as what we want to achieve is concerned? And I will start with Professor Arthur Tugume. Who should we target? The concerns have been told, the hindrances. We have had case studies from Nigeria and other successful countries that have implemented this. So who should we target, and what key messages should we give to these people or we are targeting? Starting with Professor Arthur Tugume.
Thank you so much, moderator. Yes, whom should we target? It depends on what we are aiming at achieving. If we are aiming at delivering food security, delivering a functional health system, then the population is our target. But then the population have got leaders who are responsible for making decisions. And those are the people who should be. Unfortunately, it looks like our political economy is much stronger than certain other economies. And for that reason, we perhaps may need to be able to target our leaders, our legislators, give them the right information, communicate properly, because that is something that will be important. And that goes back again to how we engage, who is communicating to who. Because even if you have the right target, but then you have wrong vessels of information flow, you won't get your message across board. So my thinking is that everyone is a target, but in a different form. We could inform journalists, we could get the journalists informed, and maybe they can also help us to translate our messages in a proper way. I think yesterday I was getting it right that depending on what you want, you can twist the story and get the message completely differently. So I agree we could target everybody, but then we may have to target differently. So it has to be, I want to call it coordinated movement of troops as opposed to random information. And then we need to know who is targeting who and who is aiming at what. Otherwise, it becomes quite confusing. Like somebody was mentioning yesterday, you get two scientists in a room and you are trying to force them to agree because you already know that the two people are on the different sides of the aisle, and you want to stimulate an unhelpful discussion that is not going to help anybody. So in that case, you are not building consensus. So we need to be aiming to be focused, to target particular people, but also target in a different way. So the message to our leaders in the parliament, in the executive, that are involved in decision-making is probably not exactly the same as we do to our farmers, because our farmers, what they need is food, and safe food and consistent food, and making sure that what they need actually to put on their table is always available. And also to make sure that if we have patients, they are sick, all they need is medical care. And that should be the way we should package. So it depends on how you package it. Thank you very much.
Thank you, Professor Herbert. I think you experienced in policy and advocacy. How best should we package this information or these messages going to policymakers, lawmakers, the consumers, the targeted consumers? Yesterday, a presenter from, I think, NPA was showing us figures that we have lost so many billions of money in delaying to adapt or implement or deploy some technologies. Should we use this kind of fear that unless we do this, we stand to lose this? Or how do we frame the messages to make sure it is beneficial to everybody, that people can easily adapt, that the infusion of innovations can take place? Or should we use the benefit or gain message to say that if we are to adapt this one, this is how much we shall be getting. How best can we frame our messages to the targeted audience?
Thank you, moderator, Angie, answering the question. So I'm not sure what I should say. But I wanted to first go back to the main reason we need to communicate. As Professor Tukume also noted, there has to be a deliberate effort. What are you looking forward to achieve with your communication? So before you package the message in a different format or in different channels, what do you want to achieve with your communication? Your audiences matter a lot, like we have already noted from presentation from a colleague at Rufus in his earlier presentation. So you must be deliberate when you're communicating. That means you must know what you want to communicate and to who. Then you must be very focused, as Professor also noted. But beyond that, you need to be very, very strategic about it. So therefore, if your question on how you package this information will depend on one, stakeholders you need to reach out to, because these different stakeholders have different ways of receiving messages. Because some are very many, like farmers, I'll give an example, farmers and consumers, you're not going to find a method of engaging them one-on-one effective. So the best way to move with the engagement is look at your audience critically and what you want to communicate to them in your area of being, and as you're focused. So if it is consumers and farmers who are the mass of the general public, the best way to reach out to them is through the media. But your message must be only and only about things that concern them, which will be benefits about your technology, and then safety concerns. So you address their concerns in terms of how they want to, what they have, any questions they might have about the science you're working in or the education system that is delivering that science to society. If you're looking at policymakers, many avenues can work for this, but typically one-on-one engagement is the best tool for the high-level policymakers. If you have policymakers at, say, regional MP level, the more effective way is to reach them in deliberate meetings that are very focused on issues that concern them, whether it's about policy or it's about the products that you're developing. And if you think you have to reach out to the general public, which normally we don't recommend since it's very difficult to be deliberate and focused when you're reaching the general public, then the media offers you the best avenue, the general media. The media can be social media, can be broadcast media like radio and television, or even the conventional newspapers can deliver a message to people. So really, how to package information depends on the audience you're targeting, because audience have different ways of accessing information. Thank you.
Thank you. Herbert has talked about the objective of communication. Why do we communicate? What are we targeting? I think we have already created a lot of awareness, in my own opinion. So what next? Are we educating? Are we sensitizing? Are we trying to influence opinion or advocacy? So that's what he's talking about. Yesterday or the other day, someone talked about the era of is it called infodemics? There is the emergence of social media, that information, negative information or bad press is traveling faster than the fact finding and the company. So Mr. Chemonges, what are the best channels or avenues or approaches of communication should we employ? We have heard the CEO from Nigeria. Given the emergence or the rampant social media and the wrong information being spread, and wrong information spreads faster. How do we counter this? Or what other appropriate channels should we use to have our messages go to the right people at the right time?
Thank you, moderator. I think the channels have already been elaborated. What I only wanted to bring forth is maybe the concerns from my experience as of our experience since 2007 to date, is that when you look at the science, it's correct. When you look at the rigorous checks, they are there. So many people many times hide behind the safety, but it's not the safety they are concerned about. My conclusion is the real problem and the real concern is sovereignty. If you listen to anyone criticizing you listen properly. They will hide behind the safety, but they are saying no, sovereignty. And why they are coming in that angle, I think it is also because I think government has left much of this work, the development partners. If you look at most of the research, even what was presented here earlier on gene therapy, where is the government's firm position? When you look at communication, where is the government's firm position? So at the end of the day, people tend to misinterpret, so if government is not interested, why are you interested? And I think that is where the real problem, and that's what we should be addressing. I'm only grateful that of recent, as a result of COVID-19, the government made up efforts and commitments to put money into vaccine research, which is home development, which was a great thing. And I think that's the direction we should be taking. As we depend on development partners, let government take efforts and dictate the direction, dictate the tune on which the research should take. And I really implore the leadership, the president, parliament, the minister, that really money should be put into, Ugandan money should be put into research. Ugandan money should be put into communication. So that at the end of the day, we want a best Uganda. We are dictating the direction. We are developing our own products, which have a hands-on of government resources in huge amounts. So I implore the president to pay a close attention to this technology and the derivatives of this technology, because it is where the future is, and the future is here. Thank you.
Thank you so much. Oh, round of applause for Chemonges. He has implored the president to really have interest in this. And earlier, Mr. Ongu talked about the concerns by the president, and the president represents our interests, all of us. So those are public concerns. We have identified them: home of the regulator, indigenous crops, so others will be protected, environment and threats. So how best, Mr. Ongu, are we going to communicate these to the authority that are sent to what we want to be enacted? How best are we going to communicate this and to convince and to persuade him to assent to this, having identified these concerns? Or if we have identified and got responses for them, what is hindering us? What is standing between us and communicating this to the president?
Yeah, thank you so much, moderator. I would like to note that actually through the office of the president, we have been told they have been the biggest sponsor for this conference. So I think he has the interest through his minister on what is taking place here. So I think out of this conference, we need to identify key issues and ask him. We know as a president, he functions better using directives. We need to ask him to issue a directive to the different agencies to wake up and incorporate regulations and guidelines into their various laws. And secondly, for the minister in charge of science and technology to come up with a framework that will bring together regulators from different agencies. We already know UNCST as the National Biosafety Committee that has representations from all the regulators, but also from civil society organizations. So I think that message we should find a way of delivering it to him through the minister.
Thank you so much. Before I call in Jacob Komakech from the audience, and I give you opportunity also to ask a few questions to these eminent persons, let me ask Professor. Yesterday he told us he enjoys teaching first years, and he told us the books we read at O-level and A-level are either wrong. Are they McKinney and others? So how best, Professor Arthur, can we take advantage of the case studies of the countries where the deployment and implementation has successfully taken place to inform our own? Okay, so Arthur, how can we use the case studies from Nigeria and other successful countries where these have been deployed?
Well, thanks a lot. We wouldn't really need case studies from abroad if we are awake. The trouble is that the people who are, I think some people are sleeping on the job, and the people who are sleeping on the job have the big say. I appreciate examples from Nigeria, the examples from Kenya, from India, from Brazil, and many others we have not talked about. Yeah, but you see, when you have a country where you are asking which doctors how to heal people, you are not going to move them an inch. You understand? You are not going. Yesterday we are told that people can comment on something they know nothing about, and we believe it. So one of the things before I came to this was that I was beginning to have a thinking with some of my colleagues who are in the audience that we may have to start a training on science communications. And I'm seriously talking about this because I happen to be the dean now, and I have the opportunity for talking with my fellow deans who are working in linguistics, languages, and communication. And we are working on that path to make sure that we are the scientists, we can do these things, but we seem not to be very good at communication. There are other people who are not doing the science, but they are very good at communication. Whether it is bad or good, they excel. You know, whatever you aim at doing, if you do it consistently and well, even if it is robbing a bank, you'll be excellent about that. So as a university and as a professor who has been at this for quite some time, perhaps we may need to divide labor, and we say, let me do the science, but let me partner with the institution X or department X or so that they can do the communication. I give the information, they communicate. And you mentioned about the old science and all that. It goes back to what we are talking about here. How long does it take? To the best of my knowledge, I know science is dynamic. And since I'm a biologist by training, it seems like biology is highly dynamic, much more dynamic than any other area. And we are all hinging ourselves and our expertise on that. But then how often do we change our curriculum? Not only biology, even in other things. How often do we change the curriculum? And who is responsible? If a guy or a gentleman or a lady who is responsible also last checked his notes the last time he was in S4, then we are not going to be moving very fast. So I think some things will need some strategic movements. Others will need us to keep continuously talking and talking and talking. We do not need a Nigerian example, much as the Nigerian brothers and sisters are doing very well. We have our own example, and we may not even need an example from the USA because the Americans are not interested in cassava. They're not interested in our banana. They're not interested in our potatoes. Those are things which we do here. And so if we used our local solutions and our local capacity, and I disagree with the fact that Uganda is a poor country. We are only poor managers of our own resources. We can be able to do much better than we are doing, and we have sufficient capacity. Now, allow me to talk about this that you mentioned, that what if we cannot, the only way we can use others as an example is where we have gone to school too, because a greatest majority of scientists in this room, I beg, if not all, highly trained, no one has been trained on government of Uganda funding. Maybe they are there, I put that possibility, but the greatest majority of us have been trained on friends' money. And then so when you look back, you say, but how come the government is not the one which is involved in training? So these people must be agents of foreign interests. And that, unfortunately, will not be the case. So, Mr. Chairman, how we can be able to use it, we do not need examples from very far away. We already have examples in Naro, we have examples at UVRI, we have examples at the Joint Clinical Research Center and many other institutions in Makerere University, so many of them, we can be able to even work on them because we work for our local communities. Thank you very much.
Thank you so much. I understand Mr. Vitumiko is online, is that true? Vitumiko is online? OFAB Kenya. Okay. CEO from Nigeria, would you like to make a very brief response to what your counterparts in Uganda are saying?
Yeah, thank you very much. I just want to add to what Avelia said. You see, the one problem we are having among scientists and developers is that they lack communication ability. No matter how good your products are, if you do not make them known to the public value, nobody is going to recognize your products. They are going to be frustrated with suspicion and being considered irrelevant. So communication should be part of R&D, research and development. Is very, very key, so that can be fashioned into research and development. Another major thing is that the area of communication, you must also give example. Because the scientists are very cold, the scientists must be able to communicate their products or their research work to the members of the public. And for those countries that do not have laws, you need to concentrate on the parliament. What do you want to tell the parliament? How relevant this technology is going to be, because science and technology is going to drive the new world in the order to achieve green economy. And your country must not be lacking. And apart from that, the products of genetic engineering, what does the FAO say? And what does World Health Organization say about them? You must also convince them of that. And other countries within your region that have been able to embrace this technology, what is the benefit? Has there been any harm to those countries? Those are the things you must also look at. And again, the regulators must be knowledgeable. They must have confidence. They must also be able to convince the members of the public of the decisions they have taken, that this product is safe, is for the interests of our people. What are the benefits to the society? Because biosafety itself is a safety and economic enabler. You must be able to convince the members of the public. And how do you do that? You must constantly engage the members of the public through the media, through seminars, and apart from that, various groups. These are very, very key. And I think those are the things I just want to talk about. But science and technology will drive the new world, and Africa must be strategically placed. Countries must be able to put in funding to achieve what those countries see as their priority. Thank you.
Thank you so much. To have evidence-based arguments in order to be able to confront maybe the fallacies and lies projected by others, should be knowledgeable. Last, we are inviting like 2 or 3 questions, but there's Dr. Nambatia. Dr. Nambatia Grace wanted to say something. Please give the mic. I'm giving her less than a minute.
Thank you for the opportunity. I'm Grace Nambatia, board member, National Drug Authority. We are a board that really get training, and I felt I had to share something on communication. Having mentioned the classical groups, politics, politicians, farmers, whoever, we need to recognize that currently we are under VUCA, where VUCA is volatile, uncertain, complex, and ambiguous system. Volatility, epidemics, and all that. And there is a new normal. As you classify groups, you need to go down to classifying them in these categories. We have the baby boomers. Those are the ones born between 1946 and 1964, have been categorized as live to work. How do you communicate to those? Then the generation X, 1965 to 1980, who work to live. And then generation Y, 1981 to 1999, who live then work. And the Gen Z, those 2000 and below, who live and then love to work. It's very important. And another issue is to look at our structures in our institutions. What informed the structures? Because we are now restructuring, because this leads into a very important component, human resource. Why are we leaving out the generation Z? just because they put a pin in their nose or have a hole in the gene. Meanwhile, they're the ones holding IT to drive the system. So we need to segment further for us to communicate effectively. And then when you talk about social media, we should know WhatsApp is not social media. It's just a messaging platform, but the others, Facebook, LinkedIn, Messenger, TikTok, and how they can disrupt all this. Thank you so much. I think I've made my point.
Thank you so much. We shall continue with that discussion. Mr. Jacob Komakech had put up his hand. Please take the mic to him, and I'm giving him 1 minute. Then we shall have 3 questions from the public to these people, and they will make concluding remarks, and we go for lunch if the mistress of ceremonies allow us to do so.
Thank you so much. I'm Jacob Komakech from Uganda National Council for Science and Technology. I have about 2 questions. Number 1.
To who?
To all the stakeholders in the house, and anybody who can give the response is welcome. There are concerns that biotechnology may lead to extinction of originality, thus requires preservation. What plans do we have in place to preserve the science of God? And was it provided for in the initial biotechnology laws that was fronted before? That preservation aspect.
Question 2.
Then, just to continue on that, there is also concern that biotechnology may lead to property rights protection of the technology. And there is fears among the farmers and the population that it can lead to costly prices of the seeds and other products.
Thank you so much.
How can you remove that fears? Then the last question is that there is also perception that we can emphasize the biotechnology in the areas of non-crops, non-food crops. What is our position in this, other than emphasizing it in the areas of food crops? Thank you so much.
Thank you so much. If I might also ask you, whose concerns and perceptions are you expressing?
I'm number 1.
Okay, those are your concerns. Now, question 2, gentleman here. You can use your natural microphone if these are delaying you. Thank you, Komakech, for your concerns expressed on behalf of your constituency.
Thank you very much. My name is Kwaro Obuku. I'm a medical doctor and a student of policy studies. I'm happy with this presentation because it speaks to what I did for my PhD. Many times scientists do good work in the dark and hope the whole world should celebrate them. But they forget to work 3 things. We have identified the problem always, and you've seen the issue of biotechnology. We have come up with very many solutions. Back and forth, I saw the schema from the narrow chief showing why the law hasn't passed. Where we fail, because we all aim to open a policy window, get funding, get the law and so on, is working the politics. We leave the politics to parliament. We leave the politics to cabinet. In the medical association, we worked the politics. Doctor salaries are tripling. Whether they are working is another matter, but they are tripling. So we opened the policy window by working the politics. Scientists should not shy away from working the politics. You are the most clever people in class, but you go to the lab and die from there. Those who are last go to parliament, make the laws, decide your salary, and you think that they'll determine the law and it will come out. We needed to do better. We needed to use political science frameworks, not the Krebs cycle. Okay? When I went for my training in PhD at the McMaster, my professor said, you leave this econometrics and this business, you come and do political science. I was very hesitant, and it was winter. But by the time summer came, I saw why. So the professor was talking about training. We're actually conducting trainings, and not training scientists, but also science journalists, because they are the ones who communicate daily. So that's one of the areas that we can collaborate, College of Health Sciences, Africa Center for Systematic Use and Knowledge Translation. I'm happy to talk to you and we see how we can work together to work the politics, by the way, because you can't just write a newspaper article and change. So we need to connect. We need to know who is going to benefit from this GMO and tell him now, you see, if you want to benefit, you must do this, but it must also be good and honest. Thank you.
Thank you for the observation. Professor Tuguma, you have a window for collaboration. Next. I hope I am gender sensitive. Okay, this will be the last.
Thank you so much, Mr. Modulator. I'm Dr. Medad Twinamasko Katonera. I'm the Director of Innovations and Technology Transfer at Mbarara University of Science and Technology. I would like to thank our panelists for the great input to the legislation framework in this country. But I would like to point out a few issues that you can respond to, but also which make a very fundamental contribution to our discussion today. The first one is the lack, there's a big disconnect between regulators and academia. And I find this a bit challenging, especially in our developing countries' landscape. It is unfortunate if all the scientific evidence that has been justified is presented in this room and just ends here. And what happens outside becomes the other business of the regulators, but also the politicians. So how best can we create a forum where there is a periodic or regular engagement of regulators and academia? Because that's fundamental. It will blend what evidence is provided by the scientists and what regulators always do, especially when they implement based on hearsay, not facts. Number two is on the issue of science communication. This is pertinent, Mr. Modulator, because I want to believe that there's a lot of infodemic outside there, especially on vaccine. We have presented a number of the GM and vaccine scientific justifications, but our people there are still hesitant on the uptake. So we need a very strong science communication framework in this country so that we are able to condense what comes out from the public, because we believe that these are the end users. And if they don't understand, they will not be able to take it. But one of the challenges that has been that most of our policies and legislations are always left in the foreign language. And it is never translated in the local language for people to understand. But also, science is very complicated when it comes to communication. So there is need for a forum between media, because the media is a little bit of a darling to the public. How the media can be engaged, that can be another forum STI can be able to pursue, so that we have translated simple messages that can go to our people in the local languages. And lastly, Mr. Modulator, it's about the integrated approaches of these policy regulatory bodies. You realize that we have the problem of institutional silos, where NFA, NEMA, NBA will be having particular laws, but these are not aligned. And sometimes there are disconnections and contradictions. So I think that is something our panelists can comment on. How do we bring these bodies together so that we harmonize our legislation platform and environment in this country?
Thank you, Dr. Twinamasko. There is a lady there, then the last one will be this, then the panelists will summarize, give concluding remarks, respond or nod their heads at those who have observations.
Thank you, Chair. Thank you, organizers of this forum and participants. I want to say that it has been good. We have received good communication on biotechnology and the biosafety chain of action in implementation. We realize that there are many good things, positive results that are coming out of research.
However, we realize that there is still a fear amongst us or the public on the threats that could arise from biotechnology, especially the genetic modification of the organisms. I would want to ask or propose that we have a monitoring and evaluation team or body that is independent, aside the scientist forum of presentation that keeps bringing out this information, especially on the possible threats and the interventions of these threats, so that we can have a confident public reaction towards
Thank
our
you very much.
regulators.
Thank you very much. Please bring the mic here. The last person, then these people will summarize, give their winding remarks. Time is not on our side, but Grace Nambati, your doctor, will address your fears according to your generation.
Thank you very much. Thank you all the moderators and discussants. I just want to make one point that we need a place in the communities where our people can find this information of biotechnology. And that is, I would like to propose that each district should have a library where information is deposited.
Thank you so much for that suggestion. So back to our panelists, starting in no order. Dr. Ongol, what are your, Dr. Ongu, oh, Mr. Ongu. I'm used to Dr. Ongol. So Mr. Ongu, what are your concluding remarks and recommendation?
Yeah, it's just to point out that concerns will always be there, and biotechnology is an issue, and that means there are several options that different people are providing to come up with a solution that biotechnology is offering. And they are not about to stop engaging what Dr. Okwaro called politics. And as he recommended, I think our scientists should come out firm because they know exactly, they know better the techniques of biotechnology. And also on public engagement, there's a challenge of going to the consumers, and yet we still don't have this product. When they say, show us the product, the yield and all that, but I think public engagement will be improved if we get to have our product released for farmers.
Thank you so much. Before I call Professor Arthur Tugume, I would like to say Philip Chemonges and OFAB usually encourage the media to write about biotechnology and biosafety, and their awards, annual awards that have been disrupted by COVID. Maybe he will talk to us later, or we shall circulate it online. Professor Arthur Tugume.
Thank you. I could comment on one or two things. One, thanks, Dr. Ebuku. I need to get in touch with you because I'm passionate about communications. It is the other day when I decided that going forward, every paper that I publish will be having a PR. No discussions, no negotiations. If you're under me, we'll always do a PR and try to simplify the jargons out of Krebs cycle and all that type of stuff. So we want to make sure that this one actually gets an impact, not only for me, but let it be a national, take a national shape. When we created Bachelor of Science Biotechnology, we had a lot of problems. And everyone who is in this room now agrees that we were seeing many, many miles ahead. And that was in 2006. So I like the vision that we have that going forward in the next 10 years, we should not be worried about who is going to communicate what in science. We want people who know what to say and when to say it and how to say it. And I want to really get in touch with him together with my other fellow deans who are interested and other participants, we can be able to put together something that can be able to help our country. Number two is somebody who mentioned about the science of God. You know, people think that God is there somewhere confused, not knowing DNA, not knowing RNA, these molecules, the DNTP is not. That's not true. One of the best scientists I know outside God is Daniel in the kingdom of Babylon, who was leading the whole team of people, and he ended up being like a prime minister, and is recognized as one of the most articulate and perfect and efficient scientists, to the extent that his work put everyone in the shades. So we are not saying here that we are going to be gods, not at all. How do we manage nature? I don't want people to begin blaming that biotechnology is going to cause extinction of species. You human beings, you have already caused extinction without biotechnology. Check about your climate, check about your environment. If anything, it should be the other way around. And I remember immediately a man called Jacob, who worked out a method of breeding animals and made sure that the animals of the trait that he wanted are the ones which he got. This is in the Bible. What is that? Or maybe we are not yet recognizing that these things are actually we are talked about. So what are we trying to say? We are trying to say that the means are good, but I don't know who voted against COVID-19 vaccine. And I don't know whether there is anyone here who can refuse if the government wants to be vaccinated against COVID-19, which is a GM technology anyway, that you can refuse to be vaccinated. Why are we discussing things, some of us on which we have no competence about to discuss? Why don't we let those who know what to communicate or what to do say them? I mean, you come and challenge President Museveni that you are against COVID-19 vaccine in Uganda. You try to do it, and you will see what will happen to you. So because we allow everybody to contribute, people even come and sit on your back, sit on your toys, they kick you. Can we divide where the boundaries are? Let the people who are, and one of the African problems is that we want to be everything and anything. So you are a farmer, you are a medical doctor, you are a politician, you are a religious leader, we want to be everybody and we want to commit on everything. So, but if we allow specialization as a result of industrial revolution, so that the sciences, let's do the science, those who can communicate it, let them communicate it properly. We will be able to move forward. Thank you very much.
Thank you so much. I said, you know what to say, where to say it, and when to say it. The young people have a concept called Gamba Nobu. So, Mr. Abatoloka, I hope Jacob Komakech's question has been answered. Dr. Akankwa Bariyega said God is the best scientist, and for you are saying we are working against God's science. Abatoloka?
Thank you very much, Mr. Chair. Let me start from Mr. Komakech himself, as Professor has noted.
Yeah.
The country has been conserving, especially on the plant side, since 1892.
Get closer to the microphone.
We've been conserving materials in Uganda on the crop side since 1892. That's more than 100 years. Way before we even thought about genetic engineering. So preservation or conservation of materials is should be part of the process with or without genetic engineering, because everybody knows that genetic materials are super important for our future. You asked a question about intellectual property rights. We shall discuss that. We will have more time to discuss that. But we don't expect any costs of seeds to be higher than normal, than usual. But since you sit at the UNCST, we'll also welcome you to our office at OFAB, our office at UPBC, and the reception. We have all this information placed there. We'll encourage you to have it. We also have some in the stalls out there. We've answered all these questions because we've been dealing with them for the last 26 years. And I think the others from Dr. Medad, thank you very much. I think the UNCST can take up the matter of addressing coordination among regulatory agencies together with the STIOP, but especially for biotechnology. I think UNCST is very well placed to handle that matter of coordination. Dr. Ndambatia, I think your comments are very, very welcome. We need to be mindful of the types of generations we are dealing with and segment our audiences. So I think no one will complain that this is not valid. It's a very good comment. I'll just nod my head like this. And then Professor Mutumba, thank you very much for the suggestion. We'll need to find a way to have the right information for this audience at the district level. They are critical, but as Isaac also noted, until products are ready for them, what are you going to tell them? Remember, stakeholders like farmers and consumers care about what is in it for them. Those are the benefits, and also any concerns they might have. But if there's no product, how are you going to tell them about the benefit or about a product? Vaccination, we're able to, for COVID, we're able to tell them about the products and the benefits. It was so easy.
Thank you
so
so
thank
much.
you. I have one concluding remark. As a country, we need to factor in communication and continue to do so at research level, however early, at regulatory level by the regulators, and definitely, as the last presenter asked, the second last question came, post-release of technologies, we need to find a way to continue monitoring and engaging the public and seeing if there's any potential issues that we need to address as scientists and regulators and the government. Thank you very much.
Thank you, Herbert. And finally, Mr. Philip Chemonges.
Thank you very much, moderator. I will still want to start with Mr. Kumakej. I think this technology has been misinterpreted in some way. When you look at synthetic biology, it is going to be the only savior from extinction. Because you see what controls a characteristic, or what controls something is at DNA level, at gene level, and at basis level. And with synthetic biology, we're able to identify things to the basis, A, T, C, G, and that is where characterization starts from. So biotechnology is a tool that will save us from extinction. So it's not a disadvantage, it's an advantage. So that's what I want to bring forth. And then the other thing I would like to bring forth is, like I told you, OFAP is a communication arm of the technology. And we have had media awards. The purpose of media awards is really to promote excellence in science journalism and appreciate contribution of journalists to promoting agricultural technologies or biotechnology, and reward the roles played by the media. So this year, we will have another set of awards. The call is already out, which will run from September last year to September this year. So the media has a lot of time between now and September to do work on science and on biotechnology and submitting your papers. You can go to UNC website, you can go to our pages, and you can get the information there on the details on how to apply. It's just something to stimulate still the debate, because the fourth estate is still important.
Thank you so
much. And my parting remarks is this technology is not about to go anywhere. Instead, it is improving every day. So Uganda should not afford by any means to be left behind. I think God will judge us harshly if we are left behind out of this technology, because it is where the future is. Marchers will say this is where the future is. It is not going anywhere. Thank you.
Thank you so much. Biotechnology is the tool to save us from extinction. And for those of us who want to avoid harsh judgment from God, let's go this way. I would like to thank everybody, the CEO from National Biosafety Management Agency, Nigeria. Thanks for your patience and time and your insights. Thanks for our presenters, thanks our panelists, thanks the audience that contributed very insightful ideas. And we apologize for rushing you through this. We pledge that next time we might allocate it more time. I thank you so much and wish you a very delicious lunch. Thank you, Deborah, for allowing us to go through this.
Thank you, Chair, for moderating this session. No, presenters, please don't leave, because you'll have to take a group photo just before you go away. And that will be after my brief announcements. Thank you, participants, for your patience. Please give a round of applause to our session chair and panelists. We apologize again for rushing them through the session. We have a long closing session, and we are expecting VIPs, I think, who are within the area to be part of the closing session. So we wanted to have a quick lunch and then get back and have the session going. The session after this one, which is the session 8 on biotechnology regulatory framework, and the closing session have been combined in the interest of time. And they will be chaired by the Executive Secretary of Uganda National Council for Science and Technology. Yes, and in that we'll have the closing also by the chief guest, who is the Vice President of the Republic of Uganda. Our minister will be with us in the afternoon session. So let's prepare to go and have lunch in 45 minutes and be back here in good time. Thank you, panelists and session chairs. Please move forward and take a group photo as you're all invited to lunch upstairs as usual. Thank you. I'll see you later. Yes. Please. Yes. Thank you. Hey, okay. Oh, I forgot. The afternoon will be moderated by Mr. Musa Kwehangana, the Biosafety Officer.
Biotechnology came in as a very important way of safeguarding the population, especially as a result of population increase. It became important to ensure that is there sufficient food for the population? Is there sufficient energy for the population? Is there sufficient shelter for the population? Is there sufficient clothing for the population? With an explosion of the population, it became necessary to ensure that normal formal agriculture could not provide food, clothing, energy for this export. And we all know that human beings have for a long time used living organisms to benefit themselves. Living organisms have been used in the production of beer, production of ajono, tonto, wenge bigere. All that is biotechnology. Sour milk, production of yogurt, fermenting of food. So all that is biotechnology. So it's not a new thing. The only difference now is how to enhance biotechnology at a genetic level. So, and that of course led to vaccine production and also led to drug production. For instance, the drug against diabetes, insulin, can now be produced using biotechnology. Is now the way of production of insulin. In the past, they used to produce it from pigs. and from sheep. But that was not sustainable. Large scale production was not possible. So we're now looking at how can we produce drugs using bacteria called E. coli, which if you introduce an insulin gene into them, they can produce the insulin in large quantities. That's what's happening in the world now. For plants, tissue culture was very important for production of clean material. Those seeds which are free of disease. So that when you plant seeds, you know that these seeds are safe. So clean planting material, as well as production in plants, production of disease-resistant plants. So all in all, biotechnology has been very useful in increasing production. That means you can now cover large areas of acreage without necessarily suffering the costs of labor. Right now in this country and in the world, especially the third world and in Uganda, the biggest problem we have is labor, to weed gardens. You go to the rural areas and you will see people can open land, but they will fail to weed it. In the developed world, acreage has been increased because they no longer weed. They spray and all the weeds die. So the crops must be resistant to the chemical which is used for spraying. And only the weeds will die and the crops survive. So you can find one man has acreage from here up to Ginger. Biotechnology is the reason why the prosperous world is surviving better than us. And so the question is, we in Uganda, what are we doing? What are our scientists doing? Why are our farmers suffering? What are our universities doing? What is government doing? And that's precisely why biotechnology came on board, not only to increase production and productivity, but subsequently to reduce poverty. The more you can produce, the more you sell, the better off you are. And this goes back to the Millennium Development Goals, where poverty reduction is a priority. And you cannot do that if you are not self-sufficient. Food security, again, hunger reduction, Millennium Development Goal. How will people survive if there is not enough food? So, food security or hunger reduction is a priority as a result of biotechnology. Lower cost of production. As you know, that to produce, you need to control pests. You need to control weeds. You need to make water available. Can we afford irrigation? Can we afford to fertilize soil? Can we afford to control pests like the locusts and so on? For us, using biotechnology, if we had a capacity to have plants which, if locusts ate those plants, they would die. That's biotechnology. Instead of trying to do all these things, which we have had a problem with. So indeed, food security, lowering costs of production, and lastly, reduction of use of pesticides. The more pesticides you use in production, the more you destroy the environment. And so if you reduce spraying, these days when people are growing, and they're growing maize, they're growing anything, even oranges, you have to spray 2, 3, 4 times. Where does those pesticides go? They go to the soil, the water washes them, rain washes them, rainwater washes them, they go to the water system, it ends up where we are, ends up in the food chain. It's all very dangerous thing. So if we can have plants which can resist pests, we do not need to spray them. The moment food is owned by someone else, you bake. But if you own your own food, you can eat it any time you want. You can do what you want. So do we own our own production? Do we own and do we know the biotechnology which is used in crop science, in animal science, in human science, in
I had the opportunity to represent Uganda in the negotiations under the Convention on Biological Diversity, whose main objectives are the conservation and sustainable use of biological diversity, and then the fair and equitable sharing of benefits arising from the sustainable use. Now, Uganda, of course, ratified the Convention on Biological Diversity, which was adopted in Rio de Janeiro in June 1992. And under this convention, there is a provision. There was already a concern globally about the developments in modern biotechnology or genetic engineering. And some people already thought there could be risks that could undermine, arising from the use of modern biotechnology, that could undermine the conservation and sustainable use of biological diversity. So under Article 8, subsection G of the Convention on Biological Diversity, parties to the convention were required to put in place mechanisms at national level to manage and minimize the risks to biodiversity that could arise from the development and use of modern biotechnology that arise from genetic engineering. But of course, this requirement was there. But at that time, this was 1992 and the early 90s, the level of knowledge and understanding of the products of modern biotechnology was still very limited, especially in developing countries. Now, the same convention in Article 19, Section 3, Convention on Biological Diversity, put a provision. They knew there would be shortcomings at national level for individual countries to go it alone. So they put a provision that parties shall consider the need for and modalities of a protocol to especially govern the transboundary movements of living modified organisms resulting from modern biotechnology that may have an impact on the conservation and sustainable use of biodiversity and taking into consideration effects on human health. So in 1995, July, the Convention on Biological Diversity convened a meeting of all parties to the Convention on Biological Diversity. Even non-parties attended as observers. This is a meeting of experts on biosafety and biodiversity to consider this need for an international treaty or a protocol. The meeting was held in Madrid, Spain. I had the honor to represent Uganda in that meeting. And after 5 days of deliberations, the consensus was that indeed there was need for a protocol, an international treaty to govern the transboundary movements of living modified organisms resulting from modern biotechnology that may have an impact, a negative impact on the conservation and sustainable use of biological diversity, taking into account risks to human health. Now, this working group made a recommendation to the second meeting of the Conference of Parties that was to convene the same year in November, I think November 1995 in Jakarta, in Indonesia. And the meeting of the Conference of Parties to the Convention on Biological Diversity, having considered the recommendation from this working group on biosafety, established an open-ended working group on biosafety to negotiate and elaborate this international protocol. So the negotiations took some time, and they were supposed to be concluded in the Colombian city of Cartagena, 1999, January. Then, but for some reasons, there were some disagreements with some countries not agreeing with what was the desire of the majority of the countries of the world. So the negotiations were suspended, and there were informal consultations that went on in between, both at the Africa region level and at global level. After some consultation, consensus was built that it was time to resume the negotiations. They were resumed in January 2000, and then the final text of what was called the Cartagena Protocol on Biosafety was adopted in Montreal in January 2000. And because this protocol provides for many things, but including the advance informed agreement, which makes sure that before any living modified organism or genetically modified organism is transferred into a country or released into a country, especially if it is coming from outside, the country must be consulted and must give its consent in writing. That is what is called the advance informed agreement. Then, of course, there are also provisions on conducting risk assessment to make sure that is to inform the decision on whether or not to allow the living modified organism should be allowed in the country or not. Then, of course, risk management. If there are some identified risks, are they manageable? What risk management procedures is the country going to put in place? Are they sufficient to contain? Yeah. Now, as all these things were going on at international level, the Uganda National Council for Science and Technology was not closing its eyes and ears. They were seeing what was going on at international level. And in 1996, some prior consultations took place. A workshop to consult key experts. In 1996, a National Biosafety Committee was put in place, mainly to advise the country on how to deal with living modified organisms, and also to draft guidelines on how to deal with applications of living modified organisms. And as soon as this committee was formed, they also identified a gap in capacity. So they also recommended that we need to build our in-country capacity, not only to evaluate living modified organisms generated from elsewhere, but also to be able to identify our technological needs as a country and be able to generate our own biotechnology-based products that answer our development needs as a country.
For a long time, we have had what we call the Biosafety Committee, and that was responsible to ensure that the work we do in the labs is done in a safe way in terms of how people dress themselves up, how they dispose of their waste, how they dispose of their chemicals, how they dispose of their radiation. And we had a safety committee. But there have been more developments. And now we talk about the Institutional Biosafety Committee with the new developments, especially the developments around biotechnology. We are working with a lot of organisms that have been, where genes have been inserted, and also globally and internationally. I think now there's a more talk about how do you regulate genetically modified organisms and recombinant DNA technology. And that, of course, now we're talking about pathogen economy. And I think if we're going to use our pathogens, Africa and Uganda, as many of these pathogens, microorganisms, diseases that we have, the president and the government says, how can we use this to build our economy? By being innovative, we innovate, we produce products that can be sold globally and internationally. It keeps saying that we sold our Ebola to others. Instead of us making a vaccine, others have made a vaccine using Ebola is ours, but others are making vaccines. We don't use pathogens only to treat and manage people, but also have in our mind how this can also bring in foreign exchange and money. Yeah, all of us, we are looking forward. Like my colleagues in agriculture, there are products which they can't release. They're still in the lab because the bill has not yet been passed on the GMO. I think this needs to be worked on very quickly. It can also affect us if we have products that we want to release or to use. If the bill is not passed, then it will hinder progress. I do thank the government and the Uganda National Council for Science and Technology for taking this on, mobilizing and making sure that we have a functional National Biosafety Committee and also on the ground Institutional Biosafety Committee. It is very good. We are prepared now. As we said, we are not moving back. We are moving forward. This technology has come. The technology, we are using it already. The technology will expand. So I really thank the National Council and I hope that they will continue to support both at the national and institutional level. And also I thank them for the partnership we have created.
As you know, that research work on genetically engineered organisms is a regulated process, which is the regulator is the National Biosafety Committee, which is housed under the Uganda National Council of Science and Technology. So as this is a regulated trial, I have worked closely with the NBC and the UNCST over these years to ensure compliance to biosafety regulations. These trials are approved with the special conditions which vary from one crop to another. And for bananas, there are specific regulations that we have to comply with, and we are monitored by the NBC for compliance to these conditions which has been given to us. So in my experience, there has been a lot of learning, both between us as developers of the technology and also the regulators. But overall, in my assessment, I think it has been of much benefit that that interaction happened, because there are things which, as technology developers, we may take lightly or may think it is not a very important issue. But when we get these people who come from the regulator side, those small issues are actually very, very important in our experience. And it has helped us to grow and improve in the way we conduct research with genetically engineered organisms. So we are grateful. We are very grateful to the regulator, National Biosafety Council, and the inspectors who come from there, who sometimes come from different sets of organizations. We have representatives from universities, people who come from the legal department, from the Ministry of Constitutional Affairs. We have representatives who come to inspect us. We also have people from trade. We have people from health who are inspectors who come here. Others are veterinarians. So when they come here and observe and make their contributions to this kind of research, we benefit from it and makes us answer the concerns of the general public with that representation of the NBC. So we have greatly benefited and we have learned, and we appreciate the role that they play, just as they also understand the roles that we play. So we have worked together very well over the 25 years and developed a lot of capacity in this area. We are able to carry out genetic engineering with compliance in any area of Science, Technology, and Innovation.
Do we understand biotechnology? Do we understand the science? Those who are doing it for us, are they doing the right thing? So our scientists must be well trained. We must know their capacity. We must know who's competent. We must know what comes in. We must test it. We must have good laboratories. And we must have very good SOPs. That's why the National Council for Science and Technology was playing the role of a regulator. When the Biosafety Committee started, one of the first things we did was to send people to do PhDs. For instance, the Deputy Director General of NARO, Dr. Baguma. He was one of our first persons we sent to Sweden, Uppsala. And a number of others, we sent them to train. And Baguma is an excellent biotechnologist. He understands science very well. The National Council for Science and Technology has to be aware that in science, in everything, there's what's called the continuum of knowledge. Continuum of knowledge. It does not stay static. New things come up every day, and you must be at the cutting edge of knowledge. That's why at the PhD level, to train those people, it is at the cutting edge of knowledge. What is happening in a laboratory in New York, in Japan, in the UK, in Cambridge, in Tokyo, you must know it when you are here in Makere. Cutting edge of knowledge. And you bring it to bear to your graduate students, PhD students. And that's why those people are not to be joked around with. Treat them well and they will give you the best that they can give you. So the National Council for Science and Technology must be aware that, I know they're mainly emphasizing policy, but policy is not a, policy is just a guide to action. That's what policy is. And the law regulates. From policy, you go to law, you can regulate things, so-called jurisprudence. But you must be aware that there are new things happening every other day. And in an explosion, there's an explosion of knowledge. So much so that we must continuously transform. I think going forward, the National Council for Science must make NBC a formal, regular committee of the National Council for Science and Technology. And by the way, it's the best functional committee. But it is not one of the statutory committees. It is not. Unfortunately. It's not one of the statutory committees. But it should be. And that's my hope, that the National Council for Science and Technology ensures that NBC becomes a statutory committee, they strengthen it, they ensure that it is active, and above all, they are current.
I have worked as a member of the National Biosafety Committee of the National Council of Science and Technology for about 5 years now. And my contribution is to see biosafety is about the safety of the people. And that's why it is very important to have the people you are saying you're protecting on the table. The science, yes, but increasingly the science is realizing that the social science is even extremely important. I have, for instance, participated in the NBC trials, like for cassava, like for potato, and even when you look at the testing, one of the things we struggled with was who is testing? Who is testing? Is it the consumers or the people within the agricultural area that has been restricted by the NBC? And the way, if you want legislation, you see consumers include members of parliament. We have struggled to pass the GMO, what they call the GMO bill, the genetically modified organs bill. And the problem is that the scientists have been speaking alone. And people, when they look at products that are not familiar, they tell you that is GMO. And that has swept the whole country, and nobody wants to listen to that bill. But it's important to get Parliament and the community to understand that this is also for their protection. If we don't have a bill, if we don't have a law, that means that anybody can give you anything and you will not be able to hold them accountable. So whatever the scientists are doing, it should be subjected to the community, to the users, to the people. Acceptance is an important principle of science. It's not even just a plain art. That the products you are working for, that the effort you are putting in is seen by the people as in their interest, and that the people have an input in whatever you are doing to be able to come together at the end and say, this is cassava, it is not harmful, it is in my interest, and even if it is a genetically modified. People should be able to understand that, for instance, whatever looks different, the big bananas, the big mangoes, the big onions, the big apples, people say that is GMO. You'll be surprised to see that even members of parliament say that the different one is the GMO. So it's very important to bring all of us on board and to have a representation at the NBC in order to be able to get acceptance and to move together as a country. If you even look at the vaccines, for instance, we have a lot of resistance for vaccines because people don't understand it. People have not been part of it. People here, and there's a lot of information. That is the other danger. There's a lot of information out there that is not science, that is misleading. And that is the one that most people get. However, I think that those of us who represent, for instance, the community at the NBC, we need to be able to have a forum to speak to the bigger audience of the community. When you represent people, you should be able to go back to them and tell them. I, for instance, had an input into the GMO bill at Parliament. I was asked by Parliament to go and make a presentation on what we think as consumers. And it was very enlightening, even for me to understand where they are coming from and to go back and report at the National Biosafety Committee and tell them this is what Parliament has said, this is what they think, these are their concerns, and they need to be addressed.
For me, what is a law? A law is an instrument of government which will regulate and enable the practice and execution of certain principles consistently and easily without interpretations being so varied. You've just mentioned if we had NEMA say something, we had the Ministry of Agriculture say something, Wildlife say something, another body say something. How many laws are those already? And whose interests are they? A law galvanizes interest into one, okay, and allows unison performance and allows things to be done at the same time. It is true that there are many laws in terms of jurisprudence. There are many laws which touch something about this and that and that. But the law we are talking about technology biosafety law is supposed to galvanize, uniformize, standardize, and ensure that the practice is the same. Because the environment, productivity, health, economics, because trade is important, economics comes in, and all those things are really actors at the side, implementing certain things. But there has to be someone central. It's just like we have many districts in this country. Why do we need a president? Just one president. You must oversee and regulate everything, ensure that what happens in all these districts, and moreover, even in those districts, there are RDCs. Then there are other small local governments and so on, working together. That is what that law is supposed to do. The biotechnology law or biotechnology biosafety law would enhance the collaboration, unison. It would ensure that we would reach the destination together quickly and together. And that's why in the biosafety committee, there's a member from NEM, there's someone from trade, there's someone, all those people are members of the committee, biosafety committee. There's someone from agriculture, from health, from universities, from research institutions, from farmers, from the public. In terms of all those people are brought in, and they should be able to go and feedback. Those countries which have not had a uniform law like the US, there were already laws, and I'm telling you the way those people work, you'll find that there are subcommittees which are essentially like our biosafety committees. I'll tell you that you'll find there is a committee on poisons, yeah? National Committee on Poisons. Then you'll find there's a committee, subcommittee on food poisons. There's a subcommittee, drug poisons. They all work together and they eventually report the same body. So the methods of work in those countries is because they have systems working. For us here, we don't have systems. People want to work on their own. And the laws are the ones which will bring systems to bear. Okay? So long as we do not have one law which people will follow. Just look at the roads. If they said you can drive on the right, you can drive on the left, you can drive zigzag, you can, so long as you reach where you are. One would want to ask the question, why do we need a road safety law? When there are panya roads, there are tarmac roads, there are highways, there are what? That has to be uniform. So those countries have these type of biosafety laws but they have been worked in such a way that in their systems they are working. Okay? But when you are a new entity, you don't have to begin doing what they are doing. You now start where they are. Because even the Americans will eventually have a biosafety law. They will eventually have it. Yeah. But because the other things are now working, they say, oh, what the heck, we can manage. Because for them, there is public acceptance. They trust their scientists in the first thing. In the US, they trust their scientists. For us here, we don't trust our scientists. And you know why we don't trust them? We don't own them. The research work in this country is owned by foreigners. Uganda government must take it by the bull, take the bull by the horn and own research in this country. The research work in Makerere, I was at the center of it. I was the director of graduate studies and research for 5 years in Makerere, from 1999 to 2003. 5 years. I negotiated from money from NORAD, from Sida Sarek, all these things you hear about. This Seiford, when we send the Bagumas there, I was the director of graduate studies in Makerere, ensuring that we look for money. I got $15 million to bring to Makerere. Government was giving 100,000 shillings by then, 1999. 100 million shillings. That's the money I was getting from government in Makerere to do research. That SIDA was giving 5 million, 10 million, 15 million. Look at the impact. You go to Makerere, or the medical school, which was a serving institution. Now it's a research-led institution. Go to agriculture, go to engineering, go to vet, even social sciences. That was my influence when I was there, looking for money to ensure that we own research in this country. If we do not own research in this country, we are deceiving ourselves. Other people will own it, they will tell you what to do. They will tell you what to do. They will have their own interests. We rather start small but serious. Serious, consistent, reliable, always there. A scientist does not need a lot of money. He needs small comfort and to do his job or her job consistently, without worry, without disturbance. They will sit in those labs up to 3 a.m. So long as they have their house, they have their transport, they can take their children to school, they can eat well, those small things, which is not too much. And here our best scientists are in the village. They're in rural areas. They are not being brought to the center. For me, I'm a village.
NARU's mandate is to provide oversight, coordinate agricultural research in the areas of crops, livestock, fisheries, and forestry. In the year 2003, His Excellency, the President of the Republic of Uganda, opened the first biotechnology lab in Kawanda. And basically at that time was to look and navigate ways on how basically can improve on a breeding kind of program. And as we all know, when it comes to genetic engineering, it's one of the toolkits that respond very, very well from anecdotal data that we have. You're looking at how best you can increase, say, like yield. You're looking at the tool that can be used to empower the plant in order to make the plant actually more resilient to the environment, becoming more so like immune to pests and disease kind of infection and infestation. Then again, the element of fighting what we call the hidden hunger, lack of micronutrients, especially vitamin A, zinc and iron. And again, we also in cassava, we do realize that cassava too is a very rich source of carbohydrates. And one of the fundamental areas is like one of the largest consume root crops, especially to supply carbohydrates, rank after bananas in this country. When you look at the quality of the products, we realize actually you can also introduce the genes for beta carotene to make it look yellow like carrots. And by so doing, you're more so like looking at aspects of fighting lack of vitamin A, just like the way we did it to sweet potato to come with orange fresh sweet potato. But again, we also wanted to really see the versatility of this new tool called genetic engineering. When it comes to genetic engineering, modern tools have come up. Today we are talking about genome editing. And now the key question has been posed, is genome editing part of also like genetic modification, which should actually be considered under the genetic engineering regulatory authority or the biotechnology, biosafety kind of activities to be passed. And here we're looking at non-inclusion of any foreign matter into the plan itself. I think it's being very unfair to our scientists. The element of even regulation again, you look at aspects. In the event you don't have the law, and then you happen to bring somebody else, bring up any genetically modified organism here, there's no law to stop that person. But if you have a law on biosafety or biotechnology, then you can regulate. You apply all the possible avenues that are there to enable things that we have in this country. But to me, I think we are still being very, very blind in one or another. So to my plea is to the relevant authorities. In the event we find maybe some deficiencies in the current genetic engineering regulatory bill, why don't you give it back to the scientists? We revisit it and see how best we can add value to it. And then again, we also look at other emerging technologies that are coming up. But to me, when it comes to technologies, we come up with toolkits to see the easiest way you can come with a product within a very short time, very efficiently, low cost, and then again, its effectiveness for the purposes it's intended. Otherwise, having no law at all, I think it's a problem. They're just on a time bomb.
As a biosafety expert, I really appreciate the role of biosafety, ensuring compliance with biosafety. And I really appreciate the importance of having a biosafety framework. And by having a biosafety framework, I mean, we need to have the laws, we need to have the regulations and guidelines, we need to have the institutional frameworks, we need to have the capacity, and we also need to have very good communication systems to support the ongoing work. And so as a person with this experience, I really want us as a government, as a country, to ensure that we support a comprehensive framework. There is the option of using existing laws. So let us see how we can provide for this biosafety in the existing laws or in a new law, which will then guide the regulations and the guidelines that are going to be used for this technology. One thing is for sure, we are not going to deal away with the technology. The technology is already with us. So what we need to do is to make sure we have the right legislations, we have the right guidelines and regulations, we develop the capacity. And as a country, we have done a very good job with capacity building, because we even have universities that are training people in biosafety, in biotechnology. We also have a number of scientists that have gone out and come back who can actually support this system. But we need to make sure we greatly improve the way we are communicating with the people. All our stakeholders, starting from the policymakers all the way to the farmers, and help them to appreciate the importance of biosafety and the importance of having the systems that will ensure that we comply with the requirements of the biosafety, not only for us as a country, but also because some of these products we are working with, they are going to end up on international markets. So let us ensure that we comply with the biosafety to protect our people, to protect our environment, but also to boost our competence on international markets.
The ministry has been participating in a number of activities that are led by the Uganda National Council for Science and Technology. And these activities mainly relate to issues of genetically modified organisms, and specifically the crop subsector where we have our competences. And here when we talk about the competences in the crop subsector, we mainly look at the phytosanitary concerns related to biotech products and crops for that case. And for the case of the crops that are under test, and for the case of Uganda, we have a number of crops, and these crops we have participated from the point when the trials started to where we are now. And what we do, we provide support, we provide inspectorate services for the inspection of this confined feed trials and all the trials that are ongoing in the country. And we, as I earlier on alluded to, our main concern is phytosanitary. And phytosanitary, we look at the plant health concerns. So in simple terms, these are plant health concerns. And the plant health concerns under the provisions of international sanitary and phytosanitary measures, what we focus on is we consider the genetically modified organisms as to have potential plant health effects on other commodities. Policy is not keeping pace with science. And I think that is the same message that holds with the that I would still give to the National Biosafety Committee. There are several innovations that are coming up. We have the gene editing technologies. I don't know in the provisions of the proposed legal framework whether we have those technologies, the emerging technologies that require that in the normal circumstances you may not have considered as biotech, but with the advancement of science, there are several technologies that are upcoming, and we need to keep abreast with these technologies. So a message to the National Biosafety Committee: we congratulate you first of all that you've done an excellent job in the past 25 years. But as we look at the future, and especially with biotech, there is a lot of advancement in science, and we need to really up our game. We need to make extra efforts to interest ourselves in these new technologies and how best we can regulate them. I know we don't have a complete regulatory framework in place, and that's one of the biggest challenges that we really are grappling with as a country. But going forward, I would really encourage you to the National Biosafety Committee to really interest themselves in seeing how best they can work with researchers to churn out these technologies and have them available to the farmers.
Omukyaro kyarwe bitete kirukushango mu district ya Kyuruhura notonge kishani cyitendekereri government kurabiro ministry ya science na technologia heriyo mukombe kobwe rukuteba no mu kiroto cye2020 icyo kutungura ebyamakorero
kongera kutyaza abantu abashomera ibintu bya science abokwekozesa abokwesa
so neza kugira nk'amataji mushanju yahwire yakatandika umwaka gw'2019 tekeza ngo neza kutwara nk'imyaka 5 of course umurimo mwihanyu ngo no kuombeka nkegisente bari mbombeka harwe bitete ndijo mbaza kujombeka namamve konke mpango munonga neje harwe bitete mu district ya Kiruhura.
Project ejene yombekwa ba Avic International Company y'aba China kandi hari billion bishatana n'itane zera mare ho 2095 government kazeguzo mu ihanga rya China.
Employment creation is an aspect that we intend to address. Most of our people, engineers, scientists, you find that they don't have the skills that are required in the industry. They have learned the theory. Now, our training is to provide the skill required by the industry.
Project ejene ija kuhanga hemerumero kurengo mutwaro gumwe n'enkumibiri kandi ebe bagambirwa bwa Ministry ya Science na Technologia babire ne barambuza bebembezi ba district ya Kiruhura bebembirwa mu nyishengero wa Nyabushozi Wilson Kajweng.
Ngo mwana umutware Nyamitango mu technical school nari Fort Potro usanga ugo tine bwenge kandi ebintu byo bintu eby'Uganda rukushoreza mu munonga kandi abarikuba bashomere nkebi nubo batari kubonabonesa abazire babo kubaronera imirumo narabebembezi
mbwe ningira ngo abantu bakiruhura no kugira ngo betekateke kandi n'abanyarwanda note necessary abanyakiruhura bonka betekateke bije badevelopinge ninga bamayungu bine akatare akaryo katandika n'abana babo babashomese ebinde bakorenga tubakoza ku mass street n'abaronde mirumo
omukushijuma project yahika habicweka 30 harigana kandi ye ndijo ariyo neyombe ko muri namamve mukono yahika habicweka na mushanja harigana
project aliyo najikora konga yongeremo kureba ngo tiyatwara lot of time nale tiya delaying
ibra yemeramo mu ihanga riyemerira habicweka bibero no butonyeza butana harigana kandi cyumwa habireserekezo beke no kubura ubukugubura kumara eki government yarwangiserize genereye kurwanisa Kagure TV West. Omwaka gw'2020 gutaka hwireho Uganda kabe yine kiroto cyo kuzaharengo rw'amahanga gentasi ya rwagati cyete middle income status cyinte cyaremere. Konka government negira ngo yorokerere kwikirize kiroto cyo mwaka gw'2026. Ecyabahanga bari kugira ngo ncyete ngo kushagike by'amakorero.
Aba economists nibazirebero kuzikorera esente zabushati ziri ku butwari hanga tuhonde. Tine kuzitungira mu bintu ubumirimo services zitugabura n'ibintu bitiri kuhereza.
Ecyo kureberaho government ubwa hateriyo neyombeka amatendekero abiro mu ihanga garmeri yarwe bitete omuri Kiruhura mwene rya namamve erupshango muri mukono kirukwijja kweraho kushongora barabe n'abakoro mu makorero.
Bueno hano ecyo sendane ya namamve nituza kuteka we byoma. Tineho zibari kweta laboratories n'abashomesa.
We are trying to see how we are going to help our engineers, our technicians, our graduates to attain skills, practical skills required to drive industrialization, to create employment.
Ahakachiko ka boda ka Uganda National Council for Science and Technology obukabana kashijuma emerumeri yone yakorwa hitendekere ryarwe bitete muri Kiruhura kashanga nguriya kahabucwe kasha tunabashata harigana konkendwara ya COVID-19 ino kwebara banise mukandogwe bagendereye kweza mu mwaka gw'2023
muntu bija kweza umubire bwaco obatiro mu ndagano aha hamwe na namamve abo kuba hatitu yabucweka ashatu ndabashata hari 10 kandi no gwire butabe turaganire kukoramo nabwe n'ubwingana na ecweke kikozwe
amatendekere agagarukuza kumaraho sente zero kurenga omaro billion 350 nigashajikwa government ya Uganda kandi sente zimwe bakazeguzo mu government ya China. Itendekere ryarwe bitete shitamira hari hikaze rukwikana n'itano konka habasa kwija kuba hecetengo cyo kuryongera mu buhangu.
And the initial plan was actually to maximize the utilization of this land. It is a campus, it is a learning center.
Okay.
The learning center
I can't tell it because they're showing this.
Has to be big enough to accommodate the learners.
Abanyarwanda barukurenga 15 no barije ko bana bashongorwa omyoge tarukushana kuba hwera kongera hantasa yihanga eyo mukuru w'ihanga agize ngo neija kubeyemerera hari US billion dollars anani no mu kwezi.
The establishment of a multi-billion business incubation center at Namamve has taken shape, with the construction works now at 30%. Now, according to the implementing agency, the Ugandan National Council for Science and Technology, the center will collaborate with other institutions to nurture innovations into viable businesses that create employment.
The government, through the National Council for Science and Technology, is implementing the National Science, Technology, Engineering and Innovation Skills Enhancement Project that aims at positioning Ugandans through skills training. To this effect, 2 centers have been opened, one in Kirura district in western Uganda and another one in Namamve in central Uganda. The Namamve Center will help translate innovations into viable businesses for job creation.
We are already in partnership with the National Enterprise Corporation. We have entered into another MOU with the DIT, Department of Industrial Training, and we are entering into other partnerships with the private sector foundation. The first component is the National Science, Technology, Engineering and Innovation Center, located in Revitete. The second component is this.
This center of Namamve will also provide working spaces for scientists and innovators, and the aim is to promote industrialization and manufacturing to create employment for economic transformation of the country.
We shall be outsourcing business mentors to come and impart skills, business skills. We shall be having science economists coming to teach or to mentor the people that we shall be having here, to turn their ideas, their innovations, science-based innovations into viable enterprises that can create employment for sustainable growth, inclusive growth and wealth creation.
The National Technology, Engineering and Innovation Skills Enhancement Project is co-funded by government and Exim Bank of China.
Agriculture is the backbone of Uganda's economy, and it's the country's largest employer. Data from the 29
Okay. Members, you're welcome from your lunch. We shall be proceeding to the next session. And this session will talk about biotechnology regulatory framework and also the closing session. And these sessions will be chaired by our Executive Secretary, the Executive Secretary of Uganda National Council for Science and Technology, Dr. Martin Patrick Ongol. Dr. Patrick Ongol has specialty in these areas: food microbiology, food safety and quality assurance, dairy microbiology, application of molecular techniques in food analysis, evaluating immunomodulatory effects of food, among others. Dr. Martin Ongol once served as a deputy dean and lecturer at University of Rwanda, College of Agriculture and Animal Sciences. He also served as a senior lecturer and head food analysis laboratory at Kigali Institute of Science and Technology, and previously also served as a researcher at the Institute for Technical Research, Glyco Dairy Products. But before that, he also served as a fisheries inspector at the Ministry of Agriculture, Animal Industry and Fisheries, but also serving also previously at Britannia Products Uganda Limited as a quality control supervisor, and also a quality assurance manager at Victoria Fresh Foods Industries, and also a quality assurance supervisor at Clover Game Fish and Foods. Dr. Martin Ongol holds a PhD in applied microbiology from Hokkaido University, as well as a master's in applied biosciences at Hokkaido University as well, and is an alma mater of the great Makerere University. Dr. Martin also has a patent with regards to screening of lactic acid bacteria elements with regard to immunobalance. The patent number is FERM AP21009 and number FERM AP21010. So I take this singular honor and opportunity to invite the Executive Secretary, Dr. Martin Patrick Ongol, to take charge of the next sessions as we proceed. Thank you very much. Let's welcome Dr. Martin Ongol with another round of applause. Thank you, thank you.
Good afternoon, ladies and gentlemen, and welcome to this afternoon session, which is on the biotechnology regulatory framework. And we are going to have three presenters. The first will be from the Directorate of Biosafety, the African Union Biosafety Agency. The second one will be from Dr. David Fashimana, and also we shall have the third presentation by Dr. Asea Godfrey. So I would like to start by inviting the first presenter, that is Dr. Silas Obukosia. Dr. Obukosia is the Principal Program Africa, African Union Development Agency, New Partnerships for Africa's Development, ODA NEPAD, Africa Biosafety Network Expertise, and a coordinator of AUDA, NEPAD Regional Office for Eastern Africa based in Nairobi. And they have been working closely with the Uganda National Council for Science and Technology for over a period of 10 years. Previously, he has been very active in capacity building. He has published several scientific articles. And of course, he holds a PhD in molecular genetics from Pennsylvania State University. He has a master's in genetics and plant breeding and a Bachelor of Science in Agriculture, both from the University of Nairobi, Kenya, where he also served as a senior lecturer and taught for over 15 years in the College of Agriculture and Veterinary Sciences, and have supervised several theses at MSc and PhD level. He will be with us, I think, online, and if the online team are ready, we can start. Okay. Dr. Silas, are you getting us? Please, you can start. Thank you.
How many minutes do I have? 20, okay.
Okay, roughly about 15 minutes. So it will be great if you could make it within that time. Thank you.
Okay, thank you very much. Thank you, Dr. Martin Ogol. We have been with you for about a year now. And I want to thank the organizers of the conference, the Uganda National Council of Science and Technology, and then also the Minister of STI and the participants. It's a privilege to talk to Ugandans. I worked there for 15 years, sorry, for five years at a place called Mtinda, and we appreciate you. Two, I was born on the border of Kenya, and we knew anybody who went to university, if they don't mention Makerere, then they haven't went to the best school. So we appreciate the leadership of Makerere in education in Eastern Africa. Thank you very much. So my topic is just biosafety regulatory experience in Africa. And I want to focus on what is our experience and then emerging African Union policy and regional harmonization. Okay, so yes. So I will basically focus on those 6 areas and basically let's look at functional biosafety regulation frameworks. And then the word functional means it's working. So we look at how has it worked in the commercialization of first generation and second generation crops. And then look at the approach to emerging technologies, genome editing, gene drives. I may also allude to synthetic biology and also harmonization at COMESA and ECOWAS. I would like to impress on us that we, Africa, 55 countries have got a framework for socio-economic development for 50 years, starting in 2013. So we are almost 10 years in. It's got 7 goals and 7 aspirations and 20 goals here. And the vision is a integrated, prosperous, and peaceful Africa, driven by own citizens. Now we're talking about Ugandans and a dynamic force in the international arena. And also I want to emphasize that as we're looking at STI, there is an overarching Africa 55 countries science, technology, innovation strategy called STIZA. It runs every 20 years within the 50 years of agenda. So it's coming to an end. Again, we expect another 10 years one. What it does is that is the last point there. It tries to focus on how can STI contribute to agriculture. And I want to appreciate the speech by Honorable Monica Msenero Masanza. He emphasized where Africa needs to be. And I think that was a very, very excellent speech. Thank you, honorable member. Now, the genesis of biotechnology in Africa at the continental level came from a request by AU member states to have a technical policy direction for the continent, and none other than our own, the late Juma and Cesar Regini gave this report. It looked at how can biotechnology be used to solve problems of food safety, health, and environment, and identified its use in agriculture, crops, animal health, and forestry. And it's out of the need to have a safe application of technology that now the African Biosafety Network of Expertise was established here. And sorry but then the world has moved ahead here, and now we are looking at how can we tap knowledge. And I think the minister emphasized that knowledge as a tool, as a capital for development here. And so with the rejuvenation of ADA NEPAD with 4 mandates, One of them is looking as providing advisory-based services and technical assistance to AU member states in a knowledge-based. And so that's where we are focusing as an organization here. Now, we must appreciate the history of the genesis of biosafety. UNEP-JEF was the first to do continental biosafety trainings, then later program for biosafety systems 2005, and ABNA came in 2008. Yeah. But the question is that so if you look at our map now today, yeah, you can see all that green, the countries have got biosafety frameworks, acts. If you look at the yellowish, the pinkish, they have got no acts, but they are part of Cartagena. You look then at the only few are not part of CPB and also have got no legal framework. But the question is that what are these frameworks doing to help technologies move ahead in the continent? And that's where now our efforts come in. There was a need to make these frameworks functional by looking at policy legislation, capacity building, partnerships, and also to support African Union members negotiate at international level. Yeah.
Sorry, Dr. Silas, the slides are not moving.
Oh, they are moving on my side, sorry. Let me try and see how I can do it. I'm on the 5th slide here. Are they moving now? Yeah?
Not moving. Still, they are not moving.
Maybe I sent one, if you can present from that side, although it's a long version, but that can be maybe better. I can just try and go through very fast. Because I'm trying, on the side they're moving, but your side not moving here.
Sure.
I sent one to Musa. If you can present from there, then I can see how to summarize it.
Okay, in the interest of time, you can continue as they work it over.
Okay, yeah. So the work of PBS and the work of ABNA has been to build on the foundation of UNEP-JEF to make this functional. And functional means to have the legislation and the policy enabling the technology, to have implementing regulations and other guidelines, to have human capacity with the ability to do risk assessment and decision making and monitoring. So that has been what we have been doing, and in about 23 African Union countries. So we go to each country, assess, then based on the need, we've been able to build, fill in the gaps, and that has now resulted in what now we have next is a commercialization of crops in Africa. 3 years ago, there were only 2 countries, South Africa and Sudan, that had commercialized them. Today, we are talking of 7 countries. One of them being Nigeria as our leader, then Ethiopia, Uganda, Kenya, Malawi, Eswatini, and of course, including South Africa. And then the other countries that are almost coming forth, Burkina Faso is trying to get back, and Ghana is is now looking at the copy application. In other words, there has been commercialization. In other words, the frameworks are now becoming functional to commercialize product in Africa. The other aspect that has changed a lot is that the commercialization was based on single trades. Cotton now is about 5 countries in Africa. Maize is still in Nigeria, but is coming up in other countries in Africa. I think, yes. Then the other aspect now coming up is cowpeas. Nigeria has commercialized. Ghana is now looking at an application for commercialization here. And other countries like Malawi and Burkina Faso are coming on board here. Also, there are now crops in the pipeline. The other major change, which I can say in the last 6 months, was the previous commercialization were based on single traits like insect only. But now, the last 6 months, the African Union member countries are now looking at the stacked traits, with Nigeria having commercialized drought-tolerant insect resistance. Kenya is looking at the same. Mozambique is testing a similar thing. Ethiopia is working on the same thing. So there's been improvement here, but this just don't come. People are getting trained, the regulators, in how to approve stacked traits here, and that's a very big plus for our continent here. And the other things in the pipeline, rice, potato, soybean, they are also in the pipeline, and they'll have a big impact in Africa. When it comes to emerging technologies, there is a special panel of African experts established. Sorry, you can't see my slides, but they are looking at 10 emerging technologies that can give benefit to Africa. They have already delivered on gene drives, which I'm going to cover. They have delivered on drones. I won't cover the post out of the topic. They have delivered on microgrids, and now they're delivering on genome editing. So in 2017, there was a policy statement from Africa Union that the continent, at the continental level, we commit to invest in development and regulations of gene drive technology. And that is gene drive technology for control and elimination of malaria. The point there I want to emphasize is that it was not for all gene drives, but only for control and elimination of malaria. Why? Because most of malaria is affecting African Union countries. About 95% of the deaths is in 31 countries, but 51% of that is in Africa. So we have got a guideline and a policy guidance that shows us that we need to invest in this and be able to use it here. And one of the things I want to highlight from the guideline is that it gives a pathway for developing this technology from laboratory to commercialization, so that at each stage there is ensured efficacy and safety. And you don't move from one stage to the other one unless it's efficacious or unless it is safe here. So that is what we have here. The second thing is that on genome editing, many of you who are in Sharm el-Sheikh in COP 2018, there was an international moratorium to put together genome editing, gene drives, and synthetic, and have a ban here. At that time, our interest was gene drive for mosquitoes here. But then African Union member countries also asked for capacity in genome editing. So in 2019, ABNA, with support of external experts, held a training here. And out of that, ladies and gentlemen, I can say that two countries, Kenya and Nigeria, have developed the guidelines for genome editing, and the country is by the way fast-tracking genome editing in Africa. They requested for an overarching guideline, which also has been developed by Africa Union member countries, and it will soon be launched soon. Yeah. But what comes out of genome editing is that it's come out that some crops of genome editing will be equivalent to conventional crops. Yeah. And so those should be regulated under conventional acts, like the normal varieties we normally regulate. Yeah. It also comes out that some crops from genome editing may have a GMO at the beginning, but then it's removed here. So the GMO section should be under Biosafety Act. After removal, it should be regulated under the Biosafety, the normal seed acts, conventional seeds here. Lastly, a third category is genome editing that are GMO. Those should be regulated under the normal genome biosafety action. Maybe you can't see from my map, but of the major countries, African Union major countries crops, bananas, cassava, maize, sorghum, wheat, and yams. The map shows all of them all over Africa, but there is genome editing research going on in the bananas in Kenya, maize in Kenya, yams in Kenya, sorghum in Kenya. And they also say cassava in Uganda, but you can confirm for me, then cassava in South Africa and the wheat in Egypt. In other words, unlike biotech, GMO, where the African unions were spectators, this time we are fast tracking and we shall be among the first countries to adopt and use this technology for our development. Yeah. Lastly, gentlemen, the issue of harmonization of regulations or guidelines across the continent. If you saw the vision for African Union is to have integration here. And so there has been much progress in ECOWAS in the harmonizing regulations for biotechnology, and that I think has already been approved here. COMESA has done that also to some extent, but I think they ran short of funds, and so there was some hitch. But the interesting thing, among COMESA countries, Ethiopia, Sudan, Kenya, Malawi, and Eswatini have already commercialized GMO crops. Then when you come to SADC has been lagging behind also in that, and is behind ECOWAS and also behind the COMESA. But the interesting thing is that 4 SADC countries have commercialized Eswatini, Malawi, Mozambique, and Southern Africa. May I summarize and say that, sorry, you can't see my map, that biosafety frameworks have been developed in Africa for now a long time, yes. But there has also been a lot of interest from 2005 until now to make them functional. And they have been made functional, many of them here. And they're also practical. In other words, they have been used to examine certain biotech crops and they have commercialized them here. And so it's a matter of expanding and having a regional impact based on this year. It's also noteworthy that Africa is working on genome editing on key crops here. But the joy is that Africa is fast-tracking genome editing, and this time we will not be left behind. We shall make our own decisions and decide which one is good for us, and so that this science can transform our continent. May I underline that the speech by the minister is what is at the heartbeat of African Union. How can science be used to change lives of people? Thank you very much. Sorry if you couldn't see my slides, but thank you very much and God bless you.
Thank you, Dr. Silas. And we have learned a lot from you this evening. In the morning, we had some presentations from Kenya and Nigeria, and now it's great to have from you, from the African Union perspective. And of course, regarding the regulatory landscape, you highlighted the importance of regional harmonization of the regulations and the developments that have happened within the African continent over the last 2 years. But the most interesting thing is just that map that you showed, and everywhere we could see gene editing technologies being applied across Africa. So thanks a lot, and maybe before you get off, there's something that I want to find out about the science, technology, and innovation strategy for Africa 2024. And we know that we are probably 1.5 years to the end of that strategy. I just wanted to know what efforts are in place, and of course, if there are already any initiative looking at this new biotechnological interventions that have come up, like gene editing and the like, to be captured in that new strategy, and considering that the consultative process can take time. Thank you.
Thank you very much. I will respond briefly on that. One of the efforts is one, because that strategy starts around 2014, yeah, and if you count 10 years, in 2024, it's coming to an end. Yeah. So one of the things is that it needs again to be revitalized for the next 10 years, and that is ongoing. Yeah. The thing is that STI is at the heartbeat of Africa. Now the challenge of ABNA has been, you can't be a regulator and at the same time you can't be a promoter. So there is specifically an establishment of a center of excellence for science, technology, and innovation in Midland South Africa, and it was part of the hub that held the conference on STI that was in Mozambique, and Martin attended here. But the thing is that it is fast-tracking science, technology, and innovation, especially genome editing in the whole of Africa, looking at what do we have, what do we need, where is the funding come from, what projects should you focus on at the national level, and how can you expand them to be at the regional level. Yeah. So that is being picked up at the STI Center of Excellence South Africa. Otherwise, thanks very much, Dr. Martin. Thank you.
Okay, thank you. Next, we are going to have Dr. Afashimana. Dr. Afashimana has over 35 years experience that spans through natural resources management, forest plantation establishment and management, biodiversity management and conservation, and biosafety in an environmental sector, and agricultural research and development. He has a very good understanding of rural small-scale farmers in the Lake Albert Crescent agroecological zone. He holds a BSc in forestry from Makerere University, MSc with distinction in plant biodiversity and taxonomy from University of Edinburgh, and a PhD majoring in forest biodiversity. He has extensive experience on the Cartagena protocol. He was involved in the negotiations, and of course the CV is quite long. I've just truncated it. Thank you. So Dr. Fashimana, the floor is yours.
Thank you very much, Dr. Ngor, and good afternoon, ladies and gentlemen, in your various capacities. I'm going to talk about the Ugandan experience in regulating biotechnology since 1996, lessons and opportunities going forward. A lot has already been presented since Monday, or Tuesday. So some of the things I'll go on through in a brief. The background, you very well know that Uganda National Council for Science and Technology was established by an act of parliament, the Uganda National Council for Science and Technology Act of 1990. And among other things, the act gives the mandate to this council to clear all scientific research and development activities in the country, and to advise government on all aspects of science and technology. Now, in the early 1990s, the council realized that modern biotechnology was becoming increasingly important in agriculture, medicine, industry, and other fields worldwide. And they identified the need for Uganda to also incorporate modern biotechnology in Uganda's research and development agenda. Following that identification, the council called a meeting in April 1994 of key stakeholders in the relevant fields. That's human and animal medicine, agriculture, environment and natural resources. But they also went into areas like trade, social sciences, as well as other sectors, including the private sector. Here they were targeting experts in these different fields, but looking at people who are in academia, government, ministries, departments, and agencies, research institutions, and the private sector. Now, the meeting of the experts held in April 1994 also appreciated indeed the growing importance of modern biotechnology, but also the controversy and fears the technology had generated worldwide regarding its safety. And the same group of experts identified the need for national capacity building in modern biotechnology if the technology was to be introduced safely and to the benefit of Uganda and Ugandans. Then it was resolved that Uganda needed to develop guidelines on biosafety, that same meeting of national experts. Then they also recommended that a committee be formed to oversee the formulation of the guidelines and regulations governing biotechnology research, development, and applications. Following those recommendations for the meeting of experts, the Uganda National Council for Science and Technology appointed the members of a multidisciplinary committee, which was named the National Biosafety Committee. And this was done in November 1995, but the committee was launched in March 1996. Now, to date, the committee has considered and approved and overseen close to 20 applications for confined field trials and other trials for research and the generation of safety-related data. It has also overseen the development of various biosafety assessment standards, guidelines, and tools, some of which we shall look at. It also made inputs into the draft biosafety bill, which was later renamed the Genetic Engineering Regulatory Act, which is still in draft form. But it has also overseen formation of several institutional biosafety committees. I have a number of some of the 100 activities by the NBC to date from 1996. On the area of vaccines, first came up in 1996 when there was a proposal to try a genetically engineered HIV-1 vaccine, RVAC, in Uganda. That was the first in Africa. Yesterday, you listened to Dr. Kabi, who was presenting on the development of the sabolecin-based anti-tick vaccine in cattle, which is being done in National Livestock Resources Research Institute of Naro, which is based in Nachesasa near Namulonge. Then we have also overseen the establishment of several diagnostic laboratories. Yesterday, again, we listened to the lady from Uganda Wildlife Authority who was talking about the establishment of a lab at the Mwea Peninsula in the Queen Elizabeth Wildlife Protected Area for research and diagnostics on wildlife pathogens. You may be interested to note that Mwea Peninsula is an epicenter for anthrax and a number of other animal diseases. Then there was another lab established at the Fort Portal Regional Referral Hospital. Then at the College of Veterinary Medicine, Animal Resources and Biosecurity of Makerere University. Again, of course, their business is livestock. And the one of Nariri or Naro at Nachesasa. They are not only trying the anti-tick vaccine, they are also establishing a laboratory for development of that vaccine and a number of other vaccines that will come up as need arises. Then the Uganda Virus Research Institute, Dr. Kayondo presented this morning about the insect array for the genetically modified mosquitoes. We have made an input into the development of that laboratory infrastructure. Some of the other 100 activities have mainly been on crops. I have lumped them according to the different commodities. And each of them, of course, had several applications. For the banana, we started with an application to look at genetically modified bananas for resistance to black sigatoka disease. Then fortification of genetically modified bananas with the provitamin A and iron. Then GM bananas for bacterial wilt, that's a Xanthomonas bacterial wilt disease. And then for nematode and banana weevil resistance. Each of those was a separate application. They were considered at different times. In the interest of time, I put them all together because they were all looking at genetically modified bananas. We've looked at several applications for maize and for drought tolerance, then for stem borer pest resistance. And then for, this was mainly with the narrow, and also cassava. Dr. Richai presented what has been going on with the cassava. For the modified cassava, for cassava brown streak disease resistance and cassava mosaic resistance. And the most recent, which is still ongoing, is to do with the whitefly resistance. The whitefly is the vector for these pathogens that cause the previous two diseases. Then we have also looked at rice for nutrient use efficiency, for rice to see where it can survive in soils that are impoverished in nitrogen. Then for potato, this is the Irish potato, for potato blight resistance. Yesterday, the gentleman from Kachweka Nozadi presented what has been done, and they are in advanced stages to multi-locational trials. Then for cotton, this was one of the first ones, which was looking at pest resistance and then the Roundup herbicide tolerance. The experiments were being done at Serere and Kasese. Then for the sweet potato, we also looked at an application that was looking at genetically modified sweet potato for virus resistance, the virus disease, the sweet potato virus disease resistance. Then soybean, this was from Makerere University, glyphosate herbicide tolerance, the GM soybean. And for livestock, we have looked at the anti-tick vaccine development and testing, which Dr. Kabi presented about yesterday. Then, of course, how do we operate? All these applications are submitted to the Secretariat, which is at the Uganda National Council for Science and Technology. And upon receipt, the application is checked for completeness. And if it is complete, the application is tabled before the full National Biosafety Committee meeting. And then after the first reading, if I may use the term used in Parliament, the application is referred to a subcommittee of the NBC. The subcommittee usually comprises 3 to 5 members, targeting those with particular expertise in the subject matter under consideration. Then the subcommittee is given a time frame in which to review the application and report back to the NBC. And any issues or concerns raised is referred to the applicant for response. And if necessary, the applicant is invited to present to the NBC the responses to the queries raised. Then after the presentation by the applicant, the application is approved with or without conditions or rejected, depending on the quality of the response by the applicant. Now, of course, we have not been working in isolation. There have been a lot of developments at international level. that informed the regulatory process. The Convention on Biological Diversity, which has been hinted on by some presenters, which was adopted in 1992 at Rio de Janeiro in Brazil, one of the 3 treaties that were adopted at that conference. The other one being the United Nations Convention on Climate Change, on combating desertification, and the United Nations Framework Convention on climate change. Now, Article 8G, Uganda ratified this treaty in 1993. And of course, that means we have to implement the provisions in that treaty. Article 8G of the Convention on Biological Diversity provides that, or requires that, parties shall establish or maintain means to regulate, manage, or control the risks associated with the use and release of living modified organisms resulting from biotechnology, which are likely to have adverse environmental impacts that could affect the conservation and sustainable use of biological diversity, taking into account the risks to human health. The same treaty, the Convention on Biological Diversity, under Article 19, Section 3, has a provision stating that parties shall consider the need for and modalities of a protocol in the field of advanced informed agreement for the safe transfer, handling, and use of living modified organisms resulting from modern biotechnology that may have adverse effects on the conservation and sustainable use of biological diversity, taking into account risks to human health. Now, the Convention on Biological Diversity convened a meeting of experts from all over the world. The meeting was convened in July 1995 in Madrid, Spain. And the consensus after the meeting, it was a 5-day meeting, I represented Uganda in that meeting. The consensus was that indeed there was a need for a protocol, that is an international treaty, to govern the transboundary movement of living modified organisms. So the meeting made recommendations to the meeting of the Conference of Parties to the Convention on Biological Diversity, which was to convene, that was the second meeting of the Conference of Parties, which was to convene in Jakarta, Indonesia in November of the same year, 1995. And after the Conference of Parties considering the recommendations of the experts meeting, they commissioned an ad hoc open-ended working group on biosafety to negotiate and elaborate the protocol. So the result, the first meeting of the open-ended working group took place in Aarhus, in Denmark, in January 1996. And the negotiations went on up to 1998. They were supposed to end in 1998 in the Colombian city of Cartagena. That's how the name Cartagena Protocol came on board. But then we couldn't agree on a few things. So the negotiations were suspended. Throughout 1999, we were consulting both regionally and at global level. Then it was agreed that it was time to reconvene. So we reconvened in January 2000, and the final text of the protocol was adopted on 29th January 2000 in Montreal. Montreal, that's where the Secretariat of the Convention on Biological Diversity is. So the treaty was presented for signature in 2000 May in Nairobi during the 5th meeting of the Conference of Parties, and Uganda ratified it in 2001. And among other things, the Cartagena Protocol on Biosafety requires parties to take necessary legal, administrative, and other measures to implement their obligations under the protocol. It also puts in place a decision-making process, including the advanced informed agreement, administrative procedures, risk assessment and risk management, public participation and awareness. Then in its annex, there is a guidance on risk assessment. Then it also brought on board.
Sorry, Dr. Afasimana. Time is up. I give you around 2 minutes.
Okay.
Yeah.
So the capacity building initiative, Uganda benefited from that. The funding was through UNEP GF. And this is what led to the development of the National Biosafety Framework that later produced the National Biosafety and Biotechnology Policy. Then UNEP in 1995, after the second meeting of the Conference of Parties, produced international technical guidelines on safety and biotechnology. This was used by countries that needed to draft national laws. Then the OAU, that's now African Union, developed also a model law on safety and biotechnology. Again, this was meant to help countries in drafting their national laws. Later in the process, a supplementary protocol to the Cartagena Protocol called the Nagoya Kuala Lumpur Supplementary Protocol on Liability and Redress was also negotiated and adopted and was ratified by Uganda in 2014. All these developments informed the national processes. So we kept on improving the decision-making process and procedures. Learning from these are the different products, the Convention on Biological Diversity, the UNEP International Technical Guidelines, then the Cartagena Protocol on Biosafety and the Nagoya Kuala Lumpur Supplementary Protocol, then the African Model Law on Safety and Biotechnology. Then some of the standards and tools and guidelines developed by the UNCST and NBC and partners are listed there, starting from around 2005, 2007, then guidelines for containment regulations, 2007, National Biotechnology and Biosafety Policy, 2008, guidelines for interpretation of data. This was done by the National Bureau of Standards. Then the national guidelines for field trials, so in 2011, then the SOPs for institutional biosafety committees, which was launched by the Honorable Minister at the opening of this conference. Then there are several others which are awaiting approval, listed there. These are new techniques that are coming up which need to be taken into consideration. Biosafety inspection manual for confined field trials, standard operating procedures for conducting contained and confined research involving GM animals and arthropods, guidelines for review and risk assessment of new breeding techniques, then SOPs for import and handling of food and feed containing GM ingredients. Then these last two were developed between the National Council for Science and Technology in collaboration with the National Environment Management Authority. They are also awaiting, these were done last year. The ED NEMA hinted on them yesterday. Challenges, of course, low staffing at the Secretariat, then high staff turnover has been a challenge sometimes. Then, of course, limited financial resources. The NBC membership keeps changing. This is both good and bad in that as we change, we get new members and new expertise on board, but also broadening the experience of more people. Then the slow legislative process, which the first draft bill was produced in 2012, 10 years down the road, we still have no law. Opportunities, we now have better capacity built over time in the country. We have several universities training people in biotechnology. We have a conducive political environment for the science and technology and innovation initiative. You saw to what length the minister went at the opening, trying to tell us how much the priority science, technology, and innovation is for Uganda. Then we have also international collaborators, both in terms of technology transfer and exchange of information. Lessons learned, IPR issues are getting understood. At the beginning, some of the applicants thought we were delaying their research by asking if they were collaborating with foreign institutions. Have you got any written approval, or what are the terms and conditions for using this technology? Some of them thought we were delaying them, but after realizing that the people they are dealing with are not angels, they are now realizing that you need actually to agree in writing with any collaborators on IPR issues, on whatever materials or products will come out of the research. Otherwise, you may do a lot, put in a lot of effort, but the technology is not available or free for use in the country. And of course, best technologies are best developed and funded nationally for solving national problems. No Father Christmas or donor from somewhere is going to help us to fund strategic research to solve our national problems. That's the Baganda say, "Nanyi ni mufuya kwacha waunya." Biosafety is a cross-cutting issue, and therefore, no single institution will be self-sufficient. I've already talked about the NBC being multi-sector and multi-institutional and multi-expertise. And so sector and institutional collaboration will always be necessary. And technology is developing faster than regulation. We have seen that before you finish making a law to regulate what is already on the market, some new techniques have come up. This was dwelt upon in the morning. I'll not go into that detail. Then, of course, we need to keep our ears to the ground because the developments are coming up regionally and globally. And if you don't take note of what is coming up, even if it is not within your country, we are living in a global village, they will eventually find their way into the country, and you need to know how to handle them. This is the last slide. Inception, present, and future. I can say we started kind of groping in the dark in the late 1990s. We have reached where we have some national capacity. Of course, more is still needed, with some products ready for release if the law was in place. And we need to finalize the law, taking into account concerns raised by His Excellency and other stakeholders 2 years ago, but also take into consideration new and upcoming techniques to complete the regulatory framework. And we need nationally generated funds sufficient to tailor new products that will feed into the industrial value chains that the Honorable Minister was referring to in her opening remarks to lead us into the desired future. Thank you very much.
Thank you, Dr. Afashimana, for giving us the 25 years of biosafety regulation in the country. I would like us to recognize the presence of our Honorable Minister, Dr. Musenero Masanza. You are welcome for this afternoon session, and let us give us a warm applause. Honorable Minister, we have had very fruitful discussions in the morning, and especially we were discussing about biotechnological applications in the health center, and in the health sector, and we had presentations from Uganda Virus Research Institute, Joint Clinical Research Center, and the Ministry of Health, the biosecurity team. And of course, they gave us quite a good overview of our biosecurity situation in the country. And of course, we had a discussion session on science communication, and it was a very interactive session. And of course, there was also that debate of whether we are interfering with God's work. And of course, there was also that discussion that we are actually protecting and actually doing everything for the good of our country and fostering prosperity. Now, all these have been summarized into a communique that will come out after this session. In the meantime, we are having discussions on the regulatory landscape in Uganda. And of course, we have had a presentation from the African Union, and the presenter whom you found was the second one, Dr. Afasimana. Now I shall invite the third presenter, Dr. Asea Geoffrey. And Dr. Asea Geoffrey is the Director of Research, National Crop Research Institute, a constituent institute of NARO based at Namulongwe. He is a lead maize breeder who led a team on application of biotechnology on maize for drought and insect resistance, and a country coordinator for water efficient maize partnership, and later Teller. And yesterday he gave us a very good presentation on research which is going on maize drought tolerance and pest resistance. Dr. Asea, you're welcome.
Good afternoon, everybody. Thank you, Chair, for your kind introduction. And also thank you very much, Dr. David Afsimana, for your presentation that lays foundation for the regulatory framework internationally and also domesticated nationally and where we are. So my presentation is mainly a reflection of the biotech environment, regulatory environment in Uganda. And from research perspective, I hope it resonates well with my colleagues in product development across board. So I want to start by giving you a catalog of a suite of traits that are going through testing by confined field trials. And some of them have actually been completed, ready for the regulation with compelling products, but awaiting the law. So you have this only from narrow and this only crops, but there are also, as you know, animal vaccines, and there's a lot of presentations also from Uganda Virus Research Institute. So there's a lot going in the country, and so we're in various stages. Many of us know a functional biosafety system must have components including a policy, which we have as a country, and that policy must be operationalized by a national law, which should be the biotechnology law, which we don't have. And the law should also be, from the law, the attendant regulations and also guidelines must be developed to conduct some of this work. So ideally, this wheel must be driving in a clockwise manner. You can see it is both clockwise and anti-clockwise. So that also presents a gap and a challenge. And our experiences in the last 20 years, from the last bill of 2012, we have moved to the last bill of JERA 2018, and our movement has also been challenging. The first bill was actually, in our opinion, very permissive, and it was based on the Katakana protocol that Dr. Simana presented. The later bills became a bit more prohibitive, and the last one even had provisions for strict liability, and it became more prohibitive. So you can see from 2012, we've been moving from green to orange to red, instead of moving from green to deep green. Yeah. So that is something also we need to reflect on. So in terms of approvals, I think this has been going well, from product development in the laboratories through containments to CFTs, proof of concepts, and also product testing. I think we have gone through these stages in the various commodities, but now we are stuck in stage 3 to go for multi-location and deregulation. So we can't move because we need attendant regulation, which is anchored in the law, which we don't have. But the bigger goal is actually to have these products for the farmers, because they address real issues and needs, and we think they contribute significantly to address some of the challenges currently, especially in agriculture. So this was my impression of the journey in the last 20 years in the biotechnology research. It is in terms of turns and twists. For me, who is not a civil engineer, you can see, I don't know why the engineers they didn't make a straight line using this red. They had to make the other turn, yeah? So that is how we feel. But nevertheless, a lot of things as well have worked well. For us in research from product development, we feel there is a clear pathway for approval for research and containment to confined field trials. And there's a lot of work that is anchored on this pathway and the regulations and guidelines for NBC. Number 2, there's attendant regulations and guidelines in place that that regulates this work that is going on. And 3, we're also very grateful that the regulatory system is science-based, and the regulatory system is science-based, and it is fully functional with the competent persons. you can see the experience of Sumana, who was involved in development of the first international protocols from the early 90s up to the current domestication in the country. So that is a plus. And also the new ones who come, we see they are regularly trained and brought up to speed. And also the inspectors, the regulators frequently come and visit us for inspections and also check for compliance. And the good news is all over this time, we have been 100% compliant. One of the things with this technology is compliance is very, very important. So we have been 100% compliant without any accident. So I think that has been a good journey, but there are still big rocks on the way. And the rocks on Biosafety, I think, has been alluded to. One, a lot is being done, but without a conducive legislative instrument. That's the biotech law is not in place. And you have seen the journey it has taken to develop the bills and the debates that have been going around it. So we feel there's no clear pathway for deployment and commercializing some of these compelling products, which are in the pipelines, in the safeties, and also in the testing stages. Number two, we've also seen a lot of activism work around this technology and also public perception. That is being skewed to negativity. And I think that presents a big challenge for us in product development. And yet these products, we see a lot of potential and benefit for the country, but also for the farmers and people. We think also the need for increased advocacy and also interest to analyze where the challenges are so that these issues can be well clearly sorted out and moved forward. And also lastly, coordination, I think has been mentioned, somebody mentioned it earlier. Coordinated communication and advocacy is very important for uniform messaging so that people are not confused. This is a new technology, and if there's different information going to different people, it confuses people. So with that, I think those are my reflections. But the last slide is a challenge.
Sorry, Dr. Asea, time is.
Okay, want to give me 1 minute? I think Afismana mentioned it rightly. A lot of this work is being done from competitive grants and projects. And the donors also get fatigued when there's no clear pathway for deployment. And we also know there's a lot of emerging and re-emerging patient diseases that present challenge to farmers for productivity and also production. But we also see now the scenario in other countries are rapidly changing, and yet Uganda has been a leader in terms of research and the number of technologies that are in CFTs. But we are seeing we're now left behind in terms of where we are. So our neighbors, Kenya, already adopting some of these technologies, deregulation. Also, bioeconomy is being embraced, but also it is premised on this technology. We see challenge of increasing population, rising food prices, food nutrition and nutrition security, climate change. All these are formidable challenges, and require innovations, these innovations and technology to solve them. And we think this is the best pathway forward. So with that, thanks, Chair, and thanks for your kind attention.
Thanks, Dr. Asea, for giving us the landscape from the perspective of agricultural research and development, especially the field trials. Now, we are going to have a slight change in our program. The next session was to be, still we shall have a discussion that is on biosafety legislation in Uganda, a focus on genetic regulatory bill 2019. And Ms. Harriet Itiang from Ministry of Justice and Constitutional Affairs will give us an overview. And then instead of having the discussions, yes, we shall have the discussions, but it will be focused more on two actions that we need to take after this forum. And the Honorable Minister will come and lead that session. So, and then after that, before the closing remarks, we shall then have a thank you session. So that's how we shall proceed. And I would like to take this opportunity to invite Ms. Harriet Etiang, who is a member of the National Biosafety Committee and supporting us on the regulatory aspects of biosafety regulations. So all those applications approved by the National Biosafety Committee, we always have a legal eye from the National Biosafety Committee and in collaboration with the Ministry of Justice. Harriet, you can come forward. Thank you.
Good afternoon, ladies and gentlemen, all protocol observed. My name is Harriet Etiang. I'm a state attorney in the Ministry of Justice and Constitutional Affairs, the Directorate of First Parliamentary Council. The Directorate of First Parliamentary Council is an executive office in the Ministry of Justice and Constitutional Affairs, and it's mandated to draft all legislation in the country and to keep the statute book clean. I'm here today to speak about the biosafety legislation in Uganda, but in particular to take us through JERA, where we started from up to where we got to today, and hopefully what will happen after or in the near future regarding legislation on biosafety. So I'll give a background, brief background to this. We had the National Biosafety and Biotechnology Bill 2012, which was presented to the 10th Parliament on 4th October 2017. This bill was passed into an act by the 10th Parliament, and it was forwarded to His Excellency the President for assent. However, under the law, the President can return an act if he has issues with it. And indeed, he had some issues and he returned the bill to Parliament for reconsideration with a number of comments that he wanted Parliament to have a look at and reconsider. Among those, he proposed that the title of the bill be changed to Genetic Engineering Regulatory Act. He also had issues with the approval of the different stages of research, who orders or stops a GMO activity and under what circumstances. He also had issues with labeling of the GMO and its products, as well as on the clause on liability for damages and some of the offenses and penalties that had been prescribed in that act. The 10th Parliament considered the bill and passed it as the Genetic Engineering Regulatory Act 2018, and again presented it to the President for assent. However, the President on 22nd July returned the bill to Parliament, again with some concerns, rightfully so under the law. And this time, some of the clauses he had issues with was the one on benefit sharing, isolation measures, and how to deal with offenses that are committed by a body corporate. Unfortunately, the 10th Parliament lapsed before it was able to consider those provisions, and the 11th Parliament didn't save the act. The saving is normally done within a certain time frame, and if it is not saved, that means we go back to the drawing board. We have to go back to Cabinet to consider and make consideration of the principles and determine whether they want to continue with that bill or totally come up with something different. So on 15th May, His Excellency the President directed that the bill that had lapsed, the JERA, the Genetic Engineering Regulatory Act 2018, be returned to cabinet for fine tuning and to include issues of use of gene therapy in human beings. But as most of us recall, JERA catered for mainly agriculture and just some aspects of human and environmental safety, but it didn't cater for aspects of research and regulation of human genome manipulation as an aspect of human health. The scope of JERA would therefore need to be expanded to include regulation of research and use of gene therapy for treatment of diseases in human beings. So the Ministry of Health wrote to the Attorney General seeking advice on how this could be handled. Should we continue with the bill and add those aspects, or should we totally take new principles to cabinet to handle the new aspects that were being proposed by the Ministry of Health and other stakeholders? The Attorney General advised that the Ministry of Health, Ministry of Science and Technology, Ministry of Agriculture, UNCST, and any other relevant stakeholders who have an interest in this need to come up with totally new principles for a new legal framework to streamline that the use of biotechnology in all the relevant sectors, so that we don't have different groups coming up with proposals and yet an act has already been passed in that line. So that was the advice of the Attorney General. However, I would like to take you briefly through what JERA 2018 was all about and how it changed from the National Biotechnology and Biosafety Act to the Genetic Engineering Regulatory Act, and what were the new aspects, if any. So JERA didn't change a lot from the Biotechnology and Biosafety Act. However, the main issue there was changing from GMOs to genetic engineering activities. So the long title of JERA now reads an act to regulate genetic engineering activities and facilitate safe development, transfer, application, and utilization of genetically engineered materials. It was also supposed to designate a national focal point, establish a competent authority, establish an interministerial policy committee. This was a new element which wasn't in the Biotechnology and Biosafety Act, and to provide for establishment of institutional genetic engineering committees, which were previously the IBCs, and to provide mechanisms to regulate research, development, and general release of genetically engineered materials and for other matters. So the Act applies to research and general release of genetically engineered materials, but specifically leaves out drugs, because the proposal was that drugs should be handled under the National Drug Policy and Authority Act. The objectives of this act were to regulate genetic engineering and facilitate safe development, transfer, application, and utilization of genetically engineered materials, to facilitate and promote research, development, and use of genetic engineering, establish procedures for bioethical considerations in genetic engineering research, to strengthen consumer protection, public understanding of products and benefits of genetic engineering, facilitate safe use of genetic engineering to address national development challenges in food security, health care, biodiversity, and conservation, and to build capacity in genetic engineering research, development, and innovation, to promote technology transfer and benefit sharing the development and use of genetic engineering, and to build strong institutional relationships among genetic engineering stakeholders. So again, this didn't change a lot from the previous act. The institutional arrangement, however, changed a bit, where we now had the competent authority as a Directorate of Genetic Engineering, which was going to be based in the national focal point, which was the Ministry of Science and Technology. Where we had the IBCs, we were going to have institutional genetic engineering committees. And the newest element there was the interministerial committee, which had about a membership of 20 or so members from different MDAs. The inspectors will be very central in ensuring compliance of the act. And we're going to have a national genetic engineering committee at the national level to oversee issues of biotechnology and biosafety And of course, the major stakeholders there are those MDAs, agriculture, environment, health, NEMA, standards, and trade. So the review process also remained the same. It was going to depend on the nature of the activity as outlined there. There was going to be review at both institutional and national levels, as was the case in the previous act. The National Genetic Engineering Committee was going to be the main national body for biosafety matters under the competent authority, which is the Department of Genetic Engineering in the Ministry of Science and Technology. All relevant government agencies were to have an input into specific applications as and when. If it had to do with agriculture, agriculture had to be consulted. If it was health, health had to give an input into this application. All approvals and denials were to be based on sound, scientifically acceptable risk and safety assessment, which was also a provision previously in the previous act. So approval processes for GM research and development, for laboratory experiments, the approving authority was going to be the Institutional Genetic Engineering Committee, and it was supposed to work within 30 days, and the competent authority had to be notified at all times. Contained testing, Engineering Genetic Institutional Genetic Engineering Committee was supposed to do its work within 28 days. And they had to report to the competent authority through the Institutional Genetic Engineering Committee. And the National Genetic Engineering Committee was supposed to review their applications. For confined field testing, the reporting was to the competent authority within 90 days. And it had to go through the Institutional Genetic Engineering Committee and reviewed by the National Genetic Engineering Committee. And general release, 270 days, and all other agencies had to have an input, and that application had to be gazetted. For import, export, and transit, 28 days, and you had to get a permit from the relevant government institution. So the JIRA had aspects of risk and safety assessment and management. The bill emphasized safety in using biotechnology by providing for measures to be taken to minimize or avoid risk to both human and the environment, which may arise from actual or potential contact with a genetically engineered material. It also provides for every application for research or general release to contain an emergency plan, complete with safety measures for unintentional release of a GM. Restoration and inspections, the competent authority had the power to stop an activity where it felt that human health or the environment is threatened. It could also stop an activity that was undertaken without approval, and it could also issue restoration orders in the event of damage to human health and the environment. The competent authority could also investigate claims concerning GM activities where there are any suspicions, and it could conduct inspections as and when necessary. Sorry for the font, but under the Act, there were also going to be regulations that were going to be produced to facilitate the implementation of the Act, one of which was going to provide procedures for research, prescribed procedures for general release of genetically engineered materials into the environment, for handling, transport, identification, labeling, and packaging of genetically engineered materials, specifying fees for applications and the services under the Act, specifying the safety levels and standards of safety for GMs and GM products on the recommendation of the Bureau of Standards, establish procedures for bioethical considerations, and prescribe penalties in respect of any contravention of those regulations and for any other matter that may have been left out in this list. So the concerns from scientists were in JERA were probably in section 26, labeling of a GM, and that is what it provided, that whoever was involved in research general release, importation, exportation, transit, or trade would have to conspicuously label whatever they are doing with those words in bold. They also had to indicate the relevant characteristics and then, the origin of that gem, a place where a place where an activity involving a gem was being carried out had to be conspicuously labeled by the applicant, and a person who contravenes or who doesn't do any of those things will have committed an offense and shall be, on conviction, liable to a fine not exceeding 1,000 currency points, or imprisonment not exceeding 6 years. The other was that provision on liability and redress, section 35, which required that a person who owns a patent in a gem is strictly liable for any harm, injury, or loss caused directly or indirectly by such a gem to the community. It went ahead to define what a patent is to mean the exclusive rights granted by a government to any person in respect of a gem. There were also concerns with offenses and penalties. There was a provision that a person who introduces a terminator seed or a gene that is genetically engineered to make any offspring of a crop sterile or unable to produce or uses genetic use restriction technology commits an offense and shall on conviction be liable. I've flashed those ones to give us a chance that now, as we await cabinet to give its decision, which is most probably going to be coming up with new principles. As scientists, we need to be keen and participate in getting these provisions that we had concerns with properly drafted and not go from green to red. This is the chance for all of us, and maybe we'll await the decision of cabinet on this.
Thanks, Ms. Harriet Etiang. Your time was up, but I know that since you are from Ministry of Justice, we don't tell you that time is up. So I gave you time to finish. And of course, it's very vital for us because this now brings us to the core of this forum. And I think we have reached that point. Now, I would like to find out if there is a representative or the executive director, NEMA, is around. If there is someone, you can come forward. And then the Commissioner Seed Certification in the Ministry of Agriculture. Okay. And then Executive Director, UNBS. And then the Secretary to the Authority, NDA. And I have also Dr. Okware from Uganda National Health Research Organization. Okay. Yeah. So the next session should have been a discussion, and actually it is going to be a discussion, and it's going to be very focused. And the focus is like we have had everything over the 3 days. And as the Honorable Minister mentioned before, in fact, I think this is the first event that we have been together where she has not been fully, and she always passed that message that she's a technical minister, and therefore I'm handing over to my Honorable Minister, Dr. Monica Musenero. Please come forward.
Good afternoon. Allow me skip the protocol since I'm a technical minister. I bring apologies from the Vice President. She's not able to make it. She had intended to be here, but she had some emergency assignment, so she couldn't make it here. So I've come in as a dangerous substitute once again. Thank you very much for being here for the past 3 days. When I look at the man hours which have gone into this forum, we have about how many people in the room sitting here for so many hours per day? That's a lot of time input, a lot of time. And so I really appreciate that you have been so diligently sitting here and discussing and listening and coming up with some of the things that we have come up with. And I believe that up here in the panel, I have the most important regulators in the country. If there are any who are missing, I don't know if there are any who are missing. UNCST, I think you better carry your chair and append yourself to here because I'm going to put you on the spot. So we have been here for 3 days. So what? We've talked. It's not the first time we've been talking. We have eaten food in Serena. It's not the first time. We've sat in this nice air conditioning room. The question is, how is this going to make Uganda better tomorrow? And that's what we are going to focus on now. Now that we have had all the presentations we've had, we've been listening to those presentations before. We've been to conferences. Those who have worked with me know that sometimes I'm a pain when it comes to action, because it, for me, doesn't make sense for us to come sit here and then leave and then wait to gather next year. What are we going to do because of what we have been here? How are we going to gather next year at a better place? Not again talking about a stalled Jera, not talking about, I've had a lot of lamentations in the presentations, the ones that have been made when I was here. By the way, lamentations will never end because lamentations represent problems and problems represent opportunities, unless you don't recognize it. But lamentations is a dissatisfaction in you. It's the gap between what things should be and what they are. And you're uncomfortable because we want to be where it should be. So if you run out of lamentations, that means you've stopped growing. You stopped growing. You have achieved everything and you've stopped there. So in this session, which is also the closing session, we would like to have a discussion, but more of an action discussion. We want, now that we have all the regulators here, what are we going to do? In my opening session here, I talked about the vision for the nation, the vision for science, technology, and innovation, and as it relates to where the country wants to go. And I stressed the role of relevant regulation, organic regulation, which moves, facilitative regulation. There are some things that haunted that GMO bill. Many people don't know, but in 2001, I participated in drafting one of the first drafts of that document. I was in Makerere teaching molecular biology, and I was requested to contribute to that document. 2001, and now we are in 2022, and the law has been. Now, if we could afford to do that, because at the time we started, it was like we were in the defensive. Don't bring your things to us. Let them stay there. If they come, we need to make sure we are safe. But if this is an industry that we are developing as a country, we can't afford to take 20 years just working on a law. When I became minister, the president was calling back this law. And then I learned that all those had lapsed. And so we cannot put them back. We have to go back to the beginning. And we took some time because we wanted to stabilize the structure. And so when we were coming to this conference, to this forum, I insisted that we make this one of the central things. At least we need to have some actions such that when we come back, we will have progress to measure, not simply storytelling of the same things we have been doing. So I came in when he was presenting. I don't remember your name. David Havashman. Okay, I hope I got that. At least I got David. You were presenting, and we have all those contexts. The first question I want to put to you, the panel, is first, before we go to JERA, what is the legal framework? What is the law, the general law, which we refer to generally in science? Do we have a law which guides us generally before? Because genetic engineering is a small component. Do we have a general law that we all know? Because since I became minister, I've tried to look for it. I was even asking Dr. Kware, do we have this law which governs research in the country generally, apart from the UNCST Act? Because if we had that general law, then some of the things in JERA, we would already have dealt with them. And we'll be dealing with very specific things. But we are trying to take a lot of science generally, but now we have to cover it in JERA. And maybe I'll ask the one from Ministry of Justice. What laws do we generally refer to, acts of parliament, which are guiding the national science ecosystem? You can use that. Because when we have that, then we are going to make, if it is not there, then maybe that's one of the things we need to work on. Because everybody is speaking things from left to right.
Honorable Minister, for now, I would think it's the NEMA Act, but it doesn't cover most of the components that deal with biosafety.
It's the NEMA Act?
It's 2019.
It's what guides 2019?
Yes, please.
That's what we hinge on. But you're saying it doesn't have most of the components.
Of what JERA would want to regulate.
What about the rest of research?
Research, I think we've been depending a lot on the National Council of Science and Technology Act.
But doesn't have a lot of details.
Yes.
It really does. I've read that act so many times. It's simply an act governing
research and science.
governing the formation of that act, that National Council of Science.
Yes. So I would say that we don't have a law that.
So really, we don't have a comprehensive law which guides research, science, technology, and innovation in the country.
Yes, ma'am.
Let me tell you this, because when I was young, there was a man on our village called Kapriaka. He didn't go to school, but this guy invented gunpowder. I don't know how he did it, and he made his own gun. He was arrested. He was arrested for making gunpowder and making a gun, I think, from wood and things. And he was using it for hunting jennifer. So if we don't have that, then for me, that should be one of the resolutions from this meeting, that we need a law which governs science, technology, and innovation in the country. Because everywhere you move, you hit a wall. I don't know. Let me open up and have some views from these people, just on that point. The legal framework for STI in the country. If you have an opinion, please give it, or if you have some information to help us.
Yes, Honorable Minister, I think Harriet has mentioned it well. There are scattered acts and the laws that govern research, and indeed there is need for one-stop STI law that should bring all this to home. For example, in NAR, we have the National Agricultural Research Act. In the ministry, we have the Plant and Seed Act that also governs some of the work we do. So these are scattered efforts. So the STI ecosystem is not all covered in those laws.
I know the reporters must be picking some of this. Yes, Dr. Perry?
Yes, Honorable Minister, I want to take this one notch up. All these laws are based on the Constitution of the Republic of Uganda. For instance, there are sections which do no harm, personal protection, human rights. There are so many things which really protect the health of the individual. I think that's really the first law. I don't know whether you call it legal law, but that is really the basic law that is governing all of us. Now, over and above that, then we have this Vision 2040. I think there is a big chunk there on emphasis on science and technology as a direction, almost as a policy, almost as an act, almost as a direction for the implementers. So maybe that could perhaps be a regulation which might support us to move forward. Now, at the sector level, I think each of the sectors, agriculture, health, I think agriculture and health, I think maybe one or two others, there is, they have their own, they developed strategic plans with research embedded into their strategic plans. For instance, in the health sector, we have a 10-year policy on research, on health research. Then we have strategic plan, and then we have specific activities, and then we work together with the council. So I'm not sure about the other sectors, but I thought as we go down, we should be able to pick up research efforts. Yesterday I was emphasizing the issue of communities. I think really that's where our research really should actually start. They should be the first people to pick up information and transmit it to us. So, Honorable Minister, I think I quite agree with you that there should be a law, an overarching law, which protects, which brings these elements of the Constitution to protect to promote research, but within the context of these aspects of the Constitution.
Thanks, Honorable Minister. And the issue of the STI laws, I think what Ms. Harriet Etiang mentioned is true. And actually, these laws are scattered through the different sections. There's the National Agricultural Research Act. There is the Seed Act. And of course, when you go to NDA, there is one that guides on clinical research. There's Uganda National Health Research Organization. And then when you go to Uganda Wildlife Authority, their act also has some regulations. And then, of course, in the Ministry of Health, there are some regulations on health laboratories. And then if you go to, again, Ministry of Tourism, there's also a bill which is in Parliament at the moment regarding research on antiquities, anthropology, and the like. And if you go to National Archives, there is also something on research regarding access to government documentation if you are doing research. And of course, there are many others. And some of these laws actually exist as guidelines in most of the government entities, but they have not transformed into regulations, making, first of all, enforcement very difficult. And because of the gap in the regulation, you find out that there are some loose ends here and there. And then, of course, also looking at the policy, I think the science policy was developed in 2008, but of course, that has been replaced by NDP3. But again, when you look at NDP3, it is not cutting edge, incisive when we look at the Honorable Minister's vision of making Uganda the best. So I think it's spot on, the need for that overarching regulation. Thank you.
Thank you, Honorable Minister. I concur with you with us having a standalone law on research and innovation, whereas we have the National Environment Act of 2019. It only makes references to other sectoral laws, given that NEMA's mandate is really coordinational. So it only makes reference to other sectoral laws that relate to biotechnology development and biosafety. It also lays emphasis on the precautionary principle, which very much is relevant in the area of biotechnology development in terms of ensuring that the environment and health is not hampered as we develop technologies towards ensuring a resilient population in terms of food security and reducing the disease burden. So I think I concur with you with regard to having a standalone research and innovation law, which the National Environment Act can make reference to. Thank you.
You want to make a comment?
Thank you, Honorable Minister. Just like the colleagues have said, the Ministry of Agriculture, Animal Industry and Fisheries has a number of laws that are implementing both for crop and animal. And when you look at the laws, the general laws, we enforce them, but in terms of them being applicable to science, we do enforcement of trying to regulate research, but in actual sense, they're specific to the sector. And we can't say these are specific to science as a general, but specific to the sector, either crop or animal. Submit.
Thank you very much. I think I'm going to throw it back to you to advise us, given what we have had, what should we do? Because I find trying to implement the industrial value chains and trying to make science work you spend so much time trying to find these laws wherever they are sequestered and then trying to interpret how does it apply now to this one. And you are the legal person from Justice. The conclusion is that we really don't have a comprehensive law that handles science, technology, and innovation in the country. We have bits and pieces applicable to different units, or if you find them, I've read quite a few of them, trying to see which one is making. They all refer to the Constitution, but you know, a Constitution is a very high-level document, and it's very difficult. For example, one of the gaps I found is that it seemed like there was nobody holding scientists accountable, apart from maybe, but if I commit some, and unless I am in trying to do a clinical trial and I try to harm a human being, but if I commit some unethical behavior in my process of science, there isn't a clear framework who should punish me or who should hold me accountable. It is not there. Because there are so many things that scientists can do other than harming a human being in the process. So whereas harming a human being is very clear, the rest of the responsibilities of scientists, there are no laws. There's nothing that you can refer to.
So, Honorable Chair, we've just been informed that the policy is as old as 2008. And I think probably one of the channels would be first going back and reviewing that policy. However, we're aware that you don't necessarily have to have a policy to do legislation. So if it is felt that the policy will take longer, then we could go in straight and advocate for a comprehensive law to regulate all those components of technology, science, technology, and innovation.
Yeah, to the best of my knowledge, that policy expired, isn't it?
Honorable Minister, it probably needs review.
I was told it was, I think it was.
Oh, it was for a
I've seen some
period.
reference to it being having expired in 2018 or something like that.
I think probably it needs review, but the key issue is that most of the articles in those in that policy now doesn't hold. It's
They
like
don't apply.
very
Okay.
outdated.
So I thought we need to leave this meeting with some key actions. And if we think that this is something that should be done, I think so, that we need to gather all these things so that there is a place of reference where you can easily find it. And you want to say something? No. And look at the policy first, and maybe then we make a decision. So could that be an action point from this meeting? That we are committing ourselves to create this comprehensive regulatory ecosystem that we can all refer to. We don't have to retable what NEMA does, but here you could say, here you apply the NEMA Act.
Yeah, Honorable Chair, that would be acceptable to commit to review the 2008 policy and to go through all the processes to have it approved. Depending on matters of business in cabinet, I don't know how soon that would come out, but it could also be done concurrently if the feeling of this meeting is that there should be legislation that addresses those three components. The process of having principles drafted and tabled in cabinet would also start while the policy is being worked upon. Because we've had legislation without policy, though the most ideal would be to have the policy first to guide the legislation.
Okay, thank you. Because I thought that it could be easier for us to draft GERA to redraft, if there was, we are not trying to cover general research and then genetic engineering in one document. So, and yet we are trying to refer only to to genetic engineering. I have read that document and it is really struggling sometimes to bring general research into genetic engineering. So in the process, it opens itself up for criticism. So that is one of the things I thought. You can sit if you are tired, but if you want, you will be my support through. The second thing is now, of course, it was about the policy, and I think we've talked about the policy. The reviewing of the policy is not only, it's urgent actually, and it's really important, not only because it has expired, but because over the last 1 year, the president has cast a completely different scope, which scope covers things that we had not thought about before as being significant in our country. And so we need to have all these primary 8 areas, plus there are things like nuclear energy. Where we want to go. We want to look into mineral beneficiation. We want to, and this has different components that we need to make sure that we have taken care of. So the scope is now broader, and we are talking a lot about domestic manufacturing. That means if I was talking about nuclear energy or something that comes out of it, we were leaving the waste there. Now we are going to deal with the waste here. We are the ones, secondly, we are the ones assuring the world that, okay, we have made our bus is okay. Our bus is fine. We have this kind of, not everybody going into some closed Mavat place, fabricating something and putting it on the road. And you don't know what consequences are going. Where is the crash testing for those vehicles which we are making? So we are making things and assuring the world by our staff it is safe. For those of you who are not here when I was opening, for me that is my major thrust. We are working on being manufacturers, on being the source of technology. And the laws and the regulations we have should be good enough to protect Ugandans, but also to assure the world that what we have done is good. So there is legislation which should be in line with global, with regional, continental standards. So we need to review the policy to see how they align. So I think that was my second point, the policy, if we take it with the law. Then specifically coming to the genetic engineering law. Where should we go next? How should we act? I'm not talking about the theory, but the practices. What do we need to have tomorrow to make sure that we have the kind of law that we need? Since I came to office, a lot of people have been handling like I'm the one who laid that law. I've had like, you should bring the law, you must bring the law, you are sitting on the law. And it sometimes you can't explain to everybody what is going on. The issue is, what I have seen is we need a law. We needed it yesterday. What actions should we take to make sure we have this law, the right law, including these things, I was copied into that letter, including those new areas. What should we do to have it as soon as possible? Volunteers to discuss this briefly before we go to the last question.
Honorable Minister, my proposal would be to avoid a situation where if we put issues of health tomorrow, Then issues of trade will come up the next day, and basically to have piecemeal incorporations into this bill. I propose that now since we have a chance to go back to cabinet with fresh principles, I would propose that all stakeholders that have a stake in this area of research are brought together to agree on areas that they feel should be regulated, so that when we go to cabinet, we have a comprehensive document to address all areas. And after that, I think the rest would fall into place, because right now what has happened is the Ministry of Health has come up and said they think issues of health should also be in here. And yet the previous document was only on agriculture. Yes. And yet we're aware that there are several other stakeholders, for example, NEMA, on issues of the environment. Standards, Bureau of Standards may also have issues there that they would like to see in there. So I think that is the starting point, all stakeholders to come together and agree on what should be regulated in that area.
Have these stakeholders presented some of their issues in the meeting? Could that be a beginning for us to use what they have presented here to draft something?
Honorable Minister, the stakeholders presented their concerns. Besides the technical presentations, they also had sections on challenges, gaps, and key issues. And the rapporteuring team have actually captured it, and we are going to share it as soon as possible. And we are hoping that this forum, we are going to have three things. One is that we are going to have the list of all participants, and then we are going to set up a virtual forum where there is continuous engagement. We hope to link with the think tanks and the like, probably to have a session or two and interact. We are also planning to have a knowledge repository whereby all those regulations and documentation and evidence can be deposited and also accessible by all the key stakeholders. So that's the proposition. Thank you.
Honorable Minister, I wanted to add that
before you come in, promises which are open-ended never deliver any results. When do we hope to have a new draft? What time? What time do we promise? Otherwise, we can be depositing those laws for the next 2 years.
it was to build on the outcome of this meeting, to make the process more consultative from the different MDAs. It will be good under your guidance to write so that there is a team formed that can build on the outcome of this meeting, so that the process is consultative, then a roadmap is drawn, again under your guidance to see how this can be actualized.
Thank you very much, Honorable Minister. I think building on what Harriet presented, to have a comprehensive law and what Dr. Seah has just talked about of consultative process, we need to be careful that what has kept us down in the past, the biggest challenge has been the process goes on and goes on and goes on. By the time we wake up, the lifespan of Parliament is ending, then whatever you had built lapses and is lost. So there is need to get on board everybody, all the other sectors that have issues to raise. But probably from this meeting, we could appoint a multi-sectoral task force where all these key ministries that have concerns are represented. Because if we leave them to generate their concerns individually, will take 2 years before we have finished consolidating their views. Then their representatives on this task force would be tasked with consulting within their sectors and coming up with the concerns from those sectors so that we have a comprehensive draft as quickly as possible, which would be taken to cabinet and hopefully from there to parliament. Also looking at this, the old bill, the bill lapsed, but the ideas there did not. Then we see what we can pick from there and get these views from the different sectors to get a comprehensive law, which can hopefully be debated by the current parliament and passed before its lifespan expires. Thank you.
Dr. Kware, then we shall.
I agree with what my colleagues have said, except I want to expand it a little bit. The speed of a convoy is decided by the slowest boat. So if we have to get all these constituencies to work, and yet they have resent this document to us, it might not be easy for us to have everybody on board, especially if in the unlikely event that there may be another emergency, like Ebola, like COVID. I was suggesting that we take advantage of what is existing. What do we have right now? We have the council, we have individual entities, interested parties in each of the sectors. I was suggesting that perhaps we get from this meeting, we say there should be a formal forum formal forum to coordinate research, formal forum in the office of the council, which will at least on a monthly basis or a quarterly basis meet to review those aspects of research, including this one.
Thank you, Honorable Minister. Having the bill passed is at the tail end of the process. One of the setbacks, especially from what we have learned in the past 2 days, is the perception of the public. So we need to either develop or, if available, disseminate baseline information, both in terms of qualitative and quantitative information to justify the need for the law, and then create massive public awareness on the need of this law, in addition to disseminating the mitigation of uncertainties that surround having such a law in place. So with that, we'll be able to arrest the fears of many people who may not be as aware as those in this meeting, to be able to support the move to assent to this bill or to even finalize it. Thank you so much.
Indeed, I think I told you that I participated in drafting the first text, 2001. And then in 2012, there was a group which asked me to help them review for the purpose of opposing the document. And up to now, so that was actually going to be my final question. How do we deal with the constraints which have really put a lot of friction on the road to this previous to JERA, that it has expired, it has died at infancy? The communication, being cognizant of the social media, we cannot ignore. I wish we could also regulate social media, but that's not within my docket. But social media, what? The infodemic, there are some people who make a business of creating scary messages. Now, I used to question myself, why would someone sit and concoct a video which looks, somebody somewhere pretends they're a professor at Harvard, and they make a very believable. I realize I've learned that it's a business. Because once you have that, you get so many hits And once you have a lot of followers and your videos, that's money. That's a business in social media. If I have Facebook and I put there things which so many people come to view, and or my Twitter is forwarded so many times, Twitter pays me. YouTube pays me if people play my videos a lot. Facebook pays me. So it is an investment, but in that process, it generates a terrible, terrible negative effect on very essential areas of life. Now we are trying to develop this law. It can be killed at infancy by an overbearing negative publicity. What strategies do we need to have in place to make sure that we'll be successful at this extremely essential and necessary law? Yes.
Thank you, Honorable Minister. I would suggest that to move this law forward, we would have education, empowerment, and engagement of all stakeholders to make sure that they are aware, first of all, of the available genetically engineered products, their benefits, and how they start to benefit from them. And then we should also, as the regulators, demonstrate our ability to effectively regulate, and so that we can create confidence in the public that the harms of these genetically engineered products can be managed, so that we are able to do an appropriate risk benefit analysis and give the right regulatory decisions. Thank you.
We have NDA here?
Yes.
You're representing NDA? NDA.
I am the one representing NDA.
You're representing NDA, yes. Good. You remember when COVID had just come?
Yes, ma'am.
And we had someone who has published some fictitious medicine on social media, saying I can cure. And NDA came in to deal with this person. And NDA turned out on the wrong side of the public. You are trying to do what you're supposed to do to protect the public, but the public ends up supporting the wrong thing. These are what we have to deal with. And I think the regulators, we also need to innovate in now how we are going to deal with it. I felt sorry. You know, you're trying to protect the same public, but the same public sees you as being in the wrong. These are the things that we need to innovate and deal with. And it's not unique to Uganda. It's everywhere. It's everywhere. So let me have one. It seems there are some people in the public who want to say something. I mean, in the audience beyond. You had a comment, then you comment, then we'll go to the audience. They want to make a contribution. How do we handle this?
Yeah, thank you, Honorable Minister. I remember in one of the presentations here, somebody from the media mentioned that the media is not our enemy, but we seem to take the media as our enemy. When this information comes out, we the scientists take the media as an enemy. So I think we need to look at the media, who are the key people that we can work with within the media, identify those that we can work with, and this can help us push this agenda. I remember this morning or early afternoon, Dr. Ebuku did mention something about pushing out this information. Who are the people? We need to identify the people that we are supposed to work with. The politicians are critical in having this information reach out to the masses, because as you said, you have videos which are made. Somebody makes a video and says a pig is growing on somebody's body, And this information moves so fast. So if we have politicians on our side, these really interact with the masses out there. I believe we need to identify those champions whom we can work with. So as the way forward, we really need to identify people, key stakeholders that we can that can help us push this agenda. Thank you.
Thank you, Honorable Minister. I'm of the view that we engage Ministry of Local Government, the district local governments and the lower units because they are closer to the people at the grassroots in terms of information dissemination, but also feedback. Thank you.
Please go ahead.
Thank you very much, Honorable Minister. Public engagement is very important and very crucial. But my view, the strategy should be that, one, we need to have the official government mouthpiece on issues of genetic engineering, biosafety, to counter what this misinformation from the public and from all sorts of sources will be churning out. They respond to that, and the public should also know where to go if they want the official government position. But as a matter of strategy, this should go on concurrently with the other process we had talked of gathering the views to put in the new bill, so that the other process doesn't delay because we don't have unlimited time. Then after we have got this official communication, this could even be actually a subcommittee of this committee we have talked about, but which is also gives what is regarded authentic information from government. Then once they have decided this is what we want to share with the Ugandan public, then it can be shared different communication outlets, the media, the local governments and the like, but should go on concurrently with the drafting of the new bill. Then they could even go on informing the public where the drafting has reached. Go to them, this is where we are. Is there anything you think we should have included, which we haven't yet put? There, you disarm them so that if you have already asked them to give their views and they haven't given, then they cannot oppose the outcome at the end. Thank you.
Okay, thank you. Let me go to the audience. The gentleman and the lady, I am too far to read your name. So please introduce yourself before you speak. If there is a microphone. Do we have another microphone which can be mobile? I want to close this in the next few minutes so we can close the. It's off.
Thank you very much, Honorable Minister. Sylvester Dixon Baguma is my name, Director of Narrow Blind Zadi in the Albertine Grabbing Zone. I have two issues. First of all, as a response on how should we go about it to make sure that we do not get these negative forces. First of all, if there is a gap, then you're allowing people to fill the gap with anything. So what's important is to make sure there's no gap. And making sure that there's no gap, then we require to have evidence-based communication, communicating facts. Whether we are going to sensitize or to engage the public, we must have the facts, evidence. And so as a STI, we need to develop a communication strategy. Who communicates what, when? And I'm glad to hear that we need to have an authority in providing the right information. If someone wants some information, should know where to get it from instead of depending on hearsay. Now, when I came here, I don't want to go back the way I came. I want to go back a better person. And therefore I want to be educated a little bit. I don't know who will. Maybe the ED, Uganda National Council of Science and Technology, maybe. I started working during the reign of His Excellency, the President of this country, Yoweri Kaguta Museveni, immediately when he had just taken over government. And one thing that the government did to champion science and technology was to create the National Council of Science and Technology. The National Council of Science and Technology went ahead to develop the act, even if it's very old, but that's the law. And then we got recently the Ministry of Science, Technology and Innovation, and now some changes. But we still have that law there. What is important, I think, for us is to revisit, to review that law in line of the current situation, the emerging issues, so that it can be all embracing. And then, of course, all the other MDS, they kind of, that's the overarching umbrella. All the other MDS, they have to borrow from there and now improve their own. We may not run away from having separate laws, because this will help in facilitating not only you, the Honorable Minister, but all of us to know when I'm talking about seed, what do I pick first? And then we don't have to have a law which is very huge, all encompassing. No, you have a seed issue, you know what to get. There might be only 3, 4, 5 paragraphs. No big deal. But to make sure that we have a good assembly, good repository of all these improved laws, including their own regulations. To me, I think that would be the right thing. Instead of coming up with a completely different law altogether, reviewing the UNC Steel law might be the right way to go, and the related regulations. I need to be educated, Honorable Minister.
Thank you. The lady next to you.
Thank you, Honorable Minister. My name is Barbara Zawede Mugwanya, Director of Research, Mukono Zadi. My submission is not very different from Dr. Dixon's. I think for this bill or the law we are planning, we need a clear communication strategy with the clear objectives of why we are communicating, to who we are communicating, at what time, what is the message to a particular audience, and how do you go there? Dr. Hafashimana said there has to be one mouthpiece, but sometimes it doesn't work like that. It's about getting the right person to deliver the message to the right people. I submit.
Thank you. We will have 3 more. There's 1, 2 behind there, and 1 here. There's some this way that I didn't see. Okay, I'll be fair, but please make your submission brief to the point so that we don't stay here overnight. So let me have those 3, then I'll take some this side. Let them finish, then I'll give you an opportunity to input.
Thank you so much, Honorable Minister and the House at large. My name is Paul Okwalinga. I work as a technical advisor for Biosafety Biosecurity at the National Health Laboratory Services, where we currently conduct capacity development for the Biosafety Biosecurity in the country. I want to weigh into and provide information. Mine is information, really. When I look at the law that we are presenting, the bill that we are, the JERA, and the previous that we had presented, when you introduce the word biosafety, the audience since morning has been asking for biosecurity because the two can never be separated. There's a marriage between biosafety and biosecurity. They are complementary and you cannot separate the two. Now, if you introduce biotechnology, then you are going to have to find a way of incorporating the 3: biotechnology, biosafety, biosecurity. So like the previous speaker who has just completed speaking said that if there is a way to find how to shorten, Let's focus on biotechnology, for example. And then we have a law passed on it. Let's have biosafety, biosecurity, which is actually globally now being implemented under the One Health approach as biorisk management. And as I speak to you right now, there's an ongoing effort in Africa to develop biosafety regulatory frameworks all over. This is headed by CDC Africa. And this is information that I needed to provide even as we think of how to move forward. Thank you.
Thank you. Next.
Thank you, Honorable Minister. My name is Emmanuel Ekulu. I am from the African Union, but particularly I'm the Executive Director of the African Forum for Ethics and Governance of Emerging Technologies. This evening, I'm actually very happy to hear from you, Honorable Minister, that I think the policy that Uganda has on science and technology innovation expired. Because the African Union, we have actually a whole framework. It's called Science, Technology, and Innovation Strategy for Africa, STISA. This was actually developed by all African countries to guide them on how science, technology, and innovation should be conducted in every country. Indeed, actually, Uganda does not have a legal framework, a broad legal framework on science, technology, and innovation. Because if you go to most of these countries and you want to know what they do in terms of science, technology, innovation, or in terms of a legal document, they have it. We keep wondering actually why Uganda developed a policy, but did not go ahead to put in place a legal framework on science, technology, and innovation, something that guides, a country on a legal basis. So we are here to help African countries to actually align their frameworks, policies, legal frameworks in line with the one of the African Union, because it's there. It just needs to be, aligned. Countries need to align theirs with the one of the African Union, because it's a 10-year document which is actually going to expire in 2024. But we want really to see Uganda really develop its own kind of policy, because the other one has expired, but also to put in place a legal framework. But I also bring good news to you is that the African Union has already gone ahead to put in place continental centers of excellence. This evening you heard that South Africa actually got the opportunity to host the Science, Technology, and Innovation Center of Excellence across the continent. They will be the one leading this. But for the East African region, we don't have this. And it's actually one of the reasons why I'm here, to identify a country that will host a continental center of excellence. And so I discussed this.
You have found it. Your search should stop. Q, I promise this side, 3. So you give those 3 opportunity.
Thank you so much. My name is Diana. I'm a student of biotechnology at Makerere University. For me, I think that for everything that we need to develop, we need continuity. Like when people have retired, we need young people to understand where we have come from and to know what we are going through now, so that they'll be able to guide those that will come later. So for me, I think that as we do all this, we need to include young people so that they see what is going on, so that they'll be able to shape the future after here.
There seems to be a lot of clapping. How many of us are below 35? I think we are including the young people. Okay.
Thank you very much.
Yeah, yes, you can shout it louder. If you watched me on TV yesterday, I was talking about liberating the science from elite capture. So let's continue.
Thank you very much, Honorable Minister. I'm very grateful actually to hear the process you are doing of making a law. I'm called Dr. Chana Francis. I'm the Vice Chancellor of Kentim University. First of all, I know very well that the element of research, as you have mentioned, we also need to include those who do research on anthropology, because anthropology is also good research people who could have a good working relationship with biosafety. So this is my humble request. Where do you include the anthropology? Thank you.
I'm Deborah Wendiro, retired. My issue is I've been hearing from several corners saying, look at it this way, look at it that way. Now, I'm wondering whether we cannot get an interministerial approach, because science operates in a system, an innovation systems framework. I had expected that, being that we are here at the cream of the nation in science and technology, really we would have other ministers also coming. Even in industrialization, people have been talking industry, but I don't see any minister from industry coming. So way forward for me is to convene an interministerial meeting. I don't know who does that, where these ministers will be the ones now to direct their ministries, because I don't know whether you would be able to go and influence policy in another ministry. Yet if they had come and known how important biotechnology, biosafety is and biosecurity, really we wouldn't be saying, how do we handle this? How do we? Biotechnology affects me as a woman, me as a mother, me in the ministry, in health, me in environment, me everywhere. So I'm suggesting that we need, from now, I mean beginning maybe today, the ministers to come together, and if they need us to come back, we'll come back and tell them how important it is for me in water, for me in food security, for me in the environment, and in every other aspect. That's what I believe. Thank you very much.
All right, I think we have only one more and we close the session because we need to go home. Just one more.
Yeah, thank you very much, Honorable Minister and the chair of this session. By the names Odoi Juventine Boaz. I work with NARO, and I'm also completing my PhD in plant breeding and biotechnology from Makerere University. My point here is looking at the aspect since we began this meeting on Tuesday. There was a session where there were recommendations which were made. One of them was to establish a joint forum to take forward all the recommendations that have been made here, so that to look into the opportunities on how to make maybe a cabinet paper or looking at a review of the current or existing policies and laws governing biosafety, biosecurity. I look at also another aspect. Sometimes we talk to ourselves. We are the technocrats. We are the implementers. And ever since from Monday, I've been trying to look, we have left mostly one wing, the legislators, our members of parliament. They are the law enactors. And you will find that somebody who had not so much embraced biosafety, biosecurity, from Monday up to now, the gospel has reached that someone can appreciate. So I feel like if select committee that handles this area had been invited, at least the chair of that committee, by the end of this workshop today, at least they would have appreciated the need to fast track the existing policy, what we have, what has not yet been completed. My last submission is on the we said that we have a number of jungled policies which are just like not very specific in different ministries and departments. This is very true. But then like now when it comes to enabling instrument, the legislation, it may appear at the level of the policy. But then when it comes to the regulation, you may find it is also adamant. We were recently having a workshop that was in Imperial Hotel, and we were talking about looking at agrobiodiversity, including in agricultural systems of Uganda, to diversify farm output. But we realized that we looked at the national agricultural policy, but we found that agrobiodiversity, like agroforestry, was very, very adamant. It was just mentioned to include one of the agrobiodiversities. But when you come inside to the details, you find it is lacking. And that is what is happening with most of our laws here in Uganda. A statement is thrown, but when to go to implementation or to practicalization of that policy, so that implementers can pick and begin using in order to bring it to practice, you find it is adamant. Thank you.
Okay, thank you very much. I know that we all have a lot to contribute, and I want to thank our panelists. My purpose, for chairing this session was to make sure that we leave this room with action, something that we are acting on. Otherwise, we come here, you know, scientists, we get entertained by science. Somebody presents and you're like, oh yeah, they are doing that. That's a wonderful paper. That's a beautiful paper, but so what? Next year again, you just come back and again get entertained by science. So we really want to move science in the country. And for us to move science, an event like this that has brought together so many people, I was presented with a summary of key things that had been captured up to lunch. And I saw a lot of statements that are not definitive actions. Maybe in the communique, there is something about definitive actions, but we need, from this session, we have made several decisions. One is that we need a law that governs science, or at the least, we need to review the policy to make sure the policy is up to date and all-encompassing and enabling in the current. I've been receiving lots of documents, you know, we need a policy on this, a policy on this, and it's like I'm going to have 100 policies in 1 year. But if we can find the good anchor documents which guide us, then we can operationalize. The second issue, so that first issue is we need to make sure that this moves forward. And I'll request our legal advisor to help us to champion this so that we can begin to draft. And within the next 2 weeks, we should have a meeting to begin to talk specifically. It's difficult to make a decision in such a big group, but in the next 2 weeks, we should have a meeting so that we can give proper direction to this. And you can also, you'll have had time to do some more research so that you can guide us. So we'll bring together a meeting of these regulators so that we can begin this process. The second thing was, what do we do with the JERA? My suggestion is in this meeting, we have had presentations. We have had presentations concerning these key areas. Let's pick those as the beginning point of what we add into the law that we had. Then we will, based on that draft document, to do consultation so that at least let's try by the middle of this financial year to make sure that we have something that we can take to cabinet. So that the president has been also asking for us to take back this law. So let's make sure that we begin to look, the people who have presented give this document. The executive secretary here will continue to coordinate us, so that brings us together, and we incorporate that, then we will call another consultation meeting to make sure that, we hold a consultation meeting with the key MDS, and the target time for us to be able to take a draft of the principles. Since we already have the big part of the body of that document already done, just let's have the new things incorporated, and we give ourselves at least latest by mid of this year. I know it takes quite a bit of time to get people to consult, to consent. The decision makers they have not been here. They are not part of the information, and yet we have to move through the hierarchy. And then the third decision we have made is communication. I think we discussed during the preparation of this meeting that science communication needs to come to the forefront. It is usually interesting when scientists are talking to each other about communication. They all know what to do, but they don't do it. And when we speak, they don't understand. Sometimes I've spoken, I think that I'm really communicating, and people are like, but you are complicated. So we needed to come up with a framework, and my ministry will work to bring together stakeholders to make sure that we build a strong platform for communicating science. When I said I've received delegations, actually, when I became minister, I received delegations on both sides. Those against GERA and those for GERA. They were among the first delegations I received. They had to make sure, all of them had to make sure I'm well briefed from their side. We don't want to get into that. We want to go ahead with the communication, because communication is critical. Government needs, as we accept that we need to up our communication. So we accept that. The other issues that people have been talking about, our colleague from the AU, Emmanuel, don't leave the country before we have a meeting, so that we can give you a home and an address here. Your search should stop, because the search should stop. Here we have taken on science like we've taken the bull by the horns, and we are ready to move forward. I think the rest were recommendations. The interministerial team, we were waiting to have 2 meetings, this meeting plus the technical working group for ITDT, which is Innovation, Technology Development and Transfer Program under NDP3. And then I'm going to write to the Prime Minister to help us to convene the interministerial committee meeting for us to deliberate. And this should be one of the issues that we are going to be discussing. And this should happen before, I can't say July, because that will be too quick, but should happen within the first half of August. July is a little tricky, but in the first week of August, the committee, the ministers who are concerned, we should have a meeting and hold a meeting, because we will have some ministers need to meet when you have something. If you meet them to ask them just, the meeting will happen, but you leave the meeting with nothing. So we need to have something. We need the technical people from the different sectors to consolidate what we are giving our policymakers, so that by the time the meeting is taking place, there are briefs to give to them. So that's why we need a bit of that time. So ladies and gentlemen, and the focal person at STI, apart from UNCST, is a gentleman called Abraham. Is he here? Abraham, are you here? National Planning, he comes from National Planning Authority, but he's our head team lead for policy at STI. He's very, very active and very good at what he does. So we will put him as part of the team to make sure that things move together with UNCST. I know there are people putting up their hands, but I'm tired of standing. So I don't think I want to take any more answers. Are you going to die if you don't say it? Okay, give him, just him, no other, please, no other. No, I am.
Honorable Minister, thank you. There is an urgency to know that every day, 8 million pieces of plastics enter into this country. And Jibril, as a medical clinician who did diploma in clinical medicine, now running higher diploma in nephrology, a graduate with a certificate on green industry.
It's okay, tell us what you want to ask. What is it you want to say?
Biochemical changes are ongoing into our bodies. Particulate particles from plastics are already with us. What can we do, or what are we doing? As Greening Uganda campaign, we are cycling plastics into pavers, into culverts, into roofing tiles. What do I mean here? What should we take home?
I'm going to charge you for that commercial. I think you've made your point. Nema is here. I think they don't sleep because of plastics. Every day they are worried about plastics and all it's. So thank you very much for bringing to light the challenge of plastics. Yes.
My name is Professor Mutumba. I'm requesting that we get a blueprint of the current jail, and we work from there, we fill in the gaps. Normally they say that you always begin from the known to the unknown. Instead of going to the drawing board, let's work to improve the current document.
Exactly.
Can we get it on our emails?
I think the executive secretary has mentioned that he's going to create an emailing list. The process of reviewing the JERA as it was to respond had started. Unfortunately, it didn't move fast. And not only did it expire, the timeline, as she used the legal word, called what? It elapsed. Not only did it elapse, so we have to take it back to Parliament, but it has also been overtaken by events, because now JERA was really strictly for plants, but now we have we have things to do with health, not as a consequence of them being in the environment, but we want to use biotechnology to treat disease. As they have presented, there are medical applications which need to be incorporated. We are also going into production of other products, not just plants. We want to produce vaccines. We want to produce, we want to manipulate microorganisms. So there is a lot that needs to be done in animals. So we want now to just, what do we have? What do we need to add? And that's what I was saying, that's exactly what we're going to do. So ladies and gentlemen, thank you for listening to me. Let me just consult a bit. Now I'm going to go home happy that I have 4 action points, and we're going to put this in a document which we are going to make sure that we act, and we've set some timelines. So I know that next year when we come, there will be a point of accountability. In the last meeting, we resolved as this, and we have made this progress so that we know that we are moving forward. Thank you very much, the panelists. Maybe let me give each of you 15 seconds for parting words from the panel. 15 seconds. Where is the clock? Your parting words. Let me start with my chairman, Dr. Kware. 15 seconds.
Honorable Minister, I want to thank you very much for bringing us together. I would like to thank the participants, and I would like to thank everybody. And I would like to say that research is a is a moving subject, so we must do it quick. We must do it today, not tomorrow.
Thank you.
Thank you very much, Honorable Minister. My last word is just to correct some, I think I was misunderstood. When I said we need one official position on GMOs, I didn't say only one person should communicate. Once the message is clear, then you can even give it to a Mutembe on the street, and as long as we know that this is the official government position.
Thank
so that's
you.
what I meant. Thank you.
Thank you, Naro.
Mine is to thank you for the purpose in this meeting from business as usual. Number two, the issue of public perception has been there for long. I think for 20 years we have learned the lessons and we know the concerns. There will be need for strategic plan and analysis around each of those areas so that every concern is addressed. Thank you.
Thank you.
Thank you, Honorable Minister. As my parting words are to thank you, but also to pray that going forward, you will be our point of whipping the other MDAs to participate in the drafting, because we are facing this challenge because people acted like ostriches, and now they realize that they need the law and they are not there. So it's taken us back a few steps backwards. So I think, I pray that going forward, all the interested parties will show a little more interest to have this bill passed as soon as possible. Thank you.
Yes, please.
Thank you, Honorable Minister. On behalf of NEMA, I wish to thank you for convening this meeting. As NEMA, this is a learning experience to help us in updating our laws and in implementing mitigation measures related to biotechnology development. I wish to say that environment management is an individual responsibility. Thank you so much.
Thank you.
Thank you, Honorable Minister. And on behalf of MAIF, we're really excited that there is hope at the end of the tunnel, because there are so many technologies that are still on the shelves. But at the end of this meeting, there is hope that soon we shall have the enabling environment. Thank you.
Thank you. NDA last.
Thank you, Honorable Minister. I would like to thank you and the UNCST for this meeting. It has really been a great forum to share ideas. And as key stakeholders in gene therapy for human health, the National Drug Authority is committed to ensuring that we'll have our input in the revised JERA bill so that this can move along fine. And we're also committed to supporting the researchers out there using the available regulatory frameworks to support research in genetically engineered drugs. Thank you.
Thank you very much for being very good panelists, for keeping the time. Even the 15 seconds, most of you have kept it, and for the good deliberations. So I'll step down as the chair of the panel of this session. Thank you.
Thank you, Honorable Minister, for chairing this session and of course, giving the strategic direction on how to go forward. And I would like to thank the panelists. I think the approach appeared like I lured you. And of course, that first session went. And of course, we had the second session, but I think the contributions were really very, very useful. So probably at this juncture now, I would request you to take your seats. I don't know whether there will be a photograph session. So probably you can probably stand somewhere there, and then we have we take a photograph of all panelists. Okay. Yeah. Honorable Minister, you have not yet relaxed, and we are giving you more work. We want you to join the panelists for a photo session. Sorry for that. Okay. Now we are going to move to the last session, which is actually the closing session. But before we get to that session, I would like to thank all the participants that we invited and managed to attend either physically or virtually. Thanks a lot. And of course, right from day 1, day 2, and day 3, the attendance has been great. The contributions have been great. And we have really learned a lot from each other. And of course, the most important thing is that we now have a very clear direction on how to move forward in relation to the JERA bill. I remember there used to be a lot of questions on where we are in relation to how we are moving on with the JERA bill, and the directions that we have got from this meeting will be very useful. Now, at this juncture, I would like to thank the organizers of this forum, which has been, I can't compare, it's my first National Biosafety Forum, but from the comments that I've heard, it's that it has been quite successful. So I would like, first of all, to thank the Honorable Minister, and first of all, for the contribution during the organization. I think we debated a lot. There was like a 360-degree turn, and we reoriented ourselves, and we managed to come up with a very useful program. So I thank the Honorable Minister, both for the financial support, but also for technical support in the organization of this forum. The STIOP was actually the greatest donor. And of course, I would also like to thank our partners, the National Agricultural Research Organization, CIFOD, Program for Biosafety Systems, the Science Granting Council's initiative, and Agricultural AATF, and quite a host of funders for this forum. So right now, I would first of all like to introduce to you very briefly the members of the organizing committee. It was chaired by Dr. Yona Baguma, the Deputy Director General, National Agricultural Research Organization. But I think due to some commitments, he has not been in position to be with us till the end. But other members of the organizing committee, if you're around, we could come briefly forward. So members of the organizing committee. Including colleagues from STIOP, Brenda. I wish there could be a portable mic and maybe just say hello very quickly. A portable. Thank you. So you can just say your name, your organization, probably this time like 10 seconds, I hope. Yeah.
Yes, good evening, colleagues. David Hafashimana is my name. I'm Vice Chairman, National Biosafety Committee. I also bring apologies from the chairman, Dr. Yona Baguma, who is also the chairman of the National Biosafety Committee. He has also been the chairman of the organizing committee. I was deputizing him. So I'm currently retired from government service. I used to work with NARO, before that with the Ministry of Environment. Thank you.
Good evening. I'm Irene Mwesigwa from National Drug Authority.
Good evening, everyone. I'm Robin Alwato, Uganda National Council for Science and Technology. Good evening, everyone. My name is Laura Nero from National Council for Science and Technology.
Good evening, ladies and gentlemen. I'm Isaac Kungu, Executive Director, Science Foundation for Livelihoods and Development, CIFOD in short.
All protocol observed. Good evening, everyone. Beth Mutumba, UNCST, Directorate for Research and Quality Assurance.
Good evening, ladies and gentlemen. My name is Helen Opolot, Uganda National Council for Science and Technology.
Good evening, Honorable Minister, ladies and gentlemen. Deborah Kasule from Corporate Affairs and International Collaboration Division.
Good evening to us all. My name is Brenda Nakazibwe, Science, Technology and Innovation, the Pathogen Economy.
Good evening, ladies and gentlemen. I'm Mary Nantongo from Science, Technology and Innovation, Pathogen Economy.
All protocol observed. My name is Barbara Zawede Mugwanya. I'm the Director of Research in NARO at Mukono Zadi, and I'm also the Chairperson of the Institutional Biosafety Committee of NARO.
Good evening, ladies and gentlemen. Mwesigwa Collins, Uganda National Council for Science and Technology.
Good evening, members. Kwehangana Musa, in charge of biosafety at the Uganda National Council for Science and Technology.
Thanks a lot. And those are the members of the organizing committee. And of course, we also have the National Biosafety Committee. So probably, I don't know if most of the members are around. How many are there? There's 1, 2. Okay. You could again introduce yourself very briefly. Thank you. You have already finished, and then the other one.
Good evening, everyone. My name is Kenneth Katonyisa. I'm the Chief Executive Officer of Uganda National Farmers Federation and a member of the National Biosafety Committee. Thank you.
All protocol observed. Good evening, members. My name is Alex Bambona, Assistant Commissioner of Food and National Security and member of the National Biosafety Committee.
Thank you. Any other member? Or that's, oh, there's one. Yeah, I think you have already introduced yourself. Okay, thanks a lot. So now I would like to take the opportunity to request the Honorable Minister, I hope you have relaxed, now to give us the closing speech. Thank you.
I hope you're not bored looking at my afro, because I've been here and then I'm back here. I'll try to be brief. First, I recall somebody talked about Parliament. I think we invited parliamentarians. Is there anybody from Parliament here? Yes. So we have, we work very closely with Parliament. So I also wanted to make sure that my protocol is right. So the honorable members of Parliament, head of government agencies present, the science and technology regulators represented here, distinguished bioscientists and bioinnovators, members of the private sector, journalists and science communicators, all the participants coming from within the country and from outside the country, ladies and gentlemen, I am very pleased to have been with you these past 3 days. Apologies because I usually sit in the meetings from beginning to end. However, there has been a moving activity that I was also assigned to lead. In Ebukedi, I had to go and set it off before I could dash back. So I apologize that I've not been in here because this is a very important meeting. But I am glad to be here to officiate at the close of the 5th Annual National Biosafety Forum, which began 2 days ago under the theme, Science, Technology, and Innovation, and Regulation for Sustainable Industrialization of Uganda. Thank you all for actively participating in this conference. The goal of this forum was to celebrate the successes in biosafety stewardship and explore how this is related now to science, technology, and innovation, and regulation for sustainable industrialization, which is currently a priority in the country. Specifically, This forum intended to provide a platform for regulatory agencies to share experiences and strategies for science, technology, and innovation. Given the new dispensation that the country is strongly poised towards industrialization. B, to link biosafety and biosecurity to national development. C, to enhance the understanding of risk assessment considerations for emerging technologies and new uses of biotechnology. To facilitate the understanding of the regulatory landscape in Uganda for the last 25 years with an appreciation of the role of a robust regulatory framework in advancing or promoting gains made in biotechnologies and innovations over the years. I was informed of the objectives and what we were supposed to have at the end. At the end of this forum, we're supposed to have an increased appreciation of the new direction of science, technology, and innovation in Uganda. Ladies and gentlemen, I hope that you are somehow started on the journey of conversion from where you were to the new dispensation of science, technology, and innovation. If there are some doubters, I am going to invite myself to your organization and speak to you a little more. We also wanted to have enhanced coordination among key regulatory agencies, to have an increased appreciation of the biosafety and biotechnology among policy leaders and regulators. I hope that we have achieved that. I hope that we have started to speak to one another more openly and seeing ourselves as complementers and not competitors of each other. To have an increased appreciation of Uganda's capacity to regulate genetic engineering technologies. I don't know whether we have had that, but we have seen that the technologies are coming, whether or not we have the law. And it is our responsibility to make sure that we have the right regulatory framework. A forum like this renews our commitment and collective efforts to have wise and regulated use of science, technology and innovation, which can boost productivity, raise our national income levels, and improve the quality of life of Ugandans. The NRM government is committed to using STI to transform the country, which is popularly referred to as science-led socioeconomic transformation. Using the different STI interventions, digitalization, artificial intelligence, modern biotechnology, agricultural technologies, space technologies, ICT, renewable energies, and many more to achieve the goal of Vision 2040 and the objective of the National Development Plan 3. We need to congratulate UNCST for having achieved, I think this is supposed to be the Silver Jubilee, 25 years of biosafety and biotechnology stewardship. I think let's give them. We know that many have come and they have gone, and the team there at UNCST, you are standing on the shoulders of those who went before you, but we thank you for continuing to carry the flag of regulation and ensuring that Ugandans are safe as they utilize science. And over the past 2 days, we have also talked about the recent advances in risk assessment of emerging genetic engineering technologies. We have also seen some displayed in the exhibition, biosafety, biomedical research, applications, public engagement in biosafety regulation, and trends with Uganda's biosafety regulatory exchange here, regulatory landscape in our last session here. Benchmarking on other nations that have made it to the middle income status through prioritizing science and technology, Uganda too, through the visionary and deliberate leadership of our President, His Excellency Yoweri Kaguta Museveni, has made STI a national priority through the NDP 3. Science and technology is looked at as the anchor pin for NDP 3. Many of you may wonder why we seem to be in haste. Is because if we do not push science and technology, we may not achieve all the objectives of the rest of the programs under NDP 3. The government of Uganda is committed to adoption of a multi-sectoral approach to science, technology, and innovation development. And ladies and gentlemen, allow me to pause here from reading my speech and address you. We are too few, we are too small to be competing. We are too few, we are too small to be competing with one another. We should instead adopt an aggressive complementary approach because we can achieve a lot together. STI does not compete with anybody. We are here to augment the work of everybody. We are here to augment the work of the research institutions. We are here to augment the work of other the regulatory institutions. We are here to augment the work of many MDAs who have research functions under them. We are here to make sure that that function is augmented. We are here to make sure that that is facilitated, it is accelerated, and we can achieve a lot together. So let's put aside the cannibalistic competition, which I observed that, you know, each institution believes that you move forward if the other one doesn't move forward. Why can't we all move forward together? Let's all move forward together. We have a small nation which is badly in need of us working together. And I would like to invite you for us to work together without, I want to see narrow. You have grown, MAIF has grown in their work, Ministry of Health has grown, UNRO is growing in their work, NDA is expanding their mandate so that we have a common agenda so that we can really, really push the nation forward. The government of Uganda is committed to this approach, multi-sectoral. It is also committed to support the prioritized STI industrial value chains, which include pathogen economy, mobility, industry 4.0 plus, export, value-added export promotion, import substitution, infrastructure innovations, and the human capital development, especially for science. The value chains align to the parish development model, aspirations for national development and transformation. We shall support the need for an enabling regulatory framework in terms of policies and laws to advance the same. And the committees and all those that have been key players, I invite you to buckle your belts because the speed is going to be quite fast. The new STI dispensation under the Office of the President through the value chain aims at reducing import dependency through product development and commercialization. As leaders, we therefore need to rally all the relevant stakeholders to build confidence and trust in our homegrown solutions and products because they are of quality, they have been nationally and internationally certified, and have followed through the process of science. In other words, we are not going to entertain shortcuts. Just because it is made in Uganda, it does not have to be of poor quality. Actually, we want, because it is made in Uganda, it should be trusted because it is of good quality. Remember, we are making Uganda the best. I would like to conclude by thanking you for participating in this Biosafety Forum 2022 and thanking the Uganda National Council of Science and Technology for leading this, organizing this forum, along with the partners who include the ministry, that's Science, Technology and Innovation at Office of the President, which is we fondly call STIOP, the Program for Biosafety Systems, PBS, AUDA, NEPAD, Africa Agriculture Technology Foundation, AATF, the Open Forum on Agricultural Biotechnology in Africa, OFAB, Uganda Virus Research Institute, UVRI, Infectious Diseases Institute, IDI, National Agriculture Research Organization, NARO, Science Granting Councils Initiative, SGCI, private sector, universities, and anybody that I could have mentioned. Allow me to once again amplify the vision for science, technology, and innovation in the country, which is reflected in our slogan, making Uganda the best. Could you kindly say that with me? Making Uganda the best. It doesn't have faith and, okay, 1, 2, 3, making Uganda the best. That is the air I breathe. That's the air the team at STI breathes, and we hope that we will infect you and you'll move with us. With these few remarks, I now officially close the 5th Annual National Biosafety Forum 2022 on the 23rd day of June 2022 at Kampala, for God and my country. Thank you.